Okay, good morning all. So shall we probably start? So
Yes, we can. We are live on YouTube and we are also live in Zoom. So please go ahead, Dr. Pankaj.
yeah, so it gives me immense pleasure to introduce our director, Professor Santanu Chaudhary. He is director at IIT Jodhpur, and we uh, we look forward to his opening remarks for the international conference on data science and biology. So this is the first time we are organizing this conference in our organizer Institute of Bioinformatics. So I welcome Professor Chaudhary, and I really very thankful that he spared some time to give some opening remarks for this conference. So thank you so much, Professor Chaudhary, and now stage is yours.
Good morning, all the delegates who have joined for this web-based conference on data science and biosciences. So it's my greetings to all of you. And on behalf of IIT Jodhpur, I welcome you all to this web conference. I think the topic which has been chosen is very relevant and extremely important. Data science actually has the potential to revolutionize healthcare and can respond to the increasing volume and complexity of biomedical and bioinformatics data. Data analytics is a collection of tools and techniques which allows us to discover patterns, discover hidden variations, hidden structure in the data, and that being applied to the data from biological sources had a huge potential in terms of applications. Data science actually brings in a significant cost advantage, enhances performance of decision making, and creates new processes and products to meet the need of the end users and stakeholders. Say, in the space of biology, from genomics to clinical records, from imaging to mobile health and personalized medicine, the data volume in biomedical research presents a huge challenge to computer scientists who are looking into techniques of data science. The way that researchers in the data science domain is playing an important role in the space of biology is really significant. And in fact, in a conference where the issues of data science and biology is being discussed together can really be significant in terms of future steps. Because data science is expected to play a very critical role in various aspects of computational biology, such as biomolecular computation, computational photonics, medical imaging, scientific community, structural biology, bioinformatics, and biocomputing. And database, because these also link to, from a computer scientist, that questions are that of databases and big databases, because the volume of data from biological sources are huge. And this really puts us to the domain of big data and the challenges of big data and big data processing. So the other important issue which comes in linked with data science in this context is that of scientific visualization. The data that we are collecting and data that we are analyzing and analyzing to discover pattern until and unless we can construct visual representation of that data, this whole acceptability of the results that data science is producing becomes always a question. See, the other very important thing is to be addressed with reference to the data science today is the majority of the techniques of data science is actually based on machine learning. And deep learning has actually leading to a variety of new possibilities in this domain of data science. But deep learning is in some sense a black box. And if we consider the traditional machine learning techniques and very simple classifier, say nearest neighbor classifier, then you can actually explain why the classification system or algorithm is given an answer for a query. But when it comes to a deep learning, it's actually that of learning a black box, okay? Because the learning system and what does this network learns, what does this network weights embed, that's not very clearly visible and understandable by a layman who is using big learning toolbox for analyzing data. I shouldn't say layman, even scientists across different disciplines, they're using deep learning toolboxes to analyze data, including biological data. And the question which comes up is the result which these deep learning systems are producing, are they reliable? Are they explainable? Because until and unless there is an explanation, you cannot really use the outcome of these kind of a black box based systems for applications like healthcare. Because no regulatory authority would accept a black box driven decision. Now, the other very important thing is, therefore, the new Vista, which is actually now a very established area in machine learning, is to build algorithms which will give me explainable learning. That this learning system should explain itself. And I think designing of such systems with specific reference to biological problems is actually an open area to explore. The other very important issue is that any of these machine learning systems or data science systems, when they give a decision, when it is used for an inferencing purposes, we know that there is always a probability of failure. Now, when we actually train a learning system, because these are very critical problems when we talk in terms of using learning and data science for domains like biology, when we train, we know that we are training on a known data set, and with the known data set, maybe a machine learning algorithm produces a result with say 98% accuracy. Fine, I know it's very high, so it can be used for unknown data. But now when you move to an unknown territory, that is new data set, therefore there exists at least 2% probability that decision will be wrong. But which decision is wrong? Okay, now that is the most critical thing. How do I assess that? See, in many cases, the deep learning algorithms, the results or uncertainty it talks about or gives in terms of classification likelihood is not true measure of the uncertainty. Because try to understand that you have trained a deep learning model or any of the machine learning models for a given distribution, and it's actually learning the characteristics of the distribution. The moment you come out of this trained data set, you are not sure and you do not know whether the distribution has changed with respect to which you are picking up the data and giving as input to the deep learning system for making a decision. So if the distribution has drifted substantially, then your result will be completely different. And even the likelihood score which the deep learning system is producing is not a valid likelihood score, because that likelihood was respect to a distribution And now you have picked up a sample from a different distribution altogether. Now, this becomes a very interesting and significant problem to actually have a true probabilistic estimate of the correctness of a decision or inference by a deep learning system. And truly speaking, this whole likelihood measure has to be derived from maybe all possible architectures of deep learning. Because when you actually train deep learning, the meta parameters are set experimentally. That means how many layers you're using, how many nodes in each layer you will be using. These are all meta parameters, and that you set up with respect to one particular architecture which we have found optimal for given result for a training data. But if you're talking about distribution distinct from training data, then the architecture itself has to be different. Now, if I have to estimate a likelihood of a decision, I should have an estimation method over all possible architectural variation of the network or the system that I'm using for making an inference. Now, these issues, why I'm bringing in here in this conference. Because in biological space, when we're using this kind of deep learning, they have implications and direct implications in terms of, say, healthcare, direct implication in terms of precision medicine, even in terms of phenomics, plant phenomics, as well as human phenomics, because human phenomics is also linked to precision medicine. In all these cases, the proper likelihood assessment is extremely important, where you can use data science results with sufficient reliability. So what we are talking about today is actually dependable data science. And dependable data science for biological systems is critical. In fact, my area of research is primarily medical imaging, has been medical imaging. And in the medical imaging, when I'm making a diagnostic decision, without a reliability of that decision, I cannot provide that decision in an automated system. So at the best, without that reliability estimate being available, my data science system based on medical imaging can simply augment the human decision-making process and can never replace a human decision-making process. In fact, that's also true for a variety of applications that we look at. And the other very important and interesting thing which is coming up with the deep learning being there is in the space of your computational synthetic biology. In fact, the generative models. So see, most of the cases data science really, which has been more popular, primarily works with a discriminative model, where we actually talk in terms of distinguishing between the things. Okay. Because given a distribution, whether an element belongs to the distribution or it doesn't belong to the distribution, or can we discover a set of distributions with respect to which we can discriminate the data which is available with us. But the next generation thing, where we want to synthesize new knowledge out of what we already have, there the generative models are most critical. And in the deep learning framework, very powerful generative techniques have been developed, and which actually, I think, being used in a big way in computational synthetic biology, and where the new interventions can be designed in terms of maybe in various applications, including healthcare. So data science is very exciting component. In fact, as a data scientist also, biology is also a very exciting domain to look at. But there are various problems in the data science itself, which also requires interventions from the biological scientists, because the whole modeling in terms of explainability, whole modeling in terms of reliability of the data science processes can possibly become much better with close collaboration between data scientists and domain experts, in this case, biological scientists. I think this conference would provide an opportunity for this dialogue, because such dialogues can only enable creation of new knowledge in these interdisciplinary areas. And today, the entire progress of science is primarily through interdisciplinary interaction. And I hope this conference would become a a regular feature and provide a forum for dialogue between data scientists and biology scientists. Thank you. If you have any questions, I can answer if there are opportunity for that.
So thank you so much, Professor Chaudhary. And it's really a very good insight towards what is the motivation of this conference that we have organized. So we, as you said, that we now we can say that the biology is not only for biologists, it's for also other fields. And so it's going to be truly interdisciplinary in nature, because we need to have a good and communication and good collaboration between different fields of science, biology, computer science, mathematics, and clinicians are also going to play a critical role in this direction. So with this, actually, I think if we have any questions, we will definitely love to take it. So if there are any questions, please ask from Professor Chaudhary.
Good morning, sir. I'm Anshu from MCSR InTech.
Yeah.
I just have one question. So you said that you deal with medical images. So how do you deal with the variability that comes along different patient data sets, because it's not generated in a controlled environment? So then the values that we receive from the clinical samples are pretty, as you said, the distribution of the data needs to be known. So what methods would you recommend there? Because data generated in lab is more organized versus data generated from clinic it has a huge variability per feature. How do you deal with that?
See, actually, let's look into this way that even if you look at standard deep learning techniques which are there today, now if you're using supervised learning, I require a label data, okay? And if the label data which is coming even from the clinic, if there are variations and it is not necessarily a parametric distribution, the whole advantage of the deep learning, it learns the distribution which may be some arbitrary distribution. So I have, even if I have data from a clinical samples, in fact, we have also done such kind of work. And if they have the inherent variability, my network can learn that variability. And we have to play with the architecture. We have to go through various modifications because the network is capturing possibly that distribution. But what is very critical is when you actually go to application, the distribution drifts. Now, there are now lots of algorithms and techniques which are being developed, which addresses explicitly this drift. And some of the techniques which are called, say, self-supervising learning, where you may not really have very detailed label samples, and semi-supervised learning, those are actually trying to do learning on a data set which may not be all labeled. And in fact, that is also very, in many cases, actually, even in COVID scenario, we have got large number of, say, X-ray images of lungs, which have not been properly labeled, okay? So therefore, when you deal with them, you have to use it with kind of a semi-supervised, self-supervised techniques. And addressing drift is something extremely important. The other very important thing which you are talking about is you will find that there are techniques which has developed, which are called probabilistic networks. Actually, you consider even each weight in a network as a distribution, okay? Not just a specific value. And these probabilistic networks, when they learn, they also give an estimate, because see, what is being modeled is that all possible distributions, if that is in some way can be at all modeled, but we can have an approximation. If I learn not just a single network, but try to learn over a probabilistic model of network. I don't know whether I'm able to explain that or not, that it is not a single network. You are actually learning over a set of networks, and each network can really represent the variations of the distributions. And then the decision that you are getting has got a likelihood measure, which is over a much more generalized distribution than what you have in your training data.
Oh, yeah. Okay. Okay. Oh, that really helps. So thank you so much.
Such networks are there. Actually, you should look at those algorithms.
Thank you, sir.
So are there any other questions, please?
Yeah, yes, I have a question. My name is Malik Yusuf. Actually, now it's 7:22 a.m. I was waking from 6 a.m. So,
Thank you, Malik.
and I am collaborating with Abishka. We have different projects. So my question is, and also it's related to my talk, most of the computational approaches we borrow from other fields, like statistics, machine learning, computer science, and we just take it and apply it in our bioinformatics field or in biology data. How do you see that we can invent something new that can use the biological knowledge and embed this information into a new machine learning algorithms? Not just to borrow stuff from other fields or doing stuff that other people doing in other data.
Okay, shall I respond?
Yes.
Yeah, actually, one point while I was making my introductory remarks, I said that we need to have what is called explainable models.
Yes.
And if we have to build such explainable models, the domain knowledge is extremely critical. So a biologist can possibly collaborate closely with a computer scientist to build an explainable machine learning model for a particular biological problem. This is something very important.
Yes.
And also, if you see today, there are also other kinds of techniques which are coming up. In fact, if you see the probabilistic reasoning models, which are being used in variety of diagnostic applications, where we talk about these with counterfactuals, with what if reasoning. Now, if I have to do a what if reasoning and what if modeling, that can never happen without the domain expert really participating in it. So I'm giving example of diagnostics because in diagnostics cases, there are different pathways for diagnostics. And that different pathways may not be directly learned in a kind of a black box machine learning model. So these reasoning with counterfactuals, probabilistic models, these are something techniques where if we want to develop any kind of an systems, without biologists working together with computer scientists is not possible. So those techniques and models would be absolutely novel in the context of the problem domain of biology in which it's being used.
Yeah, thank you very much.
Yeah, so thank you so much, Director Sir, and also
Dr.
participants.
Pankaj.
So, yeah, pardon?
Dr. Pankaj, can I say something?
Yeah, Professor Suresh Sharma-ji, please.
Yeah. I just want to supplement what Dr. Anshu Bhardwaj, she was asking about the unknown distribution. See, there are many techniques and you can find out the distributions even for unknown data using an approach that is called Bayesian analysis. And if you're using Bayesian analysis, you put up a prior and then work out the posterior. And then if you work out the posterior and then you can improve upon that posterior time and again. And then Bayesian analysis will definitely help you to get a better estimates as compared to many other techniques. And they are very useful in deep learning as well as machine learning.
That's great. Thank you so much.
Okay, so I'm now really excited to the following up talks in this session. We have talks from Australia, then we have from NCBS India, and then we also have third talk from Dr. Anshu Bhardwaj from IMTech CSIR, Chandigarh. So, with this, I would like to thank again to Professor Chaudhary for his valuable insights. So, yeah, please.
Thank you, Pankaj. I have to exit. It is a very interesting conference, but obviously other pressing engagements are there. So I would excuse myself and I would like to again greet all of you. Have a great conference.
Thank you so much, sir. Okay, so now the following program is that Dr. Abhishek Kumar will be chairing the session, and so we really look forward to the coming talks. So Dr. Abhishek, now you please proceed. Thank you so much.
Thank you, Dr. Pankaj, and it was really nice to see director sir from your institution, IIT Jodhpur, is uh, uh, is understanding data science, and out of his uh, immensely packed schedule, he prepared, and he gave a wonderful initiation. So I hope that your organization and others who are watching us here as well as in the YouTube will have an impact on it, that the topic is uh, head on by the head of the organization. So with this, I will take on the first session, and our first session has three prominent scientists, and we'll start with Professor Dr. Sobha Ranganathan. And Professor Sobha Ranganathan is working at heading bioinformatic initiatives at Macquarie University in Sydney and Australia. And apart from that, I had opportunity to meet her last December, and we had a very nice time. And she dropped me to the airport, and it was really nice. And plus, if you have grown up in last two decades or even earlier, so she did her PhD from IIT New Delhi, and uh, then she had a short stint in India, and then she moved on to Australia, and from Australia she is heading ApoBioNet initial days, and became first chair of International Society of Computational Biology Australia, and then uh, she kept on heading wherever there is a need for to head in the say different organizations, and we have listed all of the all of our major credentials. And she's working on various areas of the research. Last time, as I said, we met in December in Jalandhar, and it was a very wonderful experience to hold. And she has very nice stories about how as a human we grow, and what challenges we face other than the scientific input. And she's very actively helping students. And if you have any question, please reach out to her afterwards as a mentor. She's mentoring a lot of people. Even I feel blessed to know her. And our society and our conference, as a scientific community, we are blessed to have her here, because I just made one request, and she came, or she was ready, and she just came from finished her class uh, at the university. And madam, stage is yours. And I am very pleased to have you here. I call her madam, but she calls me to tell her Shobha, but I am just calling her madam all the time, because I respect her journey. I respect her immense pressure in 80s, that ladki hai to shaadi karo. She broke out all uh, those uh, those in 80s. He broke those social norms, what is so called old normal. And so the young ones who are seeing the students who are seeing the pressure, parental pressure or whatever, they should learn from her, actually. Both irrespective of their genders, yeah, so both gender feels pressure. And so you are especially in Indian society, yeah. And and there and also you have time, you have to learn from her, that every time you do something, there will a lot of people who will be objective, and how still keep on going. And really nice to have you here, madam. Stage is yours. Thank you.
Okay, so I thank Dr. Abhishek for giving me this opportunity. And as you can tell from the gray hair, I've been around for a very long time. But I did not break any norms. I actually obeyed all my parents and everything and did what was right. We looked after in-laws. I have a daughter who was born before my PhD. I got married during my master's because my mother said, if I got married, did a PhD, nobody will marry me. So she will be stuck with me. So I had to get married. So everything is fine. We have to do what is required of us. That is our dharma. So we have to do that and then we can proceed in life. So today I would like to show you two examples of something that we have already published on uh, machine learning applications. We also do other kinds of bioinformatics studies, but since this is a data science conference, this is important to share with you how we can actually choose data and how we can choose features that will help us to interpret the results. Because if we just did unsupervised learning, we do get excellent results, but how on earth do you figure out what has contributed to this good result? And when you make a prediction, you need to be able to tease out the contributing factors. And it's only for that that we actually use features. And once we have the features, we have explainable machine learning, explainable AI, and therefore that is easier to interpret, especially when you work with biologists or clinicians or other biological scientists. And we would like to understand the contributing factors. So that being the case, let's share the screen and get on with our presentation for today. Okay, so machine learning applications in bioinformatics, and I put my story here. So I live in Sydney, Australia, and I've lived here for some time now. We left Delhi because I just couldn't live in the pollution at that time. So having a lot of issues with my lungs, we had to move. But Sydney is a beautiful place and hopefully you will be able to travel once all the travel bans are lifted. I believe India is now open for travel. I think Chennai has opened up, my family tells me. We are here under lockdown, so we have to do everything on Zoom. So I'm stuck to my chair, and after giving one lecture, I'm here with you to give the talk today. So here are two applications that we are going to talk about. One is finding native protein-like models so that we can understand how we can use machine learning to interpret protein 3D structures. And at the same time, we have done some chemistry-oriented work, which is very significant for drug design. We are looking for bioactive ligands for human olfactory receptors. So these are two different topics, and I will take you through them to show you what can be done, what is possible, what are the limitations, how do you choose a model, how do you choose a methodology, and what data do you require to do your work efficiently. It's all a matter of trial and error, and you need to also read enormously to figure things out. Science is actually one entity, and we have ourselves put and divided it into pieces, almost like a pizza slice. And we say this is physics and that is maths and this is statistics. It's actually one. So that is why we are able to move from one area of science to the other. And we should be aware of what our colleagues are doing and developing in the other areas so we can enrich ourselves and use their techniques effectively. Mathematicians decided on graph theory. A long time ago in the 1700s, Euler came up with graph theory. We used it to understand alternative splicing. And we made a number of graph databases way back, I think in the early 1990s. So there are ways to apply these things. And that's why we need to keep an open mind how we can actually harness developments in other sciences or engineering and use them for our data, which could be from medicine, from clinician samples, from images, which is image analysis, which is also used to actually sequence DNA, and in bioinformatics to understand molecular issues. So here we go. A little bit about me. I actually studied in India and I won a National Science Talent Scholarship and studied my master's and my bachelor's in Chennai. My PhD, I moved to IIT Delhi because, as I told you, I got married and had to move to Delhi, but I did my PhD there. And then I moved to France for a postdoc. At that time, I had a 2-year-old daughter who went to stay with in-laws so I could go to France. So we did the sacrifice bit, but everything is fine. We are back together now. So then I did a small postdoctoral fellowship in the US. We were wondering at that time whether we should move to the US, but then we loved India. We wanted to be in India. So we came back, worked hard. I was a lecturer, leader, and then head of Department of Chemistry at Maitreyi College at University of Delhi. But it was teaching all the time. So I got a little fed up. I was information scientist at the Bioinformatics Center at JNU in New Delhi for 2 years. But by then my health was deteriorating. So we moved to Australia to get away from the pollution. So now I've been here since 1993. It's a long innings. I came as a research fellow and then continued on. And then I worked for a national service organization like NCBI, which no longer exists because Australia decided to close it down. Then after that, I went to Singapore. And that was the breakthrough. Going through the glass ceiling, I actually got an associate professorship. I'm still very fond of Singapore. Again, we spent 3 years there. And then I decided to return to Australia because I got the first chair of bioinformatics and I've been with this university since. I do whatever I can where is required. It's not that everybody can't do it. Some people put in more effort than others. And we also still make dinner, iron our clothes, do all the housework, do all the gardening, we do everything. So it's just a matter of organizing our time and being a productive person in science as well as at home. So what is machine learning? Algorithms can actually predict new instances based on the data they are given for learning. Learning is what we normally do when we go around. When you go or walk to your school, when you're a child, you figure out that this is shorter path than another path. So it's just a way of learning. And when you have different types of data, you can carry out different types of learning. Now these learning methods have actually got names like decision trees, support vector machines, regression, naive Bayes classification, hidden Markov model, random forest, recurrent neural network, short-term memory, long short-term memory, and convolutional neural networks. Some of these are very popular in bioinformatics today. In our current conference in COP 2020, we have received 4 papers on convolutional neural networks, which I've just assigned. And of course, we also see other methods. We have used ourselves random forests and some of the others, and we will show you how they match up and which ones work on which data. There is no rule in this. You have to try different things, and usually you take your known data and split it into 2. So while there are many, many types of applications possible, and this I borrowed from briefings in bioinformatics, which had a very nice overview of machine learning in bioinformatics, we have now we are presenting to you structure assessment and drug design as 2 of the potential applications. So in our structure assessment, what was the problem? What were the methods and what did we do and how did we do? That's the question. Protein structure has always fascinated scientists because proteins fold in weird and wonderful 3D structures. For this, you need to be good at 3D algebra, 3D geometry, and 3D vectors. So that's how we decide which atom is where. So you need to understand 3D space. So it's really exciting. Only now we have 3D printers. We had to imagine all this in our head. So for us, it was a big learning experience. The reason we are interested in the structures is that although we have millions and millions of proteins, they seem to form relatively limited number of three-dimensional shapes. And this is encapsulated by almost 1,000-fold decrease in the number of protein forms that we normally see. And the structures are also about 1,000-fold less. So these are all in millions. And as you can see, there are already 3 numbers here, whereas here we have zeros to give you the difference and what's happening. So although we know that proteins fold automatically when they are being expressed by the ribosome, we really don't know what is the little hidden secret which tells them, hey, can you please form a helix, or can you form a sheet, or can you form a combination of both? And this is still not known. So it has remained the holy grail of protein structure prediction. So this is one of the big challenges in computational biology and has been for many, many years. So predicting the 3D structure of a protein given only its sequence is still challenging, and still people are arguing about it because there seems to be no way we can crack what nature is doing, and that is the challenge. The best available method is when you have a relative which has a structure which is called homology modeling. We use that template or the 3D architecture to build a new related structure. And that gives excellent results if the structural homologue is available. But when there is no structural homologue available, we do know that structure is conserved far more than sequence. For example, we have the TIM barrel, which is the triose isomerase protein, and that fold seems to recur in totally different circumstances. Something like the wheel. So the wheel is available for the cycle or the tricycle or the aeroplane. So there are different variants on the theme. So this kind of structural evolution has occurred for many millions of years. So we can use that as one of the ways to build a model and then see whether that model fits that particular sequence and evaluate it. Maybe that will help us to understand better how to predict a 3D structure a priori. The next best method is therefore to take this fact that structures are conserved, take all the 3D scaffolds, take away their side chains, and simply build thousands of models and then evaluate them using energy functions, usually repulsion, attraction, van der Waals, etc., dipole moment, etc. And then you evaluate and then you see, okay, this one looks a bit better than that one. So I'll throw away the ones that are high energy, keep the low energy ones and come up with a model. Here we do well when we are looking at individual domains, which are sometimes subparts of the big protein and which actually fold uniquely on their own. And they sometimes are associated with a unique function. So I think Saudamini, who's coming after me, is a great domain expert, and she will be able to explain to you much better than I am. Okay. The next best method would be, if that one fails, fragment-based 3D structure building. This actually Professor Jairam from IIT Delhi has mastered, and he uses this in his CASP approach, in critical assessment of protein structure predictions, where they use pieces of proteins overlapping with each other, look for their individual structures, and then try to pull up little bits of Lego together and build your 3D structure. Again, we need to evaluate and then validate, and then we get the final structure. Combine the fragment templates, minimize them, and then hope that things work out. The least accurate of them all, when all these ideas fail, is to simply take your unfolded protein sequence and start shaking it up, doing dynamics and hoping that it folds. This was the life work of many scientists, especially from the chemistry side, where they were using force fields to evaluate protein folding. And this has been going on for centuries. Now, when structure prediction methods were very popular, that's about 10 years ago, almost 20 years ago now, 1994, scientists got together and said, we need to actually see how these methods perform. So let's have a competition. So that is called CASP, Critical Assessment of Structure Prediction. And about 20 years later, we have something called Continuous Evaluation of Structures. CASP occurs every 2 years, and that was the first competition to be started. People who are solving structures experimentally, usually using X-ray crystallography, provide only the sequence to CASP, which is then provided to predictors, people who have methods or who have some programs that use known methods in an optimized fashion to make a structure. They submit their predictions, and then all is revealed. This is like a double-blinded trial. So when everything is revealed, the structure comes out, the predictions come out, and a team of experts evaluate the predictions, and they have a meeting. This year, I think the meeting will be virtual. The CASP just got over, and there will be a meeting. I'm sure assessments are going on right now. About quite a few years later, it was decided rather than wait every 2 years, why can't we just do this in a continual fashion? So that is Cameo, where you have continuous evaluation of predicted structures. Quality assessment is done by the protein structure prediction PSP community, and a lot of scores similar to the other one are developed, and this is ongoing while this is sporadic. Okay. What is it about protein folding that makes it so difficult and interesting and challenging at the same time? A native fold is supposed to be completely extended when the protein is made at the ribosome. However, it very quickly decides that this is not an energetically favorable situation and starts folding up into various intermediate states, maybe saddle points, but finally arrives at a reasonably low energetic minimum, which is called the native structure. This is not a zero energy state, because then the protein won't be functional and we will all be dead and gone. So we need something that is reasonably stable, but is able to have some flexibility to perform its function. And that is called the native state or wild type structure, WT here for the structure people. So misfolding, on the other hand, represents a state of high energy, which can therefore expose parts of the protein which are normally hidden, can cause immune response or deposition of proteins, and can relate to various disorders. The deposition actually leads to Alzheimer's and other diseases, whereas when you have exposure, you cause or trigger an immune response. So that leads to overactive immune responses, challenges, and that sometimes happens even in COVID, as you know. When people are very sick, there is a huge immune cascade that is mounted, and the body thinks that it's under enormous threat and goes into a terrible war zone. So this is similar to that, and diseases are the problem. Transition states are intermediate states. And also when new areas from the interior of the protein get exposed, you can actually have protease activity. So it's all very simple. we believe that this entire thing is driven by only one thing. Hydrophobic patches on the surface are hidden when a protein folds. So how difficult can this be? So we got a protein that was submitted to Cas12. This is, of course, a few years ago. And we took the target, which is called T0859. And these are the details. It's from a bacteriophage. And we calculated the hydrophobic residues which are prone to aggregate. And these are shown in red. And we used a program called AgriScan. There's no need to redo what somebody else has done, just give them credit. And then we looked at the surface accessibility by using the blue background and saw which of these are on the surface. So obviously, if they're all on the surface, then we have an issue because we are not folding the protein properly. So we wanted to see how many of these hydrophobic residues are on the surface and do analysis. So we thought if we could do that, we can actually come up with some measure. And this part is, of course, from our side. While we used other people's programs, we came up with these measures ourselves. So we realized that if 50% of the accessible area is actually solvent exposed, then it should be considered surface exposed. So its SAP score in this case would be decided by the residues summed up and normalized over total number of aggregation prone residues. And then we decided rather than have a continuum of states, we will just have simply 1 and 0. So solvent accessible surface area is considered, and we take a SAP score of 1 if it is accessible, and the rest is considered 0 and carried out statistical analysis. So mapping this aggregation propensity can actually help us to estimate whether the protein is in its native state, while much of the hydrophilic surfaces, that is those that like water, are outside, and the ones that are hydrophobic, most of them, the red parts, are on the inside. So that's what we thought we should be looking for. Whereas in a non-native or intermediate stage, which is higher in energy, we would have more of the red blobs on the outside and fewer of the blue blobs on the surface. So more blue indicates native-like, and more red indicates non-native-like structures. Okay, so we took the whole of the PDB, all the non-redundant data in the protein data bank, the structural database for protein structures, and then removed the short ones. The poor quality ones chose only structures less than 2 angstrom resolution, chose only those that had proteins, no DNA, no carbohydrates, etc. No ligands were present, removed all the ligands. Less than 30% sequence identity, that means not from the same family, so we are not repeating ourselves. So there's no duplication of structure. So we ended up with only 1,500 odd structures. Out of these, almost 1,350 had a SAP score of less than 30. So 90% of the native structures have only 30% predicted aggregation prone residues as solvent exposed. This was good news because most of them are well folded and all the hydrophobes, most of them, 70% are hidden. So 90% of the proteins are in this region. Only 10 proteins had a very high score, more than 50. That is, their hydrophobic residues were on the outside. And then we went in and analyzed them. They were aggregation prone proteins. They were already known to aggregate. They were multimers. So if you took one monomer, then it is not so happy and it has hydrophobes, or it binds lipid phosphoinositide binding. So either way, we could show that this was good. So we thought, okay, we have a wonderful idea. Let's send it for publication. That didn't work because they wanted many, many, many structures to be analyzed, not just the PDB. Applying this to all the native structures that were predicted for CASP, we found that we could discriminate the best models in red from all the rest by only using this one simple score, which we digitized anyway. So it was either 1 or 0 and summed up over the whole protein. So we could actually show that the best 6149 models, the top 10 can easily be detected. Then we did that for a few of these proteins and sent it for peer review. What do you think happens? Peer review feels like this. One reviewer says, okay, the second reviewer says, okay, but the third reviewer says, kill them. So that's what happened. So he's ready to gobble you up. So although we had seemingly good results and could screen high quality models from poor predictions, reviewers came back with a big list of complaints. And this was one of our bioinformatics journals. So they said, go back and make a machine learning method. This is not good enough. You need to analyze much more data because it's become data science now. It's no longer about understanding basic principles or anything like that. So we needed a protein quality assessment, which is not just binary, but a multi-class problem because we have models that span poor quality to high quality. It's like a continuum. Structure files are very large. So we need a fast machine learning method, a scalable and interpretable one. We'll have to be able to tell the biologist why our method is performing well, which part of it is doing well, etc. Avoid overfitting. So we thought maybe random forest is better in this other than SVM or NN. So we chose that, took two large data sets, CASP11 and CASP12. 70% was made as training and 30% stuffed as a test set. Now remember, for one protein, you can have many models that have been predicted by different teams all over the world and then submitted. So we had to choose the targets and their associated protein models. So we don't actually split the target. So we chose 38 targets and all their models and 15 targets here with all their models. That's roughly a 70-30 split. So this is what we do. We collect all the data, split it into two, and then try to throw it into a black box and then figure out what's happening and then try to figure out what it's predicting and why it's predicting that. So that's what is normally done. And so we wanted to do slightly better. So we'll show you how we did this. We also have blind test sets. We had two different sets that we used in this paper. And that's what we are looking at right now. The results will be shown to you because after testing, we applied that model to look at these test sets, which the method had not seen, and then we can evaluate how good our model is doing. Now, Einstein said, for feature selection, make everything as simple as possible, but no simpler. That means select the best features and add new features, but do not add too many. Keep it as minimal as possible. So that should be your aim. Choose the best features for training and then add only those that are essential. Try to minimize, because if you have too many features, you won't be able to explain your results. This is all supervised learning. We are not doing black box unsupervised learning. So of course, we started with the SAT score, so we have to use that. So that's one of our main features. But we also used protein secondary structure percentages, helix, sheet, and loop fractions. Loops are usually most flexible. So we thought maybe the percentage of loops, the loop content would be very important to understand whether a protein is disordered or it's an orderly protein, because that could skew some of our results. And then how much of the loops are contributing to our SAP score, because how much of the loops are hydrophobic, and that's what we wanted to see. And we checked for dependencies. These were all independent, so we are okay. The diagonal is really large, so we were confident that our features are fairly independent. That's very important, because if you have dependent features, it's going to skew your results. Right. So we used random forest. So that's finding the trees. So the data set is split binarily into a tree-like structure based on the values of the features. Then the two branches are analyzed for further subclassification. So it's two-way split. And we generated 700 trees. So that's what we have done. We calculated the PCC, and we found the observed and predicted global distance template, which is a value that is used in quantifying two structures and how well they overlap with each other. We are taking a model and the true result, which is the experimental structure, to calculate GDT. We found that over 50 was a true positive, and did three-way cross-validation, and everything was consistent. So we were very happy. And then we thought, let's apply this to the blind data sets. We used the Cameo models, and then we got a reasonably good value, AUC of 0.82, which looks good. And this is like the blind test set. So as you can see, the blind man is trying to read. So that's what we were trying to do here, trying to understand what happened. Then we looked at domain-wise, how did we do? We seem to have done pretty well. We did really, and we compared to the original SAB CASP methods that were published, that are still online for CASP 12. You can see these results. And we found that we were doing quite well, and even slightly better than some of these methods when we look at our very simple approach to life. This was published in Scientific Reports. After a couple of reviews, they made our life miserable, but then we are happy with the result. The moral of the story is never give up. Even a simple idea can actually do well. Avinash did the work. And he was in Australia at that time. He was in my lab for some time. Abdul is my colleague who is in Brisbane. And he is also from Rajasthan, by the way. Apparently, he comes from a very prestigious noble family, maybe a royal family, I don't know. And Professor Jairam retired a couple of years ago from IIT Delhi, and he's like my batchmate. So we are about the same age. Australia, you don't have to retire yet. They are trying to make me retire, but I think I have to work hard. Now, in my own group, we looked at a totally different problem. Can machine learning be actually used on small molecules for drug design? Machine learning has been used for gene prediction, for microRNA prediction, for many of the other problems in genomics, transcriptomics, looking at gene expression arrays. We've also done a lot of work on proteins. So we wanted to see if it will actually work on the small molecule. For that, we are looking at the missing human proteome, which I worked with a colleague of mine who was president of UPO here in Australia in my university. And this is of particular interest to him. Of all the human proteins, we know only this much. This much, we really have some inclination it might exist, but almost 574 proteins are possibly wrongly classified, and they are probably non-coding. So our interest is in the missing proteins, which have some evidence at the transcriptome, homology, or prediction level. and are possibly proteins. And when we looked at the families of these, we found some of these names. This is just a word cloud. You can generate this yourself. And the size of the characters actually represents the number of members. And you can see what stands out are the olfactory receptors. It's a family of GPCRs, G protein coupled receptors, and it's the largest family of missing proteins. Now, what do these olfactory receptors do? They help you to smell, but many of them are also present in non-nasal or ectopic tissues. Ectopic expression means they are expressed in a place quite far from the nose, such as the brain, the heart, the kidneys, even prostate, the ear. Linda Buck and Richard Axel actually published the paper on olfactory receptors. They got the Nobel Prize in 2004. The same year, there was a paper that came out saying a similar receptor had been found in prostate cancer cells. So as you can see, they are involved in many things. They are involved in spermatozoa, kidneys, etc. And all the olfactory receptors seem to be doing totally different things. They are chemosensory receptors. They're actually looking for some molecule to bind. So that is the best suggestion that we could come up with. And it is well known that they are expressed in a huge array of tissues sorted by expression across all human tissues. 40 of the most highly expressed ones are shown here out of the 405. We have actually found a lot of evidence for many of these from wet lab, that means from mutational studies or binding studies, as well as from other associated studies. And we thought, let's take one and see if we can actually do some machine learning, because we have 143 compounds that are known to bind to this one particular olfactory receptor, OR1G1, which is expressed in enterochromaffin tumor cells in the gut. So our idea was collect all the little molecules, the compounds, the chemical molecules, do some descriptor, which is actually our fingerprint, which is like feature determination, and then process and clean the data, filter it, do some unsupervised and supervised learning, and then do some cross-validation, confusion matrix, estimate accuracy, sensitivity, do some prediction, and send for in vitro testing. In vitro testing is very difficult. There are only like 2 labs in the world which are working in olfactory receptors, maybe 3, 2 in the US and 1 in Germany. So we have to convince them that our values are very good. We managed to do that for another one because this one does not express according to our experimental collaborator in the US. So anyway, we are giving you the results. So we can show you that when we have so many molecules, we found then that there were other studies in the literature where they could find agonists using a similar method, but they used SVM, support vector machine, and predicted compounds were tested using a luciferase assay. Now this collaborator, the experimentalist in this group, is the one that we are working with, and we've submitted a paper which is under review. You know how paper reviews are, it's like a black hole, you have to wait forever. So we are waiting. So how did we do this? For small molecules, the story is very different. There is no amino acid here, there is no secondary structure. So we use what are called moderate descriptors for small molecules, small organic compounds. And these descriptors are generated using this GitHub freely available program. We have used all freely available programs in this study so other people can learn and use these different methods. And we have done the missing value filter, removed stuff that was missing. We have used the low variance filter, and then we have used the ranker, and we ended up with 13. There are 74 agonists and 74 non-agonists. So all the cleaning and pre-processing was done through KNIME and the Orange software package. Orange is a machine learning package that is freely available, and it can be used by anybody. It has a graphical interface, so it's rather easy for students to learn and use. And these pictures were also generated with Orange. So we used 3 classical machine learning algorithms and evaluated each of them: SVM, RF, and naive Bayes, as well as a deep learning model, neural networks. So we use these 4 methods for the learning. 3 are machine learning and 1 is neural network. So we made 125 trees in RF, and we use radius basis function for SVM. These are standard things. And then we use default parameters for naive Bayes. And we use 5-fold, 10-fold, 20-fold, and leave one out, cross validation, just to check how did our classifiers do. So after all this, we predicted a model, and then we used the predictions, and then we used we evaluated various measures to test how they did. So here are the classifications. We used area under the curve, the receiver operating curve, to estimate the overall performance. Then we looked at sensitivity, specificity, and F1. So when we did that, we found that these are requirements that we only did 10-fold cross-validation, but then the reviewers came back and said, that's not good enough. You have to do 5, 20, everything, and show us. So we have shown them everything. So the highlighted values are the best values. So as you can see, naive Bayes seems to be doing really well in many of these cases. So that's what we found. RF did well only in one case, where naive Bayes 20-fold cross-validation as well as leave one out looked really good. Naive Bayes in 20-fold did really the best. So we used that, and then we went for prediction. And for the prediction, we actually had to choose very large data sets, which we have done. So we, oh, sorry, I think we moved too fast. We used a human metabolite database from Canada, HMDB, which has about 114,000 compounds. We use CHEBI from EBI, European Bioinformatics Institute. This is the chemical database. And then we use the cancer odor database because we are dealing with odor receptors, olfactory receptors, and cancerous tissues, because tumor is what we were looking at. And there is another database called OdorDB. So we compiled and pulled all these small molecule data. And since the classifiers were trained on 13 selected features, the test data set was also described using the same features, so we can then mix and match. We used the Maxis fingerprints in KNIME to extract test compounds that were chemically similar. Since the data set is very large, we had to screen the number of positives, and then we selected those that matched our agonists and non-agonists very well, with a Tanimoto score of 0.85, which led us to only 1,500 compounds. And then we found that naive Bayes did the best in the earlier study, so we used that for the prediction. And naive Bayes actually predicted about 300 compounds that were really good as agonists. And since the predictivity, sensitivity of the predictive model was only this, we used a threshold to select the best predictions. Otherwise, we will end up with lots of false positives. So using that, we ended up with about 6 compounds, no, 5 compounds. I'm not even adding well, I'm sorry. I've had a very long day today. So after these predicted compounds, all 5 have known, the 5 have known to have bioactivity are shown here. And we sent them for experimental testing, but our experimental collaborator could not express this receptor. He says it doesn't work for me, so he worked on something else. But we feel that we are actually got something, and if somebody can express this OR1G1, they can test out our predictions. So machine learning has lots of limitations, and especially for GPCRs, what we found was we need sufficient training data. Data science only works when you have large numbers of data. If you had less than the 100 compounds, things don't usually work very well, and you have to go for some other way for the drug design. So we use what is called pharmacophore modeling or virtual screening. And based on this, we have sent another lot of predictions which worked. And so now we have submitted the paper and we are waiting for the reviews. So additionally, machine learning approaches can only see in compounds that lie within the known chemical space. They have no way of actually predicting something that's totally novel. So these are limitations that we have to live with, unless we can come out with a new method that can break through this glass ceiling. So, but overall, we know that through machine learning approaches, we can distinguish between active and inactive compounds in a target selective manner and predict the bioactivities for drug discovery pipelines. These are very complicated systems, GPCRs, and only a few people seem to have the knack of expressing them. So Damini has worked on olfactory receptors in insects, so she might be able to tell you a little more about those. This was published last year, and this year we are still waiting for lots of papers to come through. They are still stuck in reviewing. So I'm done. Thank you. I hope I'm on time, and I'm open for questions.
May I? I'm Sodamini. May I ask a question?
Yes, of course.
Right. Yeah. Very nice.
I don't know where my picture has gone, so I'm trying to figure out what I've done wrong. So I have to get on to. Okay, here we go. Share. Okay.
Right.
No,
Yeah.
this is the wrong one.
No problem. My question may not require a slide support.
Yeah, I'm going to switch it off. I'm sorry.
Okay, no problem.
Stop share.
Yeah.
It's not coming up.
Meanwhile, I have this question. It's a very nice talk, Shobha. I could hear some new works and that are going. Is it yet published or?
It's published. This one was published actually. The experimental work has gone for the one where we had experimental validation. That is under review right now.
Oh, right. Yeah. So because you mentioned that OR1G1 is not getting expressed.
That's right.
Perhaps we can chat and we can try to see, is it from an insect species or mammalian? Mammalian.
This is human.
Human, yeah, human. Right. But still, we could try and see if we can look at the expression here.
Okay.
Yeah, we are primarily working with insect ORs, but we could try and see if this could be taken up.
Okay.
Great.
That's a good possibility. Thank you so much, Damini.
So there is a question.
Yes. Yes. Nice talk, Shubha. I wonder, I might miss something. I wonder about the sample size of your data may also influence the result. For me, it's weird that naive Bayes is much better from random forest. So I didn't see that you mentioned the ratio between positive, negative, or the two class data that you have. So did you have balanced data or you did some?
Yes, we had a balanced data set. We had a slide which said 74 agonists and 74 non-agonists.
So that is balanced. Okay, thank you.
Yes, so it was 50-50 and we used all of them because we had only a few. If you have thousands, then you can sort and, choose the representatives and order them by families or subclass or chemical functionality. But here we had only a short data set, so we used the whole lot.
Okay.
So somebody has asked, do we have opportunities for research internship in our esteemed university? We are also, after 30 years, Australia is in recession now. So we managed to avoid all the recessions, including the GFC. So if you're coming for a research internship, you might have to come on your own steam. But most importantly, the flights are mostly closed. Very few flights are operating right now. So, but we can always do something on a distance or a virtual environment now, since we are well geared. So maybe you can think about a virtual internship. That's also possible.
Another question is coming. Can you use SAP score?
Yes, yes. And it's already published. All you have to do is put in the formula and you can calculate yourself.
Yes. So the another question is coming from YouTube. Brinda is asking, what are types of parameter turned for naive-based classifier?
The parameters are all in the publications. We used the standard filtration methods and arrived at quite a few. If I can pop up the paper, I can actually show you. So we came up with a large number, 713 or something. They're all chemically derived, so they are a bit confusing. So where are we? Cost to be, yes. So how many? They are there in the paper. Okay, classifiers. The classifiers are here. We had 13 classifiers. Maybe I can share the screen and show you if there is still interest. Share. Here you go, 13 classifiers were used, but they are basically involved in chemical terms. So this is log P values, this is characterization of hybridization state, surface electrostatics, sp3 hybridization, and aromatic ring systems, just one. And all these complicated names come from Modred. So this is what we use. So the details are all there. So this is the culled version after all the features, the recurrent features were removed, the filtration process, everything is in the publication, and we are happy to help you if you want to do something like that.
So one more question is coming in from YouTube. Mahendra is asking what happens if the protein is not able to fold properly?
What happens when a protein is not able to fold properly? There are two things that can happen. If it was already inside the ER, this is more like a biochemistry question, not a bioinformatics question. So if it is in the ER, then chaperones will try to help it to fold. If they can't help it to fold, it will be exported and turned over by the proteasome. So most of the normally unfolded proteins are turned over. They are recycled, but sometimes they can cause immune response, especially if the surface represents something that is similar to a known virus or bacterium. Then the body will start an immune cascade. So there is immune response, and usually something like an allergic or autoimmune response is created. Or they can, if in the brain, then they are very difficult to get rid of, because exporting things in and out of the brain very, very difficult. And then they settle down and form various plaques, which have led to Alzheimer's, Parkinson's, and all these diseases. So they are also involved in misfolded proteins. So there are pages and pages on this. So I suggest please do some web search and consult a biochemistry textbook. It will tell you what happens to unfolded or misfolded proteins.
Yeah. So you can, if you have a very specific question, you can reach out to Soha madam. And with this, I will thank her again for wonderful talk. And Mahendra has one more question. Maybe I can raise it. What is the fold cross validation? So 10, 20, 30. Maybe you read in the paper. Yes, something will keep you on not telling you. Then you can go and read.
That's 20. That's a mathematical technique that is used when you do a prediction. Yeah. You shuffle the data sets, 10-fold, 20-fold, 30-fold. That is, you split them into subsets. Yeah. You learn if you make a 10-fold cross validation, you split your data set into 10 bins. learn on 9 and predict the 10th, and then leave the first one out and then learn on 2 to 9 and predict on 1, and then leave 2 out, etc. So you do it 10 times. And then you average out your prediction accuracies. So this is a statistical method.
Yeah, standard
of
method actually. So 10-fold or 20-fold. So sometime you can Google also the before you ask question, because that's very common.
It's called K-fold cross-validation. K-fold cross-validation. I can type that in and you can do it over. We have done that in many instances. This is one of them.
Yes, K-fold cross-validation, and normally 10-fold is very famous one. So then most of time people are using 10-fold.
Dr. Abhishek,
yeah.
Just a little comment.
Yeah, sure, sir.
Generally, when we have large data sets, people apply machine learning and deep learning techniques. But even if there is a small data set, there is a technique which is called bootstrapping, which generates resampling and resampling methods. It is based on statistical distributions, which has to be, imagined from the given data. And those techniques can be used even if for a small data set. And further, the techniques which Professor Shobha has discussed, they can be applied even for small data sets.
Yeah,
using
you mean
bootstrap techniques.
assemble methods?
Yes.
Yeah, sure. Yeah.
So we use leave one out, which is used typically for small data sets, which is similar to bootstrapping.
Madam, leave one out is something different. It is not very much similar to bootstrapping. Bootstrapping is a technique.
It's a technique. So since we use K-fold cross-validation, we did the leave one out as well because it's a small data set.
Yeah, that's okay. That's okay, because at least at each point you will come to know what is happening by using this technique.
That was the general idea. So,
but yes, that's okay.
Thank you, madam. With this, please raise your question to madam. But before that, you make a thorough analysis of Google keywords and searches. Okay, so thank you, madam, and on behalf of organizers, we are very thankful that you've made time for us, and we are glad, and I hope that all the participants have enjoyed it, and I enjoyed it, hearing you every time I hear, and every time you present something new to learn. And with this, we will move on to the next speaker, and we have a clap for madam first, and just do it at your.
Thank you very much, Abhishek, and I look forward to Saudamini's talk.
Yeah. So with this, I invite Professor Saudamini, and she is a professor at National Institute of Biological Sciences, and she has PhD from Indian Institute of Sciences with Professor Balram, and later on she worked with Janet Thornton, if I'm correct, and also associated with Tom Blundell's group, and then she came to NCBS, and where she started her work prior to 2000, and I was one of the JRF in her group when I just finished my master's long back, and we often meet nowadays in Zoom, and so nice to hear from you, madam. And if you have a question, please raise at the end of the talk. Thank you, madam. Stage is yours.
Yeah, thank you. Thanks to Abhishek and Professor Shobha Ranganathan for inviting me for this very interesting seminar series or conference. And today I'm planning to talk about how big data can be used in a particular feature of proteins and how it can be applied in a predictive mode. And for this, I take the example of protein domain swapping. So what is 3D domain swapping? And this was observed when first, so that could be the question. And domain swapping is a particular form of protein oligomerization where there is this sort of a handshake movement where a part of the one monomer will intricately start interacting with the other part of the monomer. And this is a special form of dimer formation. And mostly these are homo multimers. And the first recorded example of domain swapping was from Professor Tom Blundell's lab, and it was observed in the beta crystallines, and whereas the gamma crystallines, which are shown underneath, form normal dimers, the beta crystallines form domain swap dimers. And sorry, the gamma crystalline is shown on top in A. And however, this term domain swapping was coined by Professor David Eisenberg when their group encountered this vivid structural transition, as it were, in diphtheria toxin. So they referred to this as domain swapping. And in this, one can envisage that the normal monomeric state undergoes structural transitions, huge conformational transitions, such that the part of the monomer that is now destined to form now strong interactions with the other protomer will be disengaged, like it happens in satellites and all. So a little part of it is just thrown out in the air, and soon, and when the environment conditions are right, it will form dimer. But now it will form this sort of a domain swap dimer. So this has been nicely described in this protein science paper many years ago by David Eisenberg. And just to recall, these were the people who coined the term. And I'm just going to quickly take you through some definitions, because this will help with our discussions on the domain swapping. So this is a schematic representation. The two monomers are shown side by side, not yet engaged in swapping. And what we show on the right-hand side is after domain swapping has occurred. So we refer to the little part that has gone from one monomer to the other as the domain, the swapped domain. That is, the little region is the swapped domain, right? And which linker gives facilitates this structural transition among the many linkers that could be in a protein, globular protein. It's a special loop region which we refer to as the hinge region, right? And this hinge is special because it facilitated the transfer of this swap domain to the other monomer. And thirdly, we talk about the newly formed interface that is across the monomers, where the bigger subunit and the smaller subunit of the other protomer are interacting as the swapped domain interface. Swap domain interface, yeah, please remember this, as opposed to the regular interface, which is called as a non-swap domain interface, NSDI, which will be there in any dimers or homodimers or homotrimers. This is not special, but this is new and very special to the swapping phenomenon. So 4 terms I'll be using again and again. So let us ask what are our questions. Okay, so we asked a question, since domain swapping is not only structurally interesting, but imagine this does not stop with dimer, and it goes on to trimers and tetramers, and in an unlimited fashion, like Professor Shobha Ranganathan mentioned, it's almost like the protein is now lending itself to become unfolded, because then it can, due to swapping, engage in unlimited oligomerization, and it's no longer fun, not even for the structural biologists, because such unlimited oligomers are usually implicated in neurodegenerative diseases. And hence, there is a strong interest to see whether we can recognize even which proteins are likely to form this sort of a swapping event. So we asked this question using computing techniques, which is, if we were to implore into the entire human genome, can we use machine learning techniques, learning from the known swapped structures, and applying machine learning tools to recognize which part of the genome and which genes are likely to be engaged in domain swapping. So that is our question. Okay, and for this, I'm going to take you through a few steps, and this will form the kind of the logic in the talk today. First of all, if you want to do some prediction, we need data. We need so the positive data set and negative data set. So the first part will be about the data curation and the little analysis, that the structural analysis that will be required to convince ourselves that indeed by knowledge-based method, by looking at the known swap structures, we will be able to apply it in a predictive scale. So the next part is to take you through the machine learning algorithms that we had applied, and then our application on entire genomes. Entire genomes, not only human genome, but a few others, and I'll tell you why. And last but one, once we have large data sets, huge data set that we trust, we can try and do what is called as a meta-analysis. Meta-analysis is like kind of interleaving other related information, such as human disease information, human genetic information, because we know that especially when we look in the human genome, our interest is not only the structural aspects, but how certain genes might be involved in neurodegenerative diseases, right? So for that, meta-analysis become very natural. And finally, I want to leave you with the thought that are we able to pinpoint which part of the globular protein, namely the gene product from the human genome, and which linker is likely to be the culprit, because that is the one that acts as the hinge. So I would take you through these steps quite one after another. So the first part is to curate the data set. So for this, we started from the entire protein data bank. At that time, there were, this is C, can see this is 2005, but then we keep updating this database. So what I'm showing the methodology alone, the flow chart. So we start from the entire protein data bank. Of course, we can make it non-redundant so that we are not looking at the same protein at different crystallization states and so on. So that gave rise to about 36,000 structures. And then we apply a domain recognition algorithm that has been developed in-house, which is called as DIAL, Domain Identification Algorithm. And that will help us to know from objective methods, such as measurement of distances between secondary structures and the loops that connect them, and clustering these elements on the basis of these average distances to then recognize spatial clusters that should correspond to structural domains. And in doing that, if we encounter cases where a domain in a PDB contains a bit of the A chain and bit of the B chain, then it would suggest to us that there is swapping going on. So this information has been exploited to pool all those entries in the protein data bank that could form, that are evidence to already have the domain swapping in place. And in addition, just as filters, we make sure that there is a symmetry, because what happens in one proteomer should also happen in the other proteomer. So therefore, we should expect some amount of symmetry. And we also had done text mining to make sure what the crystallographers think about it. Not that it is final, but if they have also recognized it, we add that value. And that means we do a lot of manual curation. And from 36,000, we were very strict with ourselves, and we created a positive data set, which consists about 2,000 domain swapped entries. So you might be wondering, what is the negative data set? And usually in machine learning, and when it applies to protein structure prediction, always the negative data set is pretty tricky, because when we say these protein entries have been evidence to form domain swapping, we can never ever be sure that those other proteins which are not showing evidence of domain swapping, which will never form swapping. So what we see is only from a crystal structure, right, in a particular pH and a crystallization state. So the same protein, it has in theory the possibility to engage in domain swapping given a totally different condition, a crystallization condition which is different, or the pH may be different. So therefore, when we create a negative data set, we are just making some safe assumptions that such proteins that are not anywhere related to the positive data set members at a superfamily or even at the fold level, so those will form our negative data set. So we curated these data set, and then that kind of shows how you can use near automatic methods to do, so that we can keep updating our database. And this is all present in a database called 3D Swap. And 3D Swap, you can see in from our lab webpage. And our lab is fondly called as Computational Approaches for Protein Science, or CAPS in short. So you will find this as 3D Swap. And in fact, as I mentioned, we have been updating, so there will be a 3D Swap Plus as well. So you're welcome to go through this. However, in the present context of structure prediction or domain swapping prediction, and being able to apply on the whole genome, this forms the positive data set, and we discussed the negative data set, which are proteins that are in the nowhere belonging to this fold. So then we will swiftly go through what sort of parameters which will go into the machine learning algorithms, and for this we performed some structural analysis just to be convinced ourselves whether indeed the domain swap protein structures, the positive data set members, are they anywhere different? Are they behaving different structurally from the negative data set? And for this, we primarily concentrated on the hinge region, that is the linker that is responsible for swapping, the swap dimer region, and also the swapped interface, as well as the non-domain swapped interface that I introduced you right in the beginning. The domain swapped interface is the special interface that has been formed after swapping, whereas the NDI is the non-domain swapped interface, the regular interface that is there in general homodimers. And you can see we had looked at features, structural features such as protrusion index, whether there are disulfide bonds, whether the interactions are different, and what about the van der Waals interactions and the hydrogen bonds at the domain swapped interface versus the non-domain swapped interface. And I have put arrows to quickly highlight to you that certain features are dramatically different between the two. The domain swapped interface, it will be interesting to know, although these are newly formed interfaces across the protomers, is not a transient, it's not by chance, because you can see that there are 3 times more van der Waals interactions at this little part that is engaging in interaction. So it seems to be a very purposeful one. And even the hydrogen bond interactions are more, and these are recorded in using our energy calculation method, which we had imparted into a related software, which is called as coil check from our lab, which will primarily start to worry about what are the energetic parameters at a protein-protein interface and dissect them into van der Waals type and so on. Okay, so we are convinced that indeed the positive data set and negative data set are different, but how can we convert it in a way that given an amino acid sequence, we can recognize them? And for this, we have to look at not only backbone conformations, but also amino acid type. And in terms of backbone conformation, interestingly, what we have in the left is a domain swapped region, the hinge region, and you can see that many more observed allowed Pfizer values are there in that swapped region. So again, it is not a chance interaction engaging into some energetically unstable states, because it seems they are, in fact, that there are more allowed regions in the swapped region compared to the non-swapped region. And this again shows you that these hinge regions are special. And how special are these? I'll show you in the next slide, which I hope, because this is stopped moving. I don't know why. Maybe, yeah, now it's okay. So what we have in general hinge, the monomeric hinge, are these reverse turn sort of structures. But what we see in the oligomeric hinge region, where there is a swapping going on, it is like an S-shaped structure. And they have distinct Pfizer values, and I would not be able to go into the details, but just it kind of encourages us to think that indeed the van der Waals interactions are different. The hinge conformation is also different. And what about the amino acid preferences? So here again, this is a very nice table. And unfortunately, since I'm interested more to show you how this applies to human genome, I'm going to go through this a little rapidly. But what we have is an amino acid propensity from our positive data set, which is merely we are asking how often do you find, say, lysine in the hinge region? And this is, if it is says 0.025, it may suggest that it is small, but we are actually comparing it for the general preference of the lysine to be in loop regions. So we are comparing the amino acid propensity of K lysine to be in general in loops in comparison to how it is in hinge regions, which is a special loop, right? And we see, therefore, positive values means it has a higher propensity to be present in the hinge, and it has been arranged in the order of preference. So if you see high values, that means it's not a chance preference. It's not as though it likes to be in any loop. It prefers to be in hinge regions. And you can see lysine, tyrosine, valine, glutamine, phenylalanine. And so after that, proline has a slight tendency to be in the hinge region. But most interestingly, residues such as alanine and valine are present more at the hinge region. And what we have shown in these sort of bold arrows in blue and red are the kind of single amino acid substitutions that biochemists all over have already recorded in their papers, and the papers are marked here. And this is from detailed literature survey where single amino acid substitution at the hinge region had dramatic effects. So for instance, there's one example in the arrow shown in red is if a residue lysine was present in the hinge region of a domain swapped protein, and if it was altered to glycine, the entire swapping has been disrupted, suggesting that single amino acids can have huge effects. And the opposite is shown in this blue. If we change a glycine to an alanine, I hope it's alanine because it's covered, but my experience tells me it is alanine, right? So the glycine to alanine mutation, on the other hand, had in fact induced domain swapping. So you see, the kind of system seems to be very suitable for machine learning and prediction of domain swapping. So on the one hand, we see overall van der Waals interactions are more, backbone conformations are special at the hinge regions, and even amino acid preferences are there for certain residues to be present at the hinge region. So with these sort of structural analysis, we went straight on to preparing our system for machine learning. And for this, what we do is to, first of all, finalize our positive and negative data set, as I said before, and create a set for the training. And then we will separately set aside a set for the testing, which will come to. So around 2,000 proteins each were kept from the positive and negative data set for training. And what are all the features? And so we brought in a number of features, such as average flexibility index of amino acid, the polarizability parameter of amino acids, the size of residues, and the net charge, their inherent preference to be in the coil region, and so on. So as you will see, almost all these parameters are designs or borrowed such that they don't rely on structural information. You may ask why? Because what we have from genome databases is only the primary structure data, namely the amino acid sequence. So therefore, it makes sense to not rely on structural features, such as we could have easily said, how often do we see residues, amino acids in beta strand conformation? So we could have fed in as parameters, but we have not done that. So bulkiness of amino acids, isoelectric point, all these features have gone into the machine learning. And together with the positive and negative data set, we use the random forest classifier, which allows us to use a combination of decision trees, depending on what sort of parameters can go into the decision. And then together, these trees will talk to each other, provide some votes by which, given that there is a positive data set, and given that we know that there is a negative data set, the system, the random forest classifier, is going to use and recognize which features are more important than the other, which features discriminate this positivity and negativity, and that is what goes into the training, and then it will be applied on test data set. And if we have any values such as measured by parameters such as accuracy, it will help us to know how well the machine learning training has happened and how well it is working on an independent test data set. Okay, and for this, I have talked to you about the data sets, and I have talked to you about the features, and then I have talked to you now about the classifier, right? So I'm just going to show you a confusion matrix, what as we call it in statistics, which is nothing but knowing that there are about 2,000 entries in the positive data set, and there are 2,000 entries in the negative data set. If the random forest based classifier training is absolutely ideal, you will find 2,000 true positives and 2,000 true negatives. However, because it is trying to learn from first principles, it is not having the structural information. And what one feature will tell may not always agree with the other. In general, we are doing this prediction with very little information, only the amino acid sequence information. So there are some false positives, some which are wrongly put into the positive data set as though it is domain swap, but actually not true. But the same way, some have left out, and they are in fact domain swap structures, but they were treated as though it's part of the negative data set. And therefore, these parameters then help us to know how much is the accuracy, how much is the specificity, and how much is the sensitivity. And you can see that all these values for the training data set of 2000 entries is quite high, around 86%. And the MCC value is a composite information. We call it as Matthews coefficient, which is also quite high. It ranges from 0 to 1. And therefore, it encouraged us to go forward and applying it on a test data set and finally on the human genome, right? And to do this, we created, we next looked at it, how it is in the test data set. And test data set is something that you save it and you make sure that you're not biasing your training. And then the test data set, if your machine learning works very well, that means you are really in a position to apply it on unknown databases, unknown protein sequences. And here again, out of 216, we find that 188 are recognized correctly as true positives. And out of 216 in the negative data set, which we had saved for the testing, 177 of them have been truly recognized. So that again shows you really high values. And for us, it depends on how challenging these questions are. And to give you an idea, I want to tell you, some proteins have signal peptides by which they go into different parts of the cell and localize themselves. And to begin with, the best prediction method available at that time had a accuracy of little more than 50%, maybe 56%, and you generally improve with time. So values such as 80%, or say ranging from 0 to 180.81 values are actually quite good. So armed with the positive and negative data set, encouraged by our structural analysis, and armed with the machine learning classifier and all these features, and knowing that it works reasonably well for a test data set, we then did something which is obvious, which is to apply it on human genome. And we first applied it on the human genome, and we found out of 20,261 genes that we had taken up, nearly 14,000 of them had rendered themselves as domain swapping positive. So nearly 71%, and that looked like a startling number to us because, you know, we wanted to know why there is so much response. Could it be true? Or is it just because our method has been overly lenient? It can't have been, because whereas the sensitivity is very high, the specificity, that is how correctly, is it just going to greedily predict many things as positive? No, because our specificity is also around 81%. So we knew that our machine learning is not overly relaxed and picking up many as domain swapping positive. So that means maybe this is how genomes are. And to ascertain and convince ourselves, we applied the same on two other genomes. One is a bacterial genome, and then another is a plant genome. And when we did, we were surprised that similar percentages are seen. Around 70% of the genomes have a tendency to engage in domain swapping. And for us, this was a very good learning and new learning. And we then asked ourselves, if 70% of the human genome are out there to get into a domain swapping, does it make sense from the angle of what we know from disease perspective? After all, this is the human genome, right? And for this, we went on to looking at what is called as a meta-analysis, by which we will like to know how many of them are already known to be engaged in unlimited oligomerization. Is there evidence that they form aggregates? And for this, we found that there are 136 human genes that are recorded to be aggregation positive. And that means maybe they are domain swapping positive. And to our surprise, we found out of 136, nearly 100 of them have been picked up by our machine learning algorithm and training as likely to be domain swapping positive. And out of this, 99 of them are enzymes. So you see that out of the many human gene products that are shown to be aggregation positive, majority of them are catalysts, are enzymes, so suggesting that the structural feature of domain swapping might suggest that not only it can form plaques and engage in neurodegenerative processes, but a vast majority of them are tuned, and the signal transduction or the catalytic machinery is kept available for under being of regulation, perhaps through the process of domain swapping. Perhaps the swapping gives them the extra regulatory keys that are required to perform their functions. And then we asked the question that how many of these domain swapping predicted members that we see in fact fall into what types of domains and what sort of enzymes, because we know that nearly 100 of them are enzymes. What sort of enzymes are these? And many of them, in fact 30% of them, are kinases. And it makes sense because we know that several of the kinases are in fact membrane bound, and some are not membrane bound. But then if there are tyrosine kinases and they are sitting on the membrane using a single transmembrane helix, perhaps the formation of the dimer and the phosphorylation, the ligand binding, they're all kind of introduces the feature of domain swapping as one of the regulatory mechanisms. And then you can see immunoglobulins, sugar kinases, and then the pH domains, and so on. And then these are protein-protein interaction motifs, and definitely we see them. So this suggests to us that domain swapping is used by nature not only to form unlimited oligomeric structures, but within catalytic machineries as a form of regulation. Okay, so this is the machine learning part, and then I will like to now stop by saying our challenging question that we had asked ourselves, whether knowing that a protein is likely to form swapping, can we recognize which is the culprit, which is the loop region that facilitates this process. And for this, we had to recognize hinges amongst 3,000 entries, and we definitely can't do this manually. So we set up several structure algorithm in our lab, and some of them are a bit like the David Rose method of recognizing domains, where we put a plane, cut across a plane on a given protein structure. And this helps us to recognize where are the two domains present, and can this help us to pick up objectively and automatically where are the hinge regions. And for this, since we are testing this approach, we had consulted the literature to look for this term hinge, and where possible, if the residue boundaries are there, we use them. And we found that it is helpful to recognize hinge regions, because you can have loops of all sorts, but certain loops that are crossing one chain to the other and are fairly extended S-shaped structures, as we saw from our analysis, and more solvent exposed. So they are like having more intermolecular interactions, they are more extended, and they are more solvent exposed. Such loop regions were picked up near automatically by us, and we call this as a HID, which means hinge identification algorithm. And we collected a data set, a subset of hinges. And then we used that again, put it through a training, which I'm not going through the details. But then when we applied it on pH domain, which one of the top ranking modules that are there in enzymes, which are known to be aggregation positive, we were able to identify or predict the hinge regions out of this whole globular structure. It has many loops. And out of this so many loops, only this loop was predicted as hinge. And indeed, it is the correct hinge region. It was also shown to be present at the structural transition region, because we do have pH domains which are in the swap form, suggesting that such an approach, being able to pinpoint which part of the structure which has been predicted to be domain swapping positive could lead to tinkering around, because if we know which is the hinge region, already we know that single amino acid changes can dramatically alter the oligomerization state. And even if we are not able to mutate it, we can identify some small molecules that maybe will sit at that hinge region and disturb the oligomerization state, in case the swapping happens to be deleterious for the system and gives rise to neurodegenerative diseases. So with this sort of description, I would like to stop by acknowledging the people who are involved. Prashant did all the structural analysis, like the positive data set accumulation and the HID algorithm for hinge, and he's now in Singapore. And Atul Upadhyaya was the brave one who applied it on genomes. including the human genome and later on Tulsi genome. And he's now a faculty in the Punjab University. And we also had previous members like Shamir and Pugalendi, who had actually contributed to the machine learning algorithm, and this helped us to predict genes that may be engaged in domain swapping in the entire human genome. With this, I think I will stop and take any questions which you might have. Thank you.
So thank you, madam. Thank you for a wonderful talk. And any question, please raise your question. Yeah, I see there is some question. Sri Ram is asking, can we study swapping using MD simulation? How will ML replace MD simulation, if
Yes.
any, in the future?
Yeah. In this talk, I could not bring it up because it was more focused on prediction. I didn't want to spend too much time on the analysis. But what we had done is a kind of a tricky MD attempt, which is we took a 4-residue hinge where, as I showed before, there is a reverse turn in the non-swapped structure, and then we had a domain swapped structure, where there was a S-shaped region as shown here. And we started off with one, and using MD simulations, we showed that it goes to the other state, suggesting that this hinge region in fact, we can use MD to follow the structural transition. So, but then it was a small hinge, only 4 residues long. So it remains to be seen whether in the midst of a protein, we will always be able to recognize structural transitions and hinge using MD simulations, because these were, loop regions were isolated and away from the protein, and indeed they show evidence of hinge transition using MD.
The next question is, Brinda is asking, what are the significant features that are used for swapping prediction?
Yeah, I had not brought it up, but what we had seen was 8 features were quite important. And these include definitely the coil, the tendency to form of amino acids to form coils. And then their hydrophobicity was an important one. And then the third I remember was bulkiness. And then size of the amino acids. So overall 8 parameters mattered. And definitely I remember that the bulkiness and then their tendency to form coils, these were the top ranking features.
Can I ask a question?
Yeah, please.
Sure.
So Domini, excellent talk. I was thinking when you showed us the picture of the hinge and the domain swapped Ramachandran plots, they looked, the shape of the loops looked so much like the 2 states of the disulfide bridges, the spiral and the left-handed hook.
Yeah.
So maybe there is something there that we can use to actually
yes.
figure them out much faster.
All right. Yeah. That's a very clever observation, Shobha. With I just flashed this slide, but then I think the world is so small. Definitely, I would like to relate it to the disulfide parameters that you had mentioned earlier, described by Professor Jane Richardson as well. However, I again didn't have the time to talk about it. And what you see down below is my first paper, where we had taken small peptide regions, and there it was proline rich. And we had seen that whereas the beta turn conformation is the preferred structure for proline rich peptides, at very low temperatures, where we are able to capture unusual structural states, what we evidenced from NMR nucleohauser effects, several non-beta turn. And in fact, one of them is this S-shaped structure, and one of bisulfide bonded as well. So I think they fall into place very nicely. So perhaps the backbone conformation, certain backbone conformations are associated with certain types of disulfide bonds as well. Yeah, very interesting.
Someone is asking how the binding energy in this case is calculated.
Binding energy, yeah. So we had actually collaborated with a group in Manchester where we looked at detailed electrostatics so that it can give rise to more precise energy parameters. And then we compared it with the available kind of thermodynamic data that are there, where two proteins will form dimers. And you can imagine how hard this can be, because most of the domain swap structures that we have in the PDB are actually homomultimers. That means you have the same molecule coming and forming this dimer. So therefore, we have to do a concentration dependent experiment. So we do not have much such data in the literature, but where possible, we had used them. And we had in fact also shown that there is a pH dependence. Unfortunately, I didn't have time to talk about it, but there are measurements. Yeah. Okay.
Any more
Yes,
questions?
are there any more questions, or yeah? You can always write to me. My email address is there on our webpage, NCBS Saudami. Yeah, so you'll be able to search.
Yeah. So yeah. So as Madam said, you can always reach her and to any speaker in the conference, and that's the whole idea, that bridging, if there is any gap in searching what you're looking for. And so you can reach out to all the professors and the speakers for training or whatever you require, and a research question if you have. Thank you, madam. Very nice talk, actually. And we clap again virtually.
Thank you.
And and this is second time madam just came on my request that we are organizing something in this year during COVID. And it's always pleasure to be around madam whenever there's some meeting going on, and nice talks, are very different talks at time to time. And we are very happy to host her, and we have learned a lot. Most of us have learned a lot from her experience. And many in this talk, I'm one of her student, and then some other students of her are also presenting. Raghu will be presenting tomorrow. And so as thank you from the organizers, and let's hope to meet the madam again in another talk. And thank you, madam.
Yeah,
And
thank you so much. And it was very nice meeting you all virtually, and we hope to keep in touch. Thank you.
Thank you. Have a nice day ahead.
Thank you.
And our next speaker is Dr. Anshu Bharadwaj from CSIR IMTech Chandigarh. And those who attended yesterday the workshop, she was presenting Galaxy India. Now she's heading the organization of Galaxy India and outreach, development of the platform and outreach activities. And also she has her own bioinformatic team, and they presented yesterday as well. She and her team. And then today she will talk about antibiotic resistance and the bioinformatics behind it. So please, Anshu, Dr. Anshu, please, stage is yours.
Thank you so much, Abhishek, for having me here. Am I audible?
Yes, Anshu.
Great. So.
Oh, I mute myself rather than you. Oh, sorry. Sorry. I'm unmuting. I muted you by mistake. You can unmute yourself. I will mute myself.
I did. All right. So, okay. Good morning, everyone. Can all of you see my slides? Abhishek, are my slides visible?
Yes.
Yes.
Great. So basically, I would really like to thank Professor Ranganathan and Professor Sodamini to make my life very, very easy, because there are certain concepts on protein structure as well as machine learning that I'll be covering in the presentation today. And since you have heard the fundamental principles from them, I'll not repeat that, but may like to extend the applications of those methods to understanding and addressing antimicrobial resistance. Now, assuming that antimicrobial resistance is not something that everybody understands, so I have given a very, very brief introduction to the topic. So antibiotic resistance is nothing. If you look at a group of bacterial organisms as seen on the left side of the screen, you would see that there is one organism which is marked as antibiotic resistant, which means that when you give a specific antibiotic, all the other bacterial population will die, but a few will survive because they have acquired or inherent resistance to this particular antibiotic. And since the rest, the susceptible ones have died, the chances of this antibiotic resistant bacteria to proliferate further goes higher. Now, as we can clearly understand why this could be a problem, because in antibiotics which are functional now no longer remain functional and the infections cannot be treated. Going further, this is a very static picture. So what Harvard team did a couple of years ago, they wanted to also show this in action. Like, can you see drug resistance in action? So what they did, they made a huge Petri plate. The Petri plate is of the size of 4 feet by 2 feet. So you can imagine a huge Petri dish. They actually added media, which are required as a food for bacterial population to grow. And then in this case, they have used E. coli. Then they add an ink to the media so that, the media appears to be black. And then they divided this particular plate into several sections. So if you look at both the ends, it says 0. It means that this particular vertical does not have any antibiotic. This one has 1 times the antibiotic. This one has 10 times the antibiotic, 100 times and 1,000 times. So as you go from both the ends to the center, the concentration from 0 antibiotic to 1,000 times more antibiotic, they kept on increasing it. And then what they did, they actually had the bacteria growing in the 0 antibiotic thing because it's a natural way to grow any organism in a petri dish. What they noticed, that despite the fact that this organism was not really resistant to antibiotic initially, at 1x, 10x, 100x and 1000x, the bacteria actually could evolve and they could reach concentrations or survive in concentrations of up to 1,000 times more antibiotic. And what you see here in white in the right hand picture is the growth of E. coli. And they then have shown that there are very many mutations that happen which actually allow these bacteria to become resistant to the antibiotic, even higher increasing concentration. So this is something which actually we know that microbial population can develop resistance to antibiotics because they are evolving continuously. So it's not a problem of drugs as such, it's an evolutionary process that is happening all the time, and we want to understand this so that we can address it better. It is not just in lab, even in clinic, and since the day we discovered our first antibiotic, penicillin, you could see that as soon as the antibiotics, as you see at the top of the panel, are introduced in clinic, you start seeing resistance either the same year or a few years later of the introduction of the antibiotic in clinic, which means that it's an arms race. So basically we are trying to discover new drugs, but the bacterial systems always come up with a mechanism to overcome them. Now, this is no more a problem that is published in scientific literature. You will see this in popular media, news articles, and given that we are living in an era of pandemic, these antimicrobial resistance is a sleeping pandemic, and this is something that realization has to come sooner than later, that these are pandemics in the making and we should address them immediately before it gets too late. So in order to address that, WHO actually came up with a priority pathogens list. So some of you who are looking forward to building a career in science, there is a huge plethora of organisms against which the only solution is to develop new antibiotics. There is no other public health intervention that can actually help to address the issues. So this is an entire list for you to follow. What I'm gonna follow today is 2 examples of how we have done this in case of MTB. Now, this is World Health Assembly in May 2015. They identified that antimicrobial resistance is a major issue, and they have now a global action plan, which is being executed by all the members of the World Health team. And my lab particularly focus on education and awareness, as well as research for new therapeutics and diagnostics. And for education and awareness, my lab also develops video games so that we can engage younger kids to know more about the complex phenomenon of antimicrobial resistance. So as you can see, there can be several ways in addressing AMR, but the focus of today's talk is to see how do we do early stage drug discovery to identify new molecules that can act on these difficult to treat organisms. Again, drug discovery is not something very common to everyone. So let's get two terminologies clear. One is a drug target and the other one is a drug. A drug target is nothing but a molecule in an organism, which is usually a protein, and it is intrinsically associated with the disease process, which means if there is a chemical entity, which in this case a drug, is designed that can work via interacting to this protein, then you can have the desired therapeutic effect. That means you can treat the infection, okay? Now, in case of TB, the problem is not that you don't have drugs. You have more than 30 drugs that are being used in clinic, but TB very soon from susceptible converts into something which is called as a multiple drug resistant TB or an extremely drug resistant TB, which means that more than one antibiotic are already given for susceptible TB. Approximately 7 antibiotics are given for MDR TB, and despite that, half of the patients never get treated, which means that we need to identify new mechanisms by which we can understand and exploit the understanding of the target or the chemical space to work on it. And my lab has been engaged in approaches for identification of novel chemotypes for quite some time now. And we take two independent approaches. One is to basically how do we explore new target space, because you need to have new mechanisms, as well as exploring new chemical space. So what I thought today, and if time allows, I'll try to quickly cover it. There are various applications. So because the conference is on data science in biology, I thought I'll try and cover multiple methods that our lab applies and also several labs in the world apply. So in case of the antibiofilm space, we use machine learning on small peptides. In case of this particular activity, we developed a very new method that I'll be describing in another 1 minute on based on identification of small molecules based on understanding of the protein structure. And the last one are graph theoretical approaches to come up with drug repurposing candidates. And I'm pretty sure that we can cover all three. So again, as I told you, that our lab has been working on identifying new drug targets for TB, and we didn't go by any opinion-based strategy. We took a data-driven strategy. We went through all the published literature in TB that was published and applied several graph theoretical methods like between centrality or metabolic network analysis methods like FBA to came up with a list of targets. And these targets were eliminated from whatever is present in the host, both at sequence and structural level, to identify targets only unique to Mycobacterium tuberculosis. So now you assume, I'm not going into the details of any of this because this is all published work. So point is, now you know which protein, as we discussed earlier, a drug target is a protein that when actually interact with a chemical entity, which is a drug, should render MTB ineffective. So basically it should kill mycobacteria, which means that this protein is essential for the function of MTB. So we actually wanted to develop new lead generation strategy, which means how do you design new molecules. So we took an example for this is a TAP-B. The logic was pretty simple. As I told you, it is not present in the host. In MTB, it is responsible for generating metabolites that feed into multiple different pathways. So the point is, by choking this particular protein, you can actually have several pathways choked at the same time. And more importantly, it has 9 crystal structures solved both with the substrate, with the inhibitor, with the cofactor. So you basically, the understanding of the protein structure as well as the binding pockets is pretty good, and we didn't want to basically have protein structure predicted and then make more predictions on that. We thought we'll start with a very robust experimental data sets and build our model on top of it. So the basic hypothesis is that instead of looking at protein from a single binding site perspective, we wanted to exploit all the potential binding sites in a protein to come up with combined pharmacophore features that we can link together by various linkers. And these pharmacophore features can allow us to explore different libraries to pick up compounds that now fit in these binding pockets. So the logic is, you look at the binding pockets, look at what binds, what are the interaction features, generate a pattern of these interaction features, and look for these patterns in bigger library. That is a simplistic explanation. So as on A, you see, this is what I've already told you. So the product of DAB-B actually feeds into multiple pathways, ultimately feeding into the development of the mycobacterial cell wall, which is pretty significant for it to survive. So that is our target of interest. And on the B figure, you can see that we have generated 6 different models. It's very simple. If you see here, sorry about that. If you see here, this particular enzyme has a cofactor binding site, and it actually binds NADPH and NADH both. So what we did, we took 2 models in which this is basically the PDB-ID. We took NADPH and PDC in the bound form. Then we took only NADPH. Likewise, in 1P9L, we took NADH and PDC and only NADH. And in these 2 hybrid models, what we did, we took the nicotinamide part of NADH and clubbed it with the PDC moiety. So because as you can see, PDC, which is the only known inhibitor for DAB-B, is very small structure and the interaction features are not specific. We wanted to bring in specificity, but since the cofactor is used in like several reactions in human as well as mycobacterial cell, we didn't want to make it a very toxic molecule. So we eliminated the part which was mostly interacting with the cofactor binding sites, picked up a nicotinamide part and picked up this PDC attached to it to see if we don't, at the time of design of this strategy, we had no clue that any such molecule even exists in literature or has ever been synthesized. So this was a gamble. We wanted to check if we can find something. And this basically is to provide you understanding of where NAD, the nicotinamide binding part is and where PDC is. So if you can see, MTB DAB-B is a homotetramer. And what I have shown you here is one monomer that has the C-terminal and the N-terminal domain. And PDC and the cofactor binds here. Zooming into the cavity, which is C1, you see that PDC is at the C-terminal side of C1 and the nicotinamide part is at the N-terminal side of C1. And what we did, we took PDC, we took this part and had an isopropyl linker join the two as our potential interaction features. So we took we generated 6 model systems. And then don't worry about all this. This was done in Schrodinger, but we can also, we already are trying to basically do it with open source applications. So you basically do pharmacophore model generation. So we actually let this MP simulation run for 200 nanoseconds and pick up trajectories which are representative of the interaction features that are being generated. And it led to conventional, as in the cofactor binding pharmacophore features, as well as the hybrid molecule, the hypothetical molecule interaction features. And then we took zinc, Acinex, and several other libraries put together, and we did map. We did a pharmacophore screening, a e-pharmacophore screening on entire libraries. Now this gave us two types of data set. One which was edge set, which is coming from your hybrid molecule. Now this is interesting because we had no clue that such a molecule ever existed, but you do find it. And then of course the end set molecules are there. Once we know that these molecules exist, like particularly the edge set, we generated another library, which is the M set and N set for mycobacteria. So we did a curation of all the potential antimicrobial inhibitors in literature, and it is like 3,400 molecules. And we actually did a structural comparison of the H set versus the M set. Now I'll tell you why we did that in a minute. So this is basically to discuss the initial observations. So what we see for the best top H set 1,000 molecules and top N set 1,000 molecules, you see that the docking score as well as the ligand efficiency is better for the H set. So our hypothetical molecule has a better binding to the binding pockets of DAB-B as opposed to the cofactor. And structurally, the H set versus the N set are very different. Likewise, the hybrid set versus the mycobacterial set are very different, which means that we have managed to identify new molecules. And as you can see here, the H type and the M type, the interaction features don't match one-to-one, which means that we have been able to discover new interaction features, and now we can use this to design molecules that have that are more drug-like in nature. Okay. Now, why we did that? We did this because the success rate of target inhibition in any drug discovery project in antimicrobials is practically zero. You never start from a target and reach to a drug. It is always that hits that you get from a wholesale screening, brute force, that has given you all your antibacterial so far. So this strategy, what we did different was, we knew this target was important. We knew this library is shown to kill microbacteria in experimental settings. We generated hypothetical pharmacophore features from the protein structure, and then we mapped it to this library. And what we have now are compounds that are already shown to kill microbacteria, but now we also know that they might be doing it via DAB, because targets of this particular library are not known in MTB. So it's a two-pronged strategy. Now we know the target, and of course that these compounds can be tested. Now we are doing those tests to find out if they really inhibit DAB. You have something which is ready at a very advanced level for a drug discovery project. So this is our first contribution. It's a new method, and this paper is available in bioRxiv, and it is under review in a journal. And Chinmay is my collaborator on this project. And as opposed to several lacunae in the conventional practices, our new strategy actually allows us to understand the mechanism of action. We can bring in target specificity. It has no cell penetration issue, which is a huge problem in case of target-based design. And then, of course, we have chemotype diversity. So we have expanded the chemical space for MTB that we can look for, and the method is generic. It can be applied to any pathogen. Now, having said that, this is one part of our lab work, but we also believe and are very well aware of the fact that any new drug discovery project has a huge attrition rate, as has been shown here in the figure. So you start with like 1 million molecules and you won't end up even with one drug, and that too can take several years. So in parallel to this, like investing energy in new drug discovery, our lab also invests time in trying to see if we can develop new methods for drug repurposing strategies. So for the seniors here, they'll know what this is. I'm basically explaining what drug repurposing is for people who are new to this terminology. So a simple example, and this is not by data science, this is by serendipity. So sildenafil was a drug that was being developed by Pfizer for heart disease, hypertension, and angina pectoris. But when they did their trials, they realized that the drug has absolutely no effect on any of the heart conditions. But subsequently, they realized that it is basically was inducing as a side effect penile erections. Now, a side effect then became the main effect of the drug, and Pfizer decided to launch this drug as Viagra, which all of us are very well aware of. It's a very classical example of drug repurposing, where something was not thought to be working in another indication, but now it is being used. So point is that you make a drug and you know it is acting via a specific target for disease 1, but subsequently you also realize that it can have another target that can treat disease 2. So the point is, there can be similarities between the action of the drugs, or there can be similarities in the function of the target that can actually allow you to basically repurpose and reuse this drug in addition to the indication in which it was originally discovered for. And there are several reasons why we believe that happens, and you can read more about it. What I want to say is that drug repurposing, there are many, many methods. There are genome-based, network-based, machine learning-based, deep learning-based, and we actually reviewed all these methods very extensively in our review last year. Today's focus is to tell you a network-based method for understanding graph, like using graph theory for drug repurposing. So what we did here, now this is a simple data. You go to drug bank, okay? When you go to drug bank and you acquire all drug target interactions. So when I say that, what do I mean? You take a drug, see via which target it is interacting, okay? And you draw an edge between them. Now you know this drug works via target 1. This drug also works via target 2, or target 1 can be acted upon by drug 1, drug 2, drug 3, right? Now what this graph shows us, that both in case of individual drugs acting on targets and individual targets being acted by multiple drugs, there is a huge level of promiscuity, which means 1-to-1 interactions where one target is being acted upon by one drug and vice versa, it's only 7%. Rest everything, rest 93%, it's not 1-to-1 interactions. So there are multiple interactions, and we thought that we can harness this promiscuity to predict potential new drug target interactions. So if you actually start looking at drug target interactions in drug bank, there are almost 15,000. and they are a result of 6,000 odd drugs and 4,000 odd targets. And of this, there are 49 mycobacterial proteins in this list already. So what we did, we simply did one thing. So you have heard of gene ontology. So we mapped each target to their gene ontology hierarchy. And wherever the leaf nodes are actually similar, we generated another edge between the drug target interaction pairs, going from 15,000 DTIs to almost 1,74,000 DTIs. So basically we enriched the network just by not looking at the name of the target, but we looked at the function of the target. So the geo that we used is molecular function. Assuming that if the function is same and the drug is binding at the functional part of the protein, which we didn't do any structural analysis, this is a hypothesis that we, this is an assumption that we made, and we actually made the network more dense. And once we made this network more dense, we added the two networks and then removed duplicates. So we actually had a final DTI network of 26,000 odd nodes. Now, I will not really have time to explain how did we apply network-based inference method. The only thing that we need to understand here is that there are ways in which information gets passed on and is received back by each node in a network. And this is what NBI suffices on, like it actually is built on that logic. And once we actually applied NBI, as you can see here, all the red dots here are new interactions that we identified from NBI. So one is geo, added new networks, and second NBI added further new networks, which with a very high score. Now, when we did all this exercise, we came up with a certain set of targets, which are like 377 new DDTIs and 233 new drugs. What we wanted to ensure that any of these targets, which is either not essential for mycobacteria, or has a role in drug resistance, or is present in human, should not be taken forward. So we applied those three as filters and came up with a list of these 10 targets, which I'll show you the list in the next slide. So as told you, we applied a combined evidence approach, essentiality, role in drug resistance, because that is what is required. You don't want a target that is contributing to the resistance profile of an organism. And homology with protein led us to 10 targets. And if you look at the drugs predicted against these targets, they actually are with reported indications of being antiviral, antifungal, bipolar disorders. Some of them are nutraceuticals or obesity drugs, which practically means now we can actually repurpose these drugs to be tested for these individual targets for applications in tuberculosis. And I'll not again describe in details, but when we started mapping these individual targets to different pathways, some of them belong to pathways that are linked, like say, for example, from corismate to folate or menaquinone. So you actually can identify combination of inhibitors also from the study that we have done. One thing, if you remember, never in the analysis we looked at the structure of the protein or the chemical compound, right? So what we wanted to also do some kind of validation to say what we have received or what we have in the end of the exercise, is it even meaningful? So that is when what we did, we looked at the predicted, so basically this is the known DTI and this is the predicted drug. And when we did a structure-based similarity at a very high Tanimoto coefficient, we could see that for some targets, the known drug actually has structural similarity to predicted drugs. Now, this is interesting because this information was never fed in the algorithm. But for certain other targets like DART or INHA, there is absolutely no structural similarity. But at least in 5 cases, we found it. So this gave, this actually gave us confidence that the method is working fine. And despite the fact that it is not structurally similar, the new predictions are not structurally similar, they are offering us new molecules to work with. Now, given that the first 2 sections of my talk focused only on small molecules, it is for the benefit of the students out there. Small molecules are not the only way to address antimicrobial resistance. There are several other things, and these are not proposed. Most of them are actually in clinic right now, and some of them will be clinicking very soon. So there are bacteriophages, methods of plasmid curing, CRISPR, probiotics, antibodies, immunomodulation, and peptides. All of this can be used for antimicrobial drug discovery. And what we did in our lab was to use peptides. And I'll show you where. I've been talking only about MTB so far, but if you talk of antibacterial resistance, it is not just TB. It is Enterococcus, Staphylococcus, Acinetobacter baumannii, Klebsiella, Shigella, and all of these have been nasty pathogens. The problem is not just in infections. If these pathogens cannot be treated, anything from diabetes to chemotherapy to surgical procedures to transplantation will fall flat, because all these procedures require antibiotic prescription. And these pathogens are making it harder to treat even a routine surgical procedure done properly. So it's not just infections, it's the entire healthcare system which is at stake. And the primary cause are not the planktonic or the free-living organisms, but the biofilms. And if you look at literature, most of these organisms, they account for 99% of microbial life. So biofilm structure, biofilm is nothing but, as we stay in colonies and we think it is safe, microbes also like to stay in colonies because it provides an added advantage of being safe, because then they can share their armor to fight antibiotics. So, and just the take-home message from this slide is that a biofilm can be up to 1,000 times more resistant to conventional antibiotics. So you know why do they stay there? They stay in a structure, which I'll show you how it looks like. But there are certain properties of a biofilm. First, recalcitrance, which means what? The minute the biofilm breaks open, the residents of the biofilm again become susceptible to the antibiotics. When they are in biofilm, these antibiotics don't work, but when they are out of the biofilm, the same antibiotics work. Second, the mechanisms are not the same. And third, they apply several different strategies, which I'll not go into the details, but where it matters is because almost 70% of nosocomial infection, 99% of urinary tract infections, 80% of ventilator associated pneumonias, 87% of bloodstream infections, all of this is caused by biofilms. So there, if you talk to clinicians, they'll tell you how big this is a mess and how do we address it. So this is basically to show you how a biofilm forms. So free-living, basically planktonic versions actually come adhere to a surface, start producing something which is called as an EPS. And when the EPS forms, the biofilm forms in this fashion, where there can be mostly more than one organism in the biofilm. The outer structure versus the inner structure is different. The inner side of the biofilm is more inactive organisms, the outer side is more active, and there are water channels. So basically, even in biofilm, it's like an entire ecosystem in itself. So if you see here, the metabolically active cells are susceptible to fluoroguanilins, whereas the slow-growing cells are susceptible to lipopeptides. So the way of susceptibility of these cells as you go from central to the outwards is not the same. And just to go forward, what do we have to destroy them? So our own body actually produces antimicrobial peptides that can help us find biofilms and can give us indications of that peptides can be one way of disrupting the biofilm structure, and that is what we did. We looked at a few databases like PAMS and Curumpeps, and we generated a repository of biofilm active peptides that work on all these stages. And then we generated a biofilm disrupting peptide as our initial positive data set for machine learning, and biofilm maintaining peptides as a data set, as a negative data set. So this is a very unusual way. Normally you would have taken a biofilm active versus an inactive peptide. We didn't do that. We took some peptides which were maintaining the biofilm versus peptides which were disrupting the biofilm as the negative and the positive data sets. If you go to the paper, you can read more about it. And then, as the previous professors have told you, how do you basically submit labeled data to a machine learning model and give new data and try to see if it is able to classify your unlabeled data correctly. So in this case, we generated, as you can see, both SVM and Weka-based model on feature selection. Professor Ranganathan has already told about that. And we basically identified whole amino acid as well as certain specific amino acid as features that are contributing to antibiofilm activity with a very high accuracy. So this is the first model and the best model that one can use today for prediction of antibiofilm peptides. And in order to make our application more relevant, what we actually did, we took 11 FDA-approved peptide drugs and 9 antimicrobial peptides in clinical trials. And based on that, we made a prediction. So if these are basically, since we have 6 models, these are all the predictions coming from at least 4 of them. So the point is that there are multiple models which are saying that this has an antibiofilm property. We have recommended it. And if you look at the way the original indication of these peptides is, most of them, we know these indications are because of biofilms. So the point is, you don't have any anti-biofilm peptide in clinic, but all the antimicrobial peptides in clinic actually have a biofilm phenotype. So this actually also gives us confidence that what we predicted or observed has some meaningful outcomes. And this particular work is very close to me because this is Pooja. She's a co-first author with in the paper. And at the time of publication of this paper, she was just finishing school. And now she had then later on became the Gates Cambridge Fellow. So this is basically for all the students out there. There is no age to do science. You don't need degrees to do science. It's your interest. And she's really, really brilliant. And now she's doing her PhD at York University. And I have several such examples like that because I work with school and college kids extensively. And this is, again, I'm not going to details. This is our response to the pandemic. We cannot just not contribute to what's going on with COVID-19. So we realized that there was a huge problem because everybody is reporting inhibitors of COVID-19, but nobody knows what chemical space are we looking at. So we actually came together and developed this open repository. It is accessible online. And this is an entire set of team that again came together to build this one. I'm not describing it today because this is only a pointer for some of you to join something which the government of India is doing. It's called the Drug Discovery Hackathon for COVID-19. I'm leading the Drug Discovery Virtual Tool Room along with several partners. And all the members out there, please go try, see if you can help address these problems. And this basically shows that my lab has been working extensively using crowdsourcing as a mechanism, like engaging as many number of experts as possible in several projects over the years. And as I told you, I really like working with the younger community, either it's wet lab or dry lab. And this is what my lab has been doing for a very long time. And most of that has translated into publications. And the point is that it is not about only outreaches, it is about training how to do serious work. And that takes time, that takes commitment, it takes effort. So be ready to put in that. And if you can do it, we can make some meaningful contributions. And last year I set up a lab in one of the INSERM units in Paris. And so this is basically all my postdocs there, a few students, and this is my lab in India. And again, a few colleagues from Joggle and LWB. These are the pointers where you can write to me on Skype, Twitter, or my email ID, and I'd be very happy to talk to you. Just one last thing about my institute. I belong to the Institute of Microbial Technology. I'm not sure how many of you know that, but recently a global impact analysis was done by Database Commons, and IMTech is at the 4th position in contributions of the databases that we have made to the bioinformatics community. And this is the campus, and had it not been for the pandemic, I would have really invited you all for a physical visit to this place. And with that, I would like to thank you, and we'll be very happy to take any questions.
Thank you, Ansu. Thank you for a very nice talk and a very, very comprehensive talk in a way that you presented a lot of areas of your research and what your team is doing, and you have very wonderful big team. And then now I'm looking forward for some questions. Question, please. Let me see in YouTube. Sometimes I have to refresh the YouTube. Yeah, there are some questions. Drugs can be thought as a molecular graph. No, this is not a question actually. So
okay.
Nothing. Please raise your question. We are live on YouTube as well, and we're watching you.
I think I was a little quick. I hope if they can watch the video again, they can get a few concepts. It's easy for senior or experienced members to understand everything, not for the younger guys. But yeah, I hope they can watch it again. And if there are questions, I'm available all the time.
Yes.
Yeah, actually, I have a question. It was nice talk. So my question is, when you did I saw that your data is somehow containing positive, negative data, and you did the resampling or whatever cross-validation. So in the end, how you rank the features, the significant features in your model? Because each time when you did the cross-validation, you're supposed to have a slightly different list of significant features. So in the end, you need to aggregate and find the optimal list. So how you did rank all these or aggregate all these lists together?
Yeah, I understand your question, Dr. Malik. See what we did. If you read the paper, so feature selection we did by using Beka, and in Beka you can select one or other classifier, the feature selection algorithm. What we instead did is we brute forced. We ran every algorithm available in Beka for feature selection. Then we did an estimate of what is the MCC or the true positive, true negative, that confusion matrix along with MCC. And instead of making a decision on that random forest we should use, or we should use J48, or we should use something else, we used everything. And whichever gave us the best result is the model that is in the paper. So instead of making a decision on how do we select features, we used every algorithm available to us and brute force the feature selection on that basis. So it is coming from data. It is not something that we intervene at any point saying, I would like to take this amino acid versus that. The final observations when we have what we have from these models are that, of course, the antibiofilm versus the quorum sensing peptides, the negative data set, there is a difference in the propensity of amino acids both at the N and the C terminal, as well as most of the antibiofilm peptides have cationic properties, which means that they can penetrate. And that is why probably they're able to disrupt the biofilms. So I hope I have answered your question.
Yeah, thank you. I see that you are using Weka. I would suggest to you to make yourself familiar with the K9. Actually, I will try to present it during my work. As actually we used before Weka, but recently K9 is much powerful tool that you can integrate different, even Weka there also you can use it. But it will be very interesting that your lab become familiar with these tools. Just a suggestion. It's my suggestion.
Oh, thank you so much.
Sure.
Yeah, so let me see if there is a question. YouTube. Sometime YouTube is slow here. So let's see. Brinda is again asking for clustering, which algorithm was used? So can you hear me? Brinda is asking which algorithm was used for clustering.
Clustering in which project? I presented 3 different works. I'm sorry.
It's not clear, Brinda. You can re-raise your question. But in between, there is one more question for antibiotic resistant. The natural traits of
Can you all hear me? I don't hear Abhishek.
The question is. Oh my God.
Oh, you are saying this question, AMR is one of the natural trait of bacteria,
Yeah,
and
there
new
is a
antibiotics
question. Yeah.
can also become resistant. So how can we tackle this problem?
Yeah, that's it. That's
So
it.
the point is very simple. Use what we have judiciously. You know that no matter what amount of antibiotics you are going to discover, and believe me, one discovery takes too much effort. So best case scenario is to use them judiciously, which means targeted, underuse, overuse. All 3 have to be very seriously avoided. And of course, public health interventions are critical. Of course, you need to take antibiotics in some scenarios where none of this really works. But the point is, judicious usage can really control this issue. And it has been shown in several studies way back in 1999 also that the minute you start using your antibiotic properly, the antimicrobial resistance pattern also changes accordingly. So it's a very real life response that you get. So judicious use is the only way out. And of course, right now, what is being tried is probiotics. If we understand the composition of the human host well, we can basically customize it to fight infections. But again, a long way to go because we don't know we really have not mapped everything in the human microbiome so far. So a lot more studies have to be done there. And again, it's a huge data intensive science. So I would recommend you to follow the probiotics vertical as well. And yeah, the only way is to be judicious in use.
And then, yeah, so if there's no more question, I will like to, and Sobha is giving a compliment to you. So it's a great talk, and thank you to you. And yeah, so this is our uh, effort in pandemic to keep ourselves active. And yeah, so thank you, Dr. Anshu, and thanks for joining both the days, yeah, because it's from your hectic schedule. And so the students and trainee, and even from my group, they think we can do something together. Just reach out to Anshu, and then I saw that somebody wanted to join my lab, join your lab.
That's good.
So,
Thank you so much, Abhishek. And I really see this as an opportunity for us to see
yeah.
how do we work together. Yeah.
So she was shy in telling where she joined, but she told me the Chandigarh. But yes, so tell her there is not a problem, and it's in life it happens. So people have to take better choices, yeah. So and choose out of two, right? So that's with this we'll end the session, and we'll have a virtual coffee. Mike and may ask if you are I heard that many places, I just come to know that many places again offices are getting closed in the across the country, and different labs are getting closed today. So we'll have shorter break maybe, and then we'll start the session with a new set of speakers, a new chair. So let's have coffee, please. We'll have a break. Yeah. So let me see just the schedule again. Okay. So yeah, so we have the next session chaired by Dr. Pankaj, and then keynote speaker Professor Devnath Pral from ISC is already here with us, and Prash and Yusuf Malik is speaking as a keynote speaker as well at the number 3, because he needed some time. Initially, we thought we'll keep him together with Professor Devnath Pal, and yeah, so we'll have some minutes of break, and then we can start 11:30. Oh yeah, we are running late actually. So can we make a 2-3 minutes break, a biological break, and start if you have your coffees in your hand. Yeah, so we are running out of time actually, so we'll start session in 5 minutes maybe.
Shall I share my screen?
Yeah, you can share, sir. Dr. Pankaj, are you there? Yeah, he's there. So sure, sir, you can share your screen. Yeah.
Yeah, so I am here. Thanks.
Yeah, please. Yeah, let's without further delay, because the speakers may have to go to the next meeting or so. So let's go ahead.
All right. So thank you, Dr. Abhishek. So let's move to the next session now. We have a another keynote speaker with us, Professor Devnath Pal from Indian Institute of Science, Bangalore, India. Today, he's going to talk about extending the horizons of biological function inference, leveraging protein dynamics. Professor Pal has obtained his PhD in structural bioinformatics from Jadavpur University in Kolkata, India. He's currently working at the Department of Computational and Data Sciences at ISC Bangalore. His areas of interest are in research spans from genomics to proteomics, metabolomics, structural biology, drug discovery, algorithms, and methods, with a focus on addressing the biological problems through computational biology and bioinformatics approaches. His methods include the uses of data from NGS, mass spectrometry, X-ray, crystallography, NMR, and other biophysical techniques. His lab works on biological problems that attempts to understand the biological function at various scales through development and integration of new methods and techniques. His recent project include inference of protein function from molecular dynamics, genome secondary analysis, hemoglobin structures and functions, and so on. So yeah, we are very fortunate to have Professor Pal here on board, and we really look eagerly and excited to look towards his talk. So Professor Paul, please, stage is yours now.
So my video feed is there.
Yeah, we can
and
see it.
my slide is also there.
Slides are also here. Yes, sir.
Great. So shall I get started?
Yeah, please.
Okay. So I thank the organizers for giving me this opportunity to speak in this virtual symposium. Thanks to Abhishek and others in the team. So my talk today is on extending the horizons of biological function inference, leveraging protein dynamics. The whole idea of giving this talk is to try to simplify things. We know, we live in an era where we have a lot of data, and coming up with methods that are simple enough to understand things is the need of the hour. Otherwise, we will get overwhelmed with data. So the outline of my talk first is that I will tell you the motivation for whatever I'm doing, the key concepts involved, the implementations and the applications, and then some conclusions. So if you have a protein, and you want to determine its function, so typically what we do, so you may have a sequence at your hand, you may have a 3D structure, or you may have gathered some high throughput data. So if you have a sequence, you may want to do some alignment. The alignment could be global, local, or you might want to use some genomic context based method to infer functional linkage, or you may want to use some features and then leverage machine learning. And you can get subcellular locations, functional class, and so on, which allow you to understand what's going on with the protein. And similarly, if you have 3D structures, you'll have again global and local, matching, and then you would have a 3D motif, or you can also leverage structural features here to train your algorithm and come to some inference. If you have high throughput data, then gene expression or protein-protein interaction information can allow you to gather new insights into the functionality of the protein. But if you look at whatever we have been doing, there's a catch in it. The conundrum is that the protein function is a biological activity. So what is an activity? Is actually a dynamics in space and time of the protein molecule. And it is governed by the physicochemical properties of the parts in action and the surrounding environment. But given that we have a limitation, we are currently only using static information to infer function. And as in the case of protein, where you're leveraging, primary data or the tertiary data or the quaternary data that's available with us. And we typically use what is called the sequence structure interaction, linkage to understand function. And whenever motive information is available, we try to leverage it as well. So this is currently where we are. And you may be knowing that, that 98% of whatever we understand about functions of proteins is computational. And this is, as per the latest available information in the GEO database, and only 2% is experimental. So understanding function of new proteins, trying to find out more functions of what they do is very important. And I'll show you, why, dynamics is so important. So this is one simple example that is at hand here. So we have a structure for an avian defensin protein. This is from chicken. So the defensin protein is the NMR structure for the wild type on the left-hand side. And you can see this protein here, it's very flexible and it has the activity that you'd expect in the wild type. It's available in the PDB database to LG5, and there's only one single, mutation from lysine to alanine, and you can see that the fluctuation has entirely stopped, and the activity is almost gone. So this tells you, that the flexibility of the protein is very important for function. And the region where this mutation happens, where this alteration happens, determines how critical it is. Now, if we look at only structural perspective, we may not be able to gather simply by looking at the mutation K to A. Most of us typically, when we do mutations, we try to replace any amino acid to alanine, hoping that that mutation will not alter the structure, so that we will know the function. So this is not straightforward. There's another example from work that we have published recently in scientific reports this year. This is from a serpin protein. So we have a single mutations, and these mutations are all seen in patients. So the serpin is an antitrypsin protein, which is metastable, but the wild type protein, is not so prone to aggregation. But when you have a single mutation, for example, glycine, sorry, not a glycine, the glutamate to lysine, it becomes pathogenic, which means that this protein is no more, very stable. It aggregates, and you get a disease which is called chronic obstructive pulmonary disease. Recently, we have identified another mutation from glutamate to aspartate, and this mutation is highly prevalent in Kashmiri population. And we have found that this supposedly benign mutation, because you can see there is no change in the functional group, both are acid, but still the protein is prone to aggregation. So these kinds of analysis is very difficult when you have only sequence information, and unless you do dynamics kind of analysis, it's very difficult to understand where the changes are and how important they are. So as you can see in this plot here, I have shown you the root means the dihedral fluctuation of the backbone and the different secondary structures in the 1 microsecond simulation. You can see one change in amino acid, and there are several parts of the protein structure which are different. Most importantly, the electrostatic signature of the molecule also changes. Uh, Another interesting aspect that one would like to keep in mind is that there is another class of functions which we typically uh, don't bring to our attention regularly, but are very important, which is called the moonlighting functions. So what are moonlighting functions? Moonlighting functions are functions that are done by the protein other than its main function that it has inherited from its ancestors, which means the orthologous function. For example, you have E. coli put a protein, it does a different function when it is bound to the cell membrane as opposed to a DNA. There's another example. The enzymatic activity uh, is different when it's inside the cell, but this converts to a growth factor when it is secreted. The protein in C has a different function when it is in an endothelial cell as opposed to a neuron. Similarly, if you have a different substrate bound, proteins can switch activity. If the multimer or a monomer in different oligomeric states, it can have different function. Similarly, different complex formations and different binding sites. So the number D, E, F, and G are more commonly known, but obviously A, B, C, D are not so common. But the emerging view is that the proteins are versatile, so much so that when they change their location, the environment change, they can, recruit different functions that is present inherently in the sequences that from which they are coded. So the quest for understanding protein function from protein structure is not new, right? Many have tried to do this, and one of the papers published about 8 years back tried to do this in a comprehensive manner, where they tried to find out what kind of analysis would be suitable, trying to link protein structure and function. So this is one of the most comprehensive attempt to link the dynamics to function, and where they tried to decipher 34 features, which means there's a 34-dimension vector to match the dynamics of two proteins. So typically these dimension vectors characterize the eigenvalue spectrum of the protein, analyze the principal component of the trajectory, the ruggedness of the free energy landscape, and the atomic fluctuations. So these attempts have been ongoing. So we also started our project, more than a decade back. My graduate student who joined in 2009, I gave her this problem, and we decided that we'll pursue such a project. So the first thing, the first concept that we had in our mind is that the functions of proteins usually reside in the flexible regions of the protein molecule. And so which means that we have to look at it locally. And also, because we want to annotate novel proteins, our method has a non-evolutionary basis. And the dynamics, because it can relate to all parts of the protein, it's also a holistic aspect. As you can see from the right-hand side, this figure here, when we do, try to see the time scales of different motions that are ongoing in the protein, the bond vibrations are at the picosecond level, the side chain rotations at nanosecond level, the loop motions are in the microsecond level. So because the loop motions are primarily, determine the functions, So we aim to do a economical microsecond simulation. And in those days, doing microsecond simulations were still hard, not so much so with the advent of high-powered GPUs. But to fully conquer the collective motions, we should be able to go to millisecond level. So when we started this work, we faced several bottlenecks. Okay, so the time scale of motions that we were trying to deal with were microsecond and above. The time taken for each simulation was large, and because the computing capacity was limited in those days, GPUs were not that fast or are not so much available as well. The cost was also very high. And if you were doing a large scale simulation of 1 microsecond, the amount of the data that you acquired was difficult to store. I mean, those were the days where you did not have 4 terabyte disks, in a commodity scale. So we had a solution that we tried, which was coarse graining the whole thing. So what do you mean by coarse graining? When you do coarse graining, essentially it means that you replace the protein molecule, each residue with some pseudoatom. And based on this pseudoatoms, you can represent your protein using those pseudoatoms. In this slide, I'm showing you that this, through the cartoon, that you had all atom model on the left-hand side, and you just use the C alpha atoms. So you will get a representation just like a chain of beads connected together in space. So to simulate such systems through pseudoatoms, you need a specialized force field, and you would not be able to use the normal charm or amber or an OPLS force field. So in those days, we did not have many force fields. So we tried to develop our own force field, which is called the coarse-grained molecular mechanics force field. And Martini was available in those days. So we benchmarked our work against Martini. So the works essentially led to building of potential functions, which more than 1,000 in number. I cannot go into the great detail of these potential functions that we derived, but they were made in such a fashion, they were crafted in such a fashion so that they matched the NMR ensemble structures. So we validated, we took the NMR structures from the PDB, and we ran molecular dynamic simulations using the CGMM force field. And from the trajectories, we deciphered the fluctuation graphs. We tried to match these fluctuations graphs with the NMR ensemble data. And so this is one example that we have on the right-hand side for ubiquitin, where you can see the NMR structure is given by the dashed line. We have the CGMM, our force field, which is closely matching with the NMR RMSF root mean square fluctuation data. The Martini, which is fluctuating more than what it should. So we did these kinds of analysis to ensure that the force field that we derived, faithfully represented the physiological fluctuation that we would be able to expect from the protein in the solution. And we have now this, the current state of this force field is such that we are able to do protein simulations for monomers and multimers, but we still do not have functions to incorporate ligands in it. So those are the new areas that we would be still working on in future. So once we finished, developing our force field, then we started, you know, trying to figure out how we could go about using the trajectory that we got from our coarse-grained simulation and decipher if we can make a direct match between dynamics and function. So the first test that we did used a diverse data set of orthologous acyl-CoA binding proteins. And for these proteins, we also had the experimental binding and affinity information. And as you can see, the set of proteins that we took was from 21% to 82% sequence identity, which means there were sequences which also had a very poor match. And for those sequences for which there were no structural models, we build them through templates. Now, an advantage of using coarse-grained dynamic simulation is that you don't need to do any sidechain modeling for the protein. So it becomes very easy. So if you have the template, it's just replacing the amino acid names and just keeping the C alpha atoms and then joining them. So if you can look at this next graph, so after we did the simulation for 1 microsecond for each of the proteins in the test set, the box here is the place where the binding of the acyl-CoA happens. And as you can see, the chain length of the acyl-CoA is given here. So the bovine, the yeast, the M. perniciosa, and the rat has a medium long side chain. And the falciparum and the T. brucei has short chains. So if you look at the graphs here, the shorter ones are highly flexible, whereas the ones which are medium long, they are clustered together here. So this was the first test that gave us hope that whatever we were doing using the RMSF was useful. So the goal was to see, if we could identify all the flexible regions of the proteins and then use the flexible regions to actually match and see, which flexible regions of a given protein match with each other, and if they would, give us some kind of a signature. So the task of matching, first we need to demarcate flexible regions. So what we did, we discretized the numerical values of the root mean square fluctuations for each residue that we got, and then we converted them into a string. So what happened is that when we converted all our RMSF graphs for each protein into strings, we could quickly do what is called a local search, a string-based local search, to identify all such substrings which we are matching. And we then empirically, figured out that if we had a criteria where the symbol L was greater than 35%, or a combination of G, H, or I was greater than 14%, we could annotate these regions as flexible. So once we have identified all the flexible regions of the molecules, then the goal was to see if, if we take a library of proteins and for each protein we know all the flexible regions, now which flexible region would match with each other. We used something called an autocorrelation vector. We could have used the traditional way of eigenvalues to also do this match, but we prefer to use an autocorrelation vector because this is a concept that we borrowed from cheminformatics, where it is highly used and it is very simple to compute and also very fast. So if we just forget about all the formulae that are written above, and just focus on these points that are shown here. So assume that you have a small segment, and these are the C alpha atoms in three-dimensional space. So what you need to do is essentially you just find the distances between all the pairs, and then you decide what will be the span or the bin for your vector. So for example, 0 to 2 angstrom, 2 to 4 angstrom, 4 to 6 angstrom, 6 to 8 angstrom. So simply you divide the space into certain bins, and you count the number of cases that fall into these bins. So then this becomes your autocorrelation vector. It's a very simple computation and very fast to do. So what we did, we computed the 3D autocorrelation vector, the unweighted vector, for each of the flexible regions. And then we did a matching based on that. So here is one example here. So you can see this protein here for the whole 1 microsecond trajectory. We have discretized and and extracted frames for every 100 nanoseconds, which means that from the starting 0 and to the 1 microsecond, we have 11 frames. So you have 11 such vectors computed here. Similarly, for the next protein here, you have such vectors here, and you have such vectors here. So now you simply take these vectors and compute a simple correlation coefficient to get a matrix with 121 elements in it. And you can see that if two proteins have a good match in a certain segment, then you will get a very high match. And if they don't, you will get a very poor match. So in this case, this protein has a match with this protein. So you get a correlation matrix with many elements having a very high correlation, and which is not true for the other. So what we did, we found out the thresholds. So this was the criteria that we had. So if you had 25% of the elements with correlation value greater than 0.95, and at least 50% of all the correlation values greater than 0.9, then it would deem that the two proteins have the same dynamics. Or if the total number of cases greater than 25% is counted as Y, and these are counted as X, you could also calculate what's called an Euclidean distance to get a value. And using both of these, one could decide what is matching and what is not matching. So initially we took 60 arbitrary proteins, including moonlighting proteins, and we figured out that all of these have also experimental data available. So we took these, and what was encouraging for us was that when we found out the true positive rate, it was 87%, and the true negative rate was 92%. So with this, we kind of established a framework where we knew that we could work more with this method. The interesting aspect of the work that we did was that it allowed us to identify functions of proteins that are matching, where you would never expect to get any insight if you did a sequence-based search, or even a structure-based search. For example, you can see on the right-hand side, we have a protein where both are potassium channel inhibitors, and the global match is 9% only. And only in one section of the structure where the autocorrelation vector matches, you can see that this is the most important region due to which this potassium channel inhibitor is working. So you can see that the secondary structure is helical here, and this is kind of a loop that is there. But apart from this match, there's nothing else that will suggest that these two are potassium channel inhibitor. Similarly, we got matches where the secondary structures were different, and this is binding the vitamin pantothine for dihydrogen phosphate. So in this case also, it would be very difficult, just if you did a motif-based search also, to figure out that there's a match here, because the motifs won't match as well. But obviously, when you do a dynamics, the autocorrelation vectors match. Now, for finding the moonlighting functions. So for finding the moonlighting functions, you can see an example here is for 1WZT. In this case, the ancestral, function is the biotin ligase, and the recruited function, the moonlighting function, is the DNA binding. And if we did a match, it was known, though both of these functions were actually known experimentally. But when we did this dynamic space search, we could also see that it could match to 2EJ9 for the ancestral function and 2L0K for the L0K for the DNA binding. So this way you can find both the ancestral function and the moonlighting functions. And if there are other functions as well, you would be able to find them. So we try to leverage these analysis in our work that we did subsequently, and I will try to walk you through some of them quickly. So the first work that we did was to see if it helps us improve function annotation. So this is a schema. So what we did, we wanted to see that if there are proteins with cis peptides for which we know the functions, if we could leverage this there. Now, cis peptides, as you may know, are a very conserved feature in the protein, and because cis peptides are functional. So we did an experiment where we just took the cis peptide segment and just used a sequence based search, and then we took just the dynamics of the proteins, and then identified the autocorrelation vectors for the cis peptide segments, and we independently did the function annotation, and then we saw that what happened, and then what happened if we combined it. So when we did that, so this is the summary of the results. I'm just, for the sake of time, I'm skipping all the other details. So we had 204 such cases for which the experimental data was also known, and the detailed functional analysis was available in various published papers. So when we did the sequence only analysis, the true positive rate was 0.72. When we did dynamics, it was 0.88. And in this case, we did not use the Euclidean distance, but just the thresholds that we determined. But what you would see that when we do the whole accuracy, so ACC stands for accuracy. So when the overall accuracy we did for sequence based approach, it's 0.78. This is marginal, improved to 0.80, but when we combine it, then it went to 0.88. When we use the Euclidean distance based criteria, so this is a little bit more stringent. So we find that, although the number of cases have come down from 154 to 138, we have a more accurate, answers. So which clearly told us that if we use both kind of information, if we mix them together, then the chances of improving our analysis is quite significant, because 0.78, 0.79, and 0.92 is quite a significant improvement. Another application that we did was through a blind test that we participated in last year, which is called the Critical Assessment of Genome Interpretation. It's called CAGI. This is an Olympiad similar to Capri and CASP, many of which some of you may have participated. So in this Olympiad, you are given proteins and their mutants, and then you are asked to tell which of these mutants are altered. So there's an altered phenotype. So the question is, just the genetic mutation lead to loss of a certain kind of function? Okay, so that's the whole point. There were many other targets. So I'm going to, for the sake of time, I'm going to only tell you about two. So the first is a challenge in which you have to predict the alteration of the phenotype for 34 single mutants. Okay. Now this is a protein called CHEK2, which is a kinase protein. It's a very large protein of 586 amino acids, for which patient data was available for 34 persons, and one had to tell whether these which of these mutant patients had a cancer or did not have a cancer. So basically, from these patients, these genome sequencing was done, and these mutation information was accessed. Now, CHEK2 is a very important protein because it is known to maintain genome integrity and also regulates the G2M cell cycle checkpoint. And also, it is required for the activation of the DNA damage repair and apoptosis of cell if there's a DNA double strand break. So any malfunction here leads to tumor and subsequently cancer. So this was known, and because this is a very important protein, so this was given as one of the targets. The second target that was given to us was for calmodulin. This is a cell survival protein. And for this, again, the data was available from 1,719 patients. So we had data from so many patients. This protein is also known for cellular functions such as signaling, division, differentiation, apoptosis. And it has been found that the variants, there are certain variants of CAM that alter, they alter such that, they are present in heart attacks, cardiac arrhythmias. So we were tasked to, predict the phenotype change in these two proteins. So this was our pipeline. So we did molecular dynamic simulation wherever model building was necessary. We filled in the gaps, and then we identified the flexible segments, and then the correlation coefficient was obtained through the autocorrelation vector, and we calculate the probability score or the similarity score for the CHEK2 and the CAM1 respectively. So the probability score is essentially the correlation coefficient, and the similarity score essentially identify the number of flexible regions in the mutant that were functional to the total number of flexible regions in the wild type. So this was the result that we got. So this is experimental results that was shared after the challenge got over. And you can see on the left-hand side, these are cancerous and these are benign. So, and this is the probability score that we predicted, which means that these cases we predicted correctly, and these cases we predicted correctly. Then this is the wrong prediction, and these are wrong predictions. So that is kind of summarized through some data here, where we have calculated the Pearson correlation, and we have staggered the fractions. So you can see that for about 2/3 of the cases, we are doing fine, which means there's not much difference between the predicted, experimentally predicted scores and the theoretical score. So, and if we relax the criteria, then we would say that, about 82% cases, we have some fair bit of results, but 18% cases, our results are anti-correlating, which means you can see the correlation drops from 0.47 to 0.02. So these mutations, we just, opposite predictions were made. So we have also done ROC plots, and you can see that we do have a positive signal for 75% cases. So for 75% cases, this is the graph here, which is a reasonable prediction. What is interesting is that when we compare our results with polyphen, so polyphen is one of the cases where you have machine learning involved. So we don't do that bad. So F1 is a classifier based approach. So we do 0.7 and they do 0.76. The accuracy is 0.6 and 0.7. So we do similar analysis for the CAM1, and I will not go into the detail of this for the sake of time, and I'll summarize the overall result. So in this case, as you can see, the accuracy for us was 0.73, whereas for polyphen, it was only 0.49. So you can see in this case, we have done much, much better than polyphen, which is a machine learning based method. And even our classification score is much better. Now, one can ask that why we are doing much better here, despite very a simple way of analyzing. So these are some of the things that one needs to go back and look deeply, where the functional parts are of the protein, and which helps us connect to the phenotype. So that was two examples. That were two examples that we presented for the KAGI challenge. The last one I will present is for a metal binding site prediction. As you can see, when you have a metal binding site that is absent in a protein, and you would want the metal binding site first to be deciphered before you can annotate function. So such kinds of methods that we have developed becomes very handy. So here are two examples. This feature plus is for calcium binding, and FoldX is commonly used for predicting calcium, copper, and zinc binding sites. So we have done built a method for predicting for these 5 binding sites. So currently, we in the PDB, about 62% cases are metal free and 38% are metal bound. But many of these metal free cases could actually have metal binding sites there, which, if annotated, would help in finding true function. So this was our motivation, and this is just this is just pictorial to suggest that when we have dynamics information, then we know which ligand will fit exactly to which pocket, because when you do dynamics, the pocket shape changes, and such that which pocket will fit which one, which otherwise you may not have unless the static structure is exactly matching with the ligand binding pocket. So in our case, we have have a flow chart. In the flow chart, there are 2 sides. One is where you prepare the data base with the template library, and in our database we have these many structures which are incorporated. The total is given here, and this is the test set here. So what we do is we first identify the metal binding sites, and then use 4.5 angstrom radius, identify the residues, and then we have a question there that whether this is specific binding site or not. A specific binding site is one that is functional. So we have used OrthoDB database to find out which sites are specific and are functional sites. We take those and we create a template library using geometric hashing. So once we have done this, then essentially uh, we have to take the query protein when we do the dynamics, and then we take the sample frames, and then we use the autocorrelation vector and we do the matching. So once we have done the match, then we enforce some ligand binding specificity on the matches, and that helps us to get the final prediction. So this is a paper that is currently under peer review. So this is a quick overview of what we do. So you have like 4 pseudo atoms, and in the center you have the metal atom. So you essentially store information for these 4 pseudo atoms, and then you do this for all data in your training set, and when you get the query, you build these things again and then you match. So that's what you do. So this is schematic for that. So you have initial configuration, and then you do the dynamics, and then you will get all kinds of residues, which will be extracted from these conformations. And once they are done, then you enforce the ligand binding frequency. So this ligand binding frequency is pre-calculated based on the known propensities for metal binding, and a score is calculated. So this is the comparative result of what we have done so far, and the results for the test. So for this zinc, you can see the comparison is being made for 2 methods called MIB and IronCom. For those of you who are working in the area, you may be knowing about these 2 methods. So you can see that for the data set that is chosen for test, so we do 79 and MIB does 62, and this is 75. We do very good at 95 for iron, and they do 81 and 74. For copper, of course, MIB is doing better than us, and IronCom is 77. For magnesium, IronCom is better. So we can say that for copper and magnesium, we are not doing as good as superior, not much superior than the others. But for copper and iron and zinc, we seem to have a fair bit of advantage in the method that we are using. So my time is up. So I will summarize what I spoke about today, the take-home lessons. So what we saw is that if we do simple analysis of protein flexibility to MD simulations, and we pass it through certain filters like autocorrelation vector, or you might want to use eigenvectors also, that will allow you to match subsegments based on flexibility, and that'll help you to understand function in a much more robust manner, and which can be extended to understand phenotype and then diseases. So you can mix and match, you can incorporate this in your pipeline to improve function annotation and metal binding site predictions. This is a great way to understand moonlighting functions of proteins, and not the least, coarse graining has helped us to do these things economically, because if we were to use all atom dynamics, then I'm not sure if we would have been able to run so many thousands of MD simulations. So totally, based on the early work that we did on coarse graining, we were able to pursue the next set of analysis that I shared with you today. Now to acknowledge the people. So this work was done by 3 ladies in my lab. Pratiti, who started this work, she has already graduated, now a postdoc in Germany. Sritama also has graduated. So she works in a German company right now. Aditi has also graduated. She is working in GE right now. So these 3 ladies did this fantastic work in my lab. And if you want to use this, use the force field to do your own simulations and also try it out. This is available in my website. You can go there. So this is a snapshot of the web page, CGMM. So feel free to download it as a tutorial that allows you to go through the whole thing. And if you have any problem, you can contact me. So with this, I will end. My time is up. I'm sorry I overshot by 3 minutes. I'll be happy to take questions. Thank you.
Yeah, thank you so much, Professor Paul. It was very valuable and resourceful talk for the audience. And I now look forward to some questions. There is one from Shashank, I think. Can you tell me? Okay, no, that's not. Yeah, can you please share your email ID? Oh, it should be there, so that's not. So there is from Sriram Pila regarding the clinical aspects of antibiotic resistance. Is it? It's also probably the last for the last speaker. So if there are questions, please ask. Are there any insights into as cRNA protein interacting sites, such as comparison to metal binding sites? Does your method approach that? Yeah, that's a question from our next speaker, Prash. So Professor Paul, maybe you can answer that.
Could you repeat the question?
Are there any insights into in cRNA protein interacting sites as such in comparison to metal binding sites?
Yeah, so obviously the signatures of flexibility are different for different binding sites. And as you may have noticed that we started the work by trying to identify the differences in the flexibility in the region where the acyl-CoA ligand binds. So based on the side chain length, the flexibility changed. So that has been a very sensitive way of understanding how the sites behave with respect to the size and the physiochemical property of the ligand. So if you have a metal binding site and you have an RNA binding site, or any other binding site, the flexibility signatures are different and these are distinguishable. So that's the power of using dynamics in understanding binding. Does that answer answer the question?
Thank you. That's indeed true.
Yeah.
Thank you so much, Professor Paul. And if we have more questions, please feel free to contact Professor Paul directly on his email. And hopefully this tool, CGMM, will be very useful for your personal research work as well. So with this, we thank again Professor Paul, and maybe we go ahead to the next speaker.
Thank you.
Thank you, sir. Thanks for coming. Please clap hands. And he has made it from his very tight schedule. And so my students keep eye on the sir's work, because we are also working with Serpins, and sir is doing the modeling in an appropriate way, and we do it in a semi-appropriate way, right? So if we need to prove something, it will be good way to approach. Gaurang, keep eye on it, because we are using polypen shift, and then now they prove that polypen shift can be outperformed by intensive structural biology, right? So computational structural biology. So indeed, there is a possibility of a good cooperation. And so my team has, as sir already knows, that my team is new and they are now learning on a learning curve. So we need to also keep eye on it, and then we potentially have to visit once we have some good results from the variant analysis.
Sure, you're welcome.
Yeah. Thank you, sir. Have a nice time.
Thank
and thanks a lot for good talk. And yeah, so I'll give it to Pankaj back. Yeah, Pankaj, please.
Thank you. I'll stop my video.
Yeah.
Yeah, hello, Pankaj and Abhishek. If it is my talk, if I'm allowed to share my screen, please. Yeah.
Neol, can you allow Dr. Prash to be co-host now for now?
Yeah, sure.
Afterwards, Professor Yusuf Malik will present after him.
Yeah, Prash is co-host now.
Yeah, Prash, go ahead.
Yeah.
Pankaj will introduce you. Pankaj, please.
Yeah, so I'm very happy to introduce Dr. Prash. He's currently working as a senior scientist at Birla Institute of Science Research in Jaipur, in India. He's a system biologist with wide interest on functional genomics, protein informatics, and interactions. He has obtained his PhD from Aalborg University in Denmark and have gained 5 or more years of postdoc experience. His current research work focuses on developing resources, targeted genomics regions of interest, and known unknowns, non-coding RNAs linked to diseased genes and proteins. He is also a very active researcher in the field of bioinformatics, and he is also leading the BioClue organization in India and also in abroad. So we welcome to Dr. Prash, and we look forward to his very valuable talk. So Prash, please, you may start.
Yeah, thank you so much, Pankaj, for your kind introduction, and thank you, Abhishek, my good friend, for having invited me again. I think this is the third conference that he is organizing ever since he's back to India. So my talk, would be on prostate cancer systems genomic analysis, which we have been doing for over the last 2.5 years. This was a kind of a grant, that a seed grant, that we obtained from BrainPan, for which I'm as well an advisor. So my talk, would delve on the rationale behind this prostate cancer project. Where are we now, and what are the kind of problems and challenges, and the kind of pilot study that we have done, taking into the whole exome sequencing capture with respect to the samples that we have taken, and the kind of mutations that we have found in high-grade tumors and their inherent pathways. And since the conference theme is on data analytics or data heuristics, a small component, I would probably delve on the data science heuristics that this project would cater upon. And finally, the kind of future projects, especially taking the long non-coding RNAs and the regulatory elements that we would work on. So I come from Birla Institute of Scientific Research. Ours is a not-for-profit research institute. Our academic setting is based in Birla Institute of Technology, Mestra, which also has got a kind of a campus in Jaipur as well. And our current chairman is C.K. Birla, and our current executive director is Dr. Punendra Ghosh. And ours is headed by Dr. Krishna Mohan, who is the head R&D. And we are a group of about 14 scientists, about 7 PhD fellows and 2 postdocs now working at Birla Institute of Scientific Research with 2 major laboratories. One is biotechnology and bioinformatics, and the other one is remote sensing, where we take geospatial satellite imaging to collect the flora and fauna, and do some kind of an exciting research. So my talk would delve into 3 small stories. So the first part is about the prostate cancer and the kind of mutations relevant to India. It is unfortunate that prostate cancer is burgeoning in India. It is the 3rd most prevalent cancer in India, and it is absolutely burgeoning with as many as the Globocon statistics saying that the current Globocon statistics and the population-based cancer registries, giving a kind of an alarming incidence rate. So overall, if you really see, 1 in 9 men are usually diagnosed with prostate cancer during his lifetime. So it's a male organ-specific disease. So it's usually caused with people aged above 55 years. And it also leads to the so-called declining fertility rates, which is counter-effective with respect to the prostate cancer tumors that are uprising right from benign cases till the malignant cases. If you really see the rationale behind this particular project, in India, there are so many prostate cancer cases, but we don't know what are the kind of inherent mutations that are relative to Indian subpopulation. And furthermore, India is considered the diabetic capital. And we were very much interested, if diabetes is really relative to prostate cancer, or the other way around, whether or not prostate cancer victims would have an inherent chance or likely chance to be diabetic in their lifetime. So while I was doing my fourth postdoc in 2014, one of my colleagues, he was working on mice and as well as cattle and pigs. He would overfeed the pigs and cows so that they would become obese, and we would probably isolate these animal tissues, the fat tissues, and check for the relative expression of the genes. So he would overfeed the cows, he would overfeed the pigs, he would overfeed the mice. And then what happens? That was a kind of troubleshooting. The lab was working on in Hajar Kadamudeen's lab. And as a result, many of these overfeeding trait efficiency would also result not only in diabetes, the type 2 diabetes, but also some of these particular cases were also prone to prostate as well. Because the animals are poor, we don't really do a lot of research in identifying whether or not an animal is a prostate victim or not. We're not really bothered. But if you really take this analogy in humans, it's absolutely the same case. Because the research that was carried out on diabetic pigs, which were prostate victims, could be extrapolated to the humans as well, because the human rumen is also something similar to the pig rumen. Okay, so the entire digestive system, the entire tract, right from the neck till the legs, it's almost the same. Only thing is, pig walks on 4 legs, and the humans, walk with 2 legs. Okay, so having said this, that is the rationale behind that. And if you see the prostate cancer cases in the far west or in the Caucasian population, that is because of due to meat-eating people. Many non-vegetarians are prone to prostate cancer. That is a well-known fact. So which means that there is a diet-induced factors, a diet-induced obesity, a diet-induced constraints because they eat a kind of half-cooked red meat. But in India, at least from the kind of the samples or the cohort that we have identified, 98% of the samples are all vegetarians, and yet we have got prostate cancer. So that is really alarming. So that begs us a question: why is that Asian Indians who eat a lot of vegetarian stuff are really related to, or really getting this prostate cancer tumors? So it is also associated with vitamin D factors as well. Although we get a lot of sunshine, we drink so-called milk, but the milk also harbors some kind of vitamin D in it, and yet there are some cases prostate victims. There are also some benign cases. Some of the renal or urorectal cases that are absolutely benign, that are not at all prostatic, like fistula, piles, none of them are really getting the prostate cancer at all. There are hardly very few cases. There's no correlation at all. But the people who have not been diagnosed with these particular cases in their lifetime, or who might really be diabetic, might be associated with prostate cancer. So that begs us a question that if you really see in terms of the economies as well, there are countries like US, UK, Western Europe, the whole of Western Europe, a lot of prostate cancer cases are seen because they're all high per capita income countries. And usually from the diagnostic front, from the clinical front, many entities are taken. There usually something called the digital rectal examination is taken. There is something called the prostate serum antigen. And of course, we also take the biopsies. There is also the TNM grading, where we see the number of lymph nodes associated with the tumor and whether or not those lymph nodes are really leading to any metastatic condition that can also be inferred. So in the earlier cases, in the earlier urogenital cancers, there were some well-known markers like the HK3, which is a kallikrein gene markers, which have been absolutely deciphered. But unfortunately, these particular markers are not really taken in analogy with prostate as such. For some reasons or the other, it has not been that sensitive. MALAT1 is one of the favorite long-known coding RNAs that has been considered as a urinary biomarker. So what we did was, our total cohort is about 120, getting this prostate cancer FFP blocks and radioprostatectomy samples is certainly a huge task. So as a pilot, we started with 10 samples, another 43 are on anvil. We might get results pretty soon. And we have taken the ethical clearance, and these are all the cases. We have taken the 5 benign prostatic hyperplasia, and 2 with intermediate Gleason scores of 6 and 7, and of course 3 with Gleason score above 7, which are the so-called the high-grade tumors. In fact, Gleason score above A from the diagnostic or from the clinical front is called as the high-grade score. And this is the inclusion and exclusion criteria. So for the malignant cases, we have taken Gleason score above 6 with PSA 4 nanogram per ml or above. And for normal or benign prostatic hyperplasia, which is Gleason score 6, less than 6. And some of the samples are also extrapolated for RNA-seq, which we're also trying to work on. And we set up our own pipeline, which is our benchmark pipeline, which we established in 2017 and published in 2018, as you could see, which is Meena et al. And we used all the well-known whole exome sequencing and ACMG guidelines to infer and call the variants. And we also got good read size, which is about 9 GB with an approximate depth of coverage of above 90x coverage for all those particular samples. And more samples are also on anvil. So on an average, we got about 9.8, which is absolutely expected. So this variable grading, as you could see, this variable grading could also be extrapolated to vegetarians. As you could see, almost all of them are all vegetarians. Maybe one, when we counsel them, he said that he was a non-vegetarian at some point of time, but he's no longer a non-vegetarian. So that was some years ago. So now coming to the results, we know that in case of prostate cancer, you get a lot of genes. There are 30 well-known genes, in fact 33 well-known genes, and of course we found mutations across all those the so-called 30 well-known genes. As you could see, the square boxes are all those particular mutations seen across those particular genes from the Caucasian population, whereas the ellipse or elliptical ones are all from the Indian population. As you could see, these mutations, CTNS mutations, are absolutely pathogenic, and they are inferred to be associated with vitamin D as well. Whereas there are some mitochondrial and as well as obesity associated genic mutations coming from OPA1, which are also influencing our Indian population, which begs us a question that there could be patients who are obese and who are prostatic, and they might have this asphyxiation problem because of loss of this mitochondrial deoxphos pathways in them. So it's just like this. The theory here is if the person is obese and if the person is prostatic, then you will have fewer mitochondria because they cannot really breathe properly, and that really leads to the so-called the loss of tumor cells, or the loss of healthy cells. I'm sorry, not the tumor cells. So we did this, and we have taken the depth of coverage with above 5 and as well as 20. These are the wonderful plots now made by Ayam and Nidhi, my PhD fellows over here. And we found some inherent mutations specific to Indian conditions. And this is what we published last week. And this was a kind of a recent paper, thanks to wonderful collaborations and samples, that were collated from my clinical collaborators, Devendra Sharma and Manish Vijay, who are reputed urologists working for Rukmini Villa hospitals. So the first part of the talk here is we knew that most of the patients in our cohort are vegetarians and yet prostate victims, and we began to understand whether or not any obesity associated risk, polygenic risk is associated with them. And not only obesity, but also diet or mitochondrial dysfunction and also vitamin D deficiency. And we also found some rare disease events associated as well, like Usher syndrome, which is more related to immune prophylaxis. And these immune prophylaxis is related to the regulatory events associated with those particular patients. And of course, the well-known other tumor suppressor, which is P53 as well. So these two are well-known tumor suppressors. And incidentally, COL6A1 has got about 7 copy number variants, and we found one important copy number variant that is seen in the high-grade tumor, which we could really detect from our pipeline. And the diet-induced obesity, the OPA1, which is a well-known candidate, it's really seen, and we got 3 pathogenic and 4 likely pathogenic mutations seen across our Indian cohort, and of course the CTNS. So now we have some data, to move forward, where we have developed a kind of a conceptualized framework of mutations, where this particular data can be further used for prioritization. So now we begin to ask, now that we know what are the genes responsible for prostate cancer in India, now we also know the mutations. Many of them are pathogenic and likely pathogenic and bona fide. And now we have our own story to work on, which we call it as a genome walking story. So it is like this. So we know the genes responsible for prostate cancer, and we know where mutations are. So for example, this is a BRCA2 gene context, the highlighted one, which is in pink color, as you could see. And our mutations are somewhere over here, all through the length and breadth of this. And what we usually do is, these are all the intergenic regions. As you could see, these are all the intergenic regions. So we might even have the non-coding RNAs coming between them. But what we preferentially do was, since this is a BRCA2 gene, we further walk 5' UTR, the untranslated region, where we have a lot of transcription factor binding sites, the CDCF motifs, the long non-coding RNAs, the enhancers, and most importantly, the so-called open chromatin elements, which fall up to 5 kb, but in a rare case, you also find some of them falling up to the 10 kb region, masking the gene of your interest. So we asked how many of these particular regulatory elements impromptu are known to be associated with the genes causing with prostate cancer in India. Of course, BRCA2 is a well-known figure. It's also seen in Caucasian population as well. So as you could see here, so all these are all the long non-coding RNAs in boxes. The circle ones are the CTCF binding sites. And of course, you have got the enhancers, which are in yellow color as well. And this is a rarity. You also find the enhancers associated with that. So we have annotated them to the fullest. We know what are those long non-coding RNAs and long intergenic non-coding RNAs. We use the Smith-Waterman algorithm. We develop a kind of a bidirectional best blast hits. We have all those particular things in our pipeline. And these are all the wonderful figures developed by Nidhi and Narmada. Narmada was a graduate student. And we isolated those long non-coding RNAs. And now our experiments are on the anvil, where about 10 long non-coding RNAs have been identified with a respectable absorbance of 1.9, 8060 by 280. And now we are trying to ask whether or not these long non-coding RNAs are known to be interacting with the proteins in question impromptu. So this is how. So these, as of now, we have taken these two. One is a long non-coding RNA non-exact 053810, which is associated with BRCA1. In fact, there's also BRCA2 gene as well. BRCA2 non-coding gene, which is non-exact 103724.2, which is somewhere over here. And we ask whether or not these are all the sure shots, because many a time there's a chance that if there is a non-coding RNA sitting upstream of that particular gene, many a time it has got highest per chance to interact. And we use some tools like LinkPro and RPA-Pred and reach consensus whether or not they are interacting. And our experiments, the physical experiments in the lab, we are just trying to check their expression as well as perform some kind of pull down assays. Interestingly, in cancers at least in prostate, we don't, no one has really identified the pvRNAs. In fact, pvRNA, one candidate also we found this to be associated with COL6A1, which is a kind of a copy number variant, which I was telling you last time. So on a different note, COVID has given us a kind of a blessing in disguise. So we have worked with Keshav Singh's laboratory, where we identified that in COVID samples, we also find some of the non-coding RNAs that are associated in COVID positive with patients. That indicates that since prostate is a male tissue, there is a likely chance that the males are more prone to be associated with COVID samples or COVID infection. And they're also more likely to be, there's a chance that the prostate victims are more likely to survive with COVID positive infection. So in that particular process, in that story, we also have these non-coding RNAs, which we also consider them to be the candidates. So the part two of the conclusions, we have the pvRNAs also identified. The pvRNAs are small non-coding RNAs. Unlike microRNAs, they're bigger than microRNAs with almost one-fold. They're anywhere between 21 to 35 nucleotides, unlike the microRNAs, which are maximum 24 nucleotides. And pvRNAs are also highly conserved, but not that conserved when we consider in relation to the microRNAs. And they're also known to be interacting with the targeted RNA and aid in regulation. And the long non-coding RNA protein interactions, the pvRNA protein interactions, which I described using our annotation, is on the anvil. And there are also some sets of pvRNAs, some bases of pvRNAs, which are impromptu interacting with the proteins of your interest. They could be any proteins. In this case, the BRCA2 example, which I was taking. So they play a wonderful role. So those RNA are called as the Fuzzy RNAs. So Fuzzy RNAs are subclades of the pvRNAs, and they also play a very important role for aiding regulation. And they could be the wonderful plausible markers on anvil. So the final part of my story, so now we have huge data, we have got huge annotation, we have got about 120 data sets, 120 samples coming sometime in a couple of months, and we have prioritized the SNPs. So on an average, if we assume that there are 120 samples, taking up to 200 samples, 200 SNPs, including the non-coding RNA SNPs as well, because I only described about the whole exome sequencing as well. But the part two of the story is we also have the RNA-seq, which I didn't really describe much. So we have about 24,000 variants. And of course, we need to validate them in the lab. So let's assume 5% as per the statistical significance, including the housekeeping genes. So that begs us to a question that we need to validate about 1200 samples. And among these 1200 non-coding RNA SNPs, if at all, if you really need to go for Sanger validation, it would be pretty expensive. So this is where our part 3 comes into picture. We really don't want to sequence, prostate cancer samples from 121st exome or 121st transcriptome. We are done. We are good for done now. We only want to ask, we have huge data sets right now. Can we take this into a better machine learning training approach? Can we really train them on a network of prioritized SNPs and ask, hey, look, we have so many samples in one column, and across the samples, we have got the SNPs pertaining to, diabetes, the vitamin D deficiency, blah, 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 blah. And now, can we use something called deep variant to extrapolate our approach, our predictions, and ask whether or not, if at all, if 121st sample has got any of these particular diseased phenotypes, can we say that in those particular patients we have, this mutation, that might be predicted. Okay, so the end goal is here to develop a kind of a panel. That's where the prostate cancer panel would really be useful. There's no panel developed for prostate cancer, taking in all these long non-coding RNAs or pVRNAs, as such. So our study is really approaching towards that, taking these particular, wonderful pictures, looking into the regulatory aspects. So this is a problem which I was really describing just for a short of time and just moving ahead. That would really give us a kind of a huge incentive, because if you really look into the prostate cancer cases in the West, people spend huge monies, although in India, the clinical market is pretty high. Even, for a simplest biopsy, as you could see here, the cost is pretty high. In US, it costs about $2,615. And even in India, for a simplest biopsy, it costs anywhere between 60,000 to 80,000 rupees, which is quite high. Do you think that we Indians with a stigma, where we have some pain in our genitalia or in scrotum, we would approach a doctor? I doubt, we'll not be able to approach a doctor. I mean, maybe even me as a scientist, probably I would probably at least think twice. So that's where, we wanted to develop a kind of a simplest pregnancy-like kit, taking a simplest urinary-based biomarker, taking this long non-coding RNA as a case with all these particular conditions, taking into picture. So the deep variant results I've analyzed for about 40 samples, taking the big data sets as well. But my group members, Ayam and Nidhi, they are currently analyzing, taking the current scenario into picture as we get more and more sequencing data. So the final lessons. So we have prioritized the long non-coding RNAs and one pVRNA. And the case here is we wanted to identify those regulatory bells or regulatory elements that are present in the 5' untranslated region, and see if any of those candidates would have an influence in the way the mutations are seen in these particular patients. So which means, there could be candidate interacting partners associated with this regulation, thus aiding a complete change in the way the mutation is heading. So the present results, with respect to multi-gene panel testing, we could probably add our coding and non-coding SNPs across the various disease phenotypes. And our immediate goal as of now is also take the Gleason score attributes across all those 120 samples. And the distant goal is to look into the hereditary prostate cancer. I doubt in India if we can really do the hereditary prostate cancer things, or the germline prostate cancer in the picture in the near future, because we started just getting these particular samples, taking out of this stigma, and of course the whole exome sequence data and phenotypic variables. As of now, the sad story here is there are about 18,000 prostate cancer exomes done all over the world. And 99% of the samples are not uploaded in GenBank. They're all part of consortium. They're all folks are working with various companies for developing certain panels or developing, some drugs. That's a very unfortunate thing. If we have at least 10% of those particular samples, 18,000 may 10% would come to about 1800. We would have really been a myriad out of it, where we could have taken our machine learning approach to a better scalability. So the role of factors like diabetes, obesity, vitamin D, and dietary induced nutrient intake is absolutely associated with prostate cancer. There could be genetic factors, which we are trying to check with the IndiGene network that Vinod Skari has established. There are also the changes that we could also see across the long non-coding RNAs as well, the SNPs, and as well as the structural rearrangements, including the copy number variants. And coming up, we are also working on the RNA-seq, as I told you, and the long non-coding RNA screening and long non-coding RNA protein interaction studies for selective biomarker studies. And of course, we wanted to come up with something like MALAT1, taking all these conditions, including the hemorrhoids as well in the near future, where we are working with collaborators. So that's where our systems genomic picture, coming to picture, coming to case, coming to a valid example. So we don't just do research. We have got a wonderful group here. We have Nidhi Shukla, who is a women scientist, and Ayam Gupta, who is a PhD fellow. So both of them are doing PhDs on prostate cancer with me. And of course, Ayam is doing with prostate cancer and vitamin K deficiency. We also counsel the patients. So this picture is taken after due permission from the patient himself. So we also counsel, once in 6 months, talk to them, and also teach them, the science behind the tumors. So finally, I would like to thank Abhi and Pankaj and the entire the conference team for your kind invitation. No words of appreciation would be enough. For Nidhi Shukla and Ayam Gupta, who are wonderful PhD fellows in making. My lab, my group, doesn't go without these, two appreciations. And 3 more PhD fellows have joined with me: Shalini, Bhuman Kaur, and Anchita. Arshita is my PhD fellow who is working on recommender systems, where she will work on these prostate cancer things. Purva and Sonal are guest postdocs, and Narmada was the one who was doing the graduate fellowship with me. She did an absolute work, staying for 3 months here. And Mamta Nehra was the other one. Thanks to wonderful funding from BrainPan, Amrit and Saurabh, and Devendra Sharma and Manish Vijay from RBH, KM, my mentor, head here. Obal is my collaborator. Praveen Mathur is another pediatric urologist. Ashwini, my good friend, who is from DNA Experts, where we do a lot of sequencing, and other people over here who are really making me happy every time, with a lot of discussions. And please join BioClues. I'm the founder of BioClues, and Abhi is also a member of it, is a core member, is a managing editor for IJCB, the journal that we are associated with. So I look forward for some kind of questions. Yeah. Thank you so much.
Yeah, thank you so much, Prash, for this wonderful and clear talk. So I hope everyone also liked it. So let's take first question maybe from Shekhar. So Shekhar is saying that the prostate cancer is a hormone-related cancer, along with cancers such as breast cancer and endometrial cancer. Other than dietary parameters, have you looked at endocrine disruptors such as phthalates and BPA, which have been also shown to create fertile grounds for these cancers to develop? He's asking if you can give some aspects in this regard.
Yeah, thank you. Thank you for asking this. That is what we have really looked into, but not really endometrial cancer as such at this point of time. Our cohort size, unfortunately, is pretty small. As we just started the project, there are funding issues. When the funding came, we had this COVID issues and all. But we are getting the other 43 whole exome, raw reads, coming up next month, I guess. and we would also have the transcriptome data sets coming up at this point of time. We would definitely be interested to look into the breast cancer and probably the dietary associated things, but not sure about the endometrial cancer at this point of time. There's also a reason behind this. I'll just tell you the reason why. By the time we take or we discover the mutations in these particular cohort, at some point of time, the kind of polygeneity associated in those particular patients, associated with those particular disease conditions, would have been subsided with the prostate cancer mutations already prevailing in those particular victims. So that is the reason why we are trying to ask whether or not, if we have a huge cohort, can we really use a deep variant kind of approach in inferring whether or not those particular patients would have the highest likability or likely chance of having those particular mutations also, with the risk, So endometrial cancer is certainly a very good suggestion. Probably, we will definitely think of that. I hope that answers my question. Thank you. Your question.
Yeah. Thanks.
I will ask Abhishek here. Thanks for nice talk. I will check the questions from YouTube, and there are two interesting questions. Have you looked prostate cancer's association with obesity in Indian samples?
Yeah. That is what we have shown, the OPA1, which is a well-known gene that is associated with it. That is what we have shown. There are a couple of pathogenic mutations, but as I tell you, our sample size is pretty small, only 20 at this point of time. As we get more, we can really take this and ask a kind of a greater picture whether or not there's a likely effect of these particular mutations across those particular samples as well. So OPA1 is a well-known candidate for this, and there are two pathogenic mutations. You can see our paper that was published last week in Frontiers in Genetics.
Yeah. So there is another question by Praveen asking, is it possible to detect in saliva the biomarkers for prostate cancer?
Oh, that's indeed a very good question. Unfortunately,
long non-coding RNA.
Yeah. Unfortunately, not Praveen. That's indeed a very good question. In case of SARS-CoV-2 or other infections, people are using it because the infection would really start from the oral cavity. But in case of the urogenital cancers, the infection would only be sublimed or restricted to only the so-called genitalia or some kind of urogenital tissues like the prostate, the scrotum, the testis, and all those alike. So what happens is the kind of diet we take, whatever diet we take, that would only be effective and meaning to that particular prostate cancer victim only after 6 hours. So it will be palatable, it will be digested, and then the very first candidate marker that could be shown as an effective is none other than the urine. Okay, so that's where, because the scrotal tissues would have the highest chance of storing these candidates like urine and all those kind of stuff. So the ideal would be the urine. The second best would be the biopsy, which is a rarity in the current conditions. We may not really get the radical prostatectomy samples. And the third one, of course, would be the blood. So there are efforts, into the saliva and all, but I don't think, they would really make any good sense at this point of time. Yeah.
I have a question now. This is a very interesting talk. So when Jyotsna Batra was visiting, Dr. Jyotsna Batra was visiting us, and they are pioneer in Australian cancer samples. So she showed one of the example where the jivas, they could detect one of the missense mutation. Yeah. Do you see that missense mutation in your sample or?
Yeah, actually, it so happened, Abhi, thank you for asking that. That's a very good question. We are, there's a small unfortunate thing here, although that mutation is well known. That is, a kind of a GWAS based mutation, which we really don't want to check it, with our whole exome sample, at least with this 20 small cohort that we have at this point of time. Although we have those inherently released from all the practical consortium. We are not officially a part of practical consortium because we needed at least 100 whole exomes, to be placed with practical consortium. Then only we'll get all those particular data, given to us. So we knew what are those mutations, but we really don't know what are those allele frequency impromptu against the disease condition for those particular disease phenotypes, like like the diabetes or the other kind of cancer association. So we really don't have that, unfortunately. So we needed to rely upon the publicly available CleanMer or the genome-made mutations that are well tagged. And probably, we would definitely be looking into those kind of, things as well.
That consortium has not released the data to the public?
No. That was what was my point. Unfortunately, consortium has not released. I mean, there are 3 consortiums in the world. One is Australian-based, this practical consortium, which we are trying to be a part of it, which we are unofficially a part of it. Officially, we'll be part once we have this 120 exomes done by December. And the other 2 consortiums, the UK and the US-based consortiums, no way, no data is really released. Yeah.
Okay, well, then it's difficult to analyze.
Yeah.
I agree.
Yeah, that's because GlaxoSmithKline, the company is coming up with a good drug, for prostate cancer next year. Yeah.
Yeah, but a really nice talk. Yeah, so
Thank you. Thank you so
we're
much.
moving towards consortium. So. And yeah, so we will this reminds me that we thought to start on cervical cancer also large project, and this somehow stuck in. Now I'll write it back to all of you that we should push it after COVID. This, I mean, just now I count it now, it's after COVID because we're back to work. Yeah, so thank you. Pankaj, you can take it over.
Yeah, thank you, Pras, again. So in the interest of time, let's move ahead faster. I think we're almost running by 1 hour late with our scheduled time. So I request also the speakers to please, just a polite request to follow this time schedule. That will also help other speakers. So our next speaker is a keynote speaker, Professor Malik Youssef. He is from Department of Community Information Systems in JFAC Academic College.
Pankaj, I'm sorry for the interruption. It is also Malik Youssef's birthday today, so it is a good idea to wish him. I'm sorry for that. Yeah.
So that's really thanks for informing, Prash. So we really, on behalf of our organizing team, we would like to wish him a very happy New Year. And and
how to say in Hebrew happy birthday? Can you teach us?
Are you around, Professor Malik?
Your voice is off. You switch on your voice.
Happy birthday, sir.
Thank you very much. Do you hear me?
Yeah, I can hear you. So how to say in Hebrew happy birthday?
No, it's my language is Arabic. Eid Milad Saeed.
Arabic. Entschuldigung, Arabic.
Arabic. Yeah, anyway, it's thank you very much.
We make language ours or theirs, but it's language never says it, I'm yours. It's for everyone. So
okay.
Yeah, so in Arabic, how to say happy birthday?
Eid Milad Saeed.
Oh, Eid Milad Saeed.
Yeah, that's great.
So we learn. Happy birthday, Anis.
Thank you very much.
Yeah.
Okay, so so Professor Malik is a data scientist who has great focus on bioinformatics applications to solve various biomedical and biological problems. He has rich experience in both research environments as well as teaching responsibilities. He has almost 3 years of postdoc experience at the Wistar Institute Cancer Center in USA, and 1 year at University of Pennsylvania and USA, and where he contributed to the development of machine learning algorithms for microRNA gene and target predictions, gene expression, classification, and gene selection. He has significant expertise in machine learning, so we really look forward to his talk today, and he's today going to talk about biological domain knowledge for grouping, ranking, and classification applied on gene expression data. So we look forward to your talk, Professor Malik. So now you can take the stage.
Okay, thank you very much. First of all, thank for you for inviting me. Thank Abishka, my friend. We also collaborating. I think the topic that I'm going to talk about it is very, very interesting. During my postdoc, I lived with biology, and I understand actually the concept and the mind of biology, how they think. Actually, I remember the first talk that I gave about data analysis. I presented only the performance and the accuracy, and the question came from my professor, from my boss there during my postdoc, Professor Louise Shaw. She said, where's the genes? Then I start to look to everything different. During my talk with biology, I start to think in different ways. So this actually, I'm going to present part of my projects. Actually, I have three topics that I'm dealing with, which is gene expression. The second one is microRNA, where also I did a lot of work there. The third one is text mining. During this talk, I'm gonna just talk about the biological domain knowledge-based feature selection applied in gene expression data. So what happened in this field when the bioinformatics start to show up and the emerging data from the biology become large and big? So most of the people start to use and borrow this algorithm, machine learning, and the feature selection algorithm from the domain of computer science, statistics, whatever. So then I start to think that we need to suggest additional algorithm that can integrate, not just borrow the algorithm and apply it. We need to integrate this knowledge, biological knowledge, into the algorithm in order to understand and to suggest more significant results in terms of accuracy, performance, and also in terms of the outcome that every biology wants to see is the list of the genes. Okay. So the big picture that I dealt with during my years of research was to deal with microRNA gene identification, microRNA target identification. Here in this two projects, actually I dealt with the sequence analysis. That means how we can discover and predict and distinguish microRNA just using the sequence analysis. As I said, I'm not gonna talk about this project here, but it's important to show that I dealt with this because after that I'm gonna integrate this information in my system. And also I did a lot of work in gene expression and text classification. Then I start to think about integration those systems together. There is an additional project also I started with Apishka is using text topics and ranking the same idea that I'm going to describe today. And I wish that somehow we get some results. So just to give you a brief summary or introduction about machine learning, I assume that some of you or most of you are coming from biology. So machine learning actually is the science of programming computers so they can learn from data. So we need, in order to provide we're supposed to provide our machine, I mean by machine the learning algorithm, with data. Then we need to perform some kind of pre-processing, then convert to structured data, and then feed this nice table that consists from column and rows to the machine learning. And then we have a model that we deploy it in some unseen data. So in general, we have data. We partition the data into training and test data, two parts. And we train the model just seeing the training set. The test set is not involved in any stage of the training part. Otherwise, we will have overfit our model. And then we apply the model in the test set. And then as we are doing in education, we score the model evaluation. We see how many times it say, okay, correct, they classify correctly. For example, say for cancer, and saying for non-cancer, and how many times he make mistakes. According to that, we evaluate our algorithm. So as I said in the beginning from the conference, I would like to suggest to use K9. I just recently moved to using K9. Before that, I was using Java, Perl, Weka, MATLAB. I figured out that K9 is very powerful tool that you can program and perform very, very powerful tools. Most of my tools is done in K9. So for example, in order to apply machine learning, you need to read data table. You partition the data into training and testing. This node actually partition the data. You specify the amount of training and the amount of the testing. You send the training part to the decision layer, which is the machine learning algorithm, could be random forest, support vector machine, whatever. Then it produces a model. The model is moved to this node. This node actually get the testing data and the score. It's very nice, very simple approach that you can explain it very well to biology. And also to teach this for people that they have no programming background is very useful to deal with and do data analysis. So in general, if we have two class data, for example, patient and control, negative and positive, and we would like to find some hyperplane or any line that can distinguish these two groups. We call this as a classifier or decision boundary that actually separate these two groups and try to minimize the error. For example, you see this is actually misclassified, this one is misclassified, and in general, actually we have a function according to the sign of the function, we can decide in which side you are, patient or control. As I said, or you can just find this one, and actually there is no learning here because this one actually partition the data randomly, and then you will have very low performance. Or the most simple approach is to use linear discriminant analysis, which actually is a line hyperplan if we're dealing with the data in high dimension. Then actually it's easy to interrupt and to understand the model, which actually consists from weights, and you have the parameter x1 to xn, which is actually the gene expression that you have. The weights actually is the solution that you find during the learning stage. And if you look too deeply, actually all these features with low weight, they consider irrelevant or they contribute less to the model, then that means you can just remove it from your model. And actually, for example, if the weights are 0, that means this specific gene or feature is not so important to create or to build the linear classifier. As I said, features with low weights are weak features, and it's simple for understanding. The most usable and strong algorithm called support vector machine. Actually, I just showing you some equation. Don't be afraid. This is an approach that is used very well in the community of bioinformatics, and actually recently people try to understand the concept or the meaning of this decision tree. So actually, this was a brief introduction about machine learning. So what I'm trying to do, as I said before, when I gave my first data analysis, my boss said, what is the genes? Actually, where is the genes? So if you deal with, for example, of gene expression with 10,000 of genes, then even if you build a classifier using all of these genes, then it's hard to understand the model. And then you would like to build a model consists from, let's say, 5 genes. And these 5 genes will be considered as a biomarker for your, for example, for diagnosis. So in order to do that, we're supposed to perform some kind of feature selection. I just read feature selection is one of the core concepts in machine learning, which hugely impacts the performance of your model. Irrelevant or partially relevant features can negatively impact the model performance. So according to this concept, we suppose to search the features space in order to find the optimal subset that can give you highest performance. So in this matter, there is a lot of approaches and tools that doing feature selection. For example, you can read about, I will not gonna explain all this algorithm, backward elimination, where you start by using the whole features and then you just remove the least significant features that actually improve the performance and so on. And there is what very famous algorithm, actually the first time this algorithm was present is applied in gene expression by Isabella. The algorithm called recursive feature elimination. So I will explain it a little bit because then I will rely in the same concept when I present my project about SVM RCE. So recursive feature elimination, actually you have data, you fit a model, then you perform feature selection, you go again and apply machine learning, and again until you remove or until you end up with a set of genes. For example, you can sit with 1 gene, 2 genes, or 5 genes on 100. This is actually depends in the performance you would like to end up. So the algorithm is actually compute an importance score for each feature. Actually, if we are talking about linear, then actually the importance is the weight of the features. For example, weights with 0, that means the corresponding feature is not so important, then you can just eliminate it. So at the least important features are then removed. For example, you can remove feature 1 by 1, or you can remove 10% or 50%, and then you go again and fit again the model and remove again and again, because that is called recursive feature elimination, because you apply this approach recursive way. Okay, so the gene expression, I assume that everybody here is hear about it. Actually, it's measure the expression pattern of many genes at once. So in the end, we have this nice data that consists from 2 groups or 2 classes. And again, it could be unbalanced data. This is something very famous in this data. For example, you have the column represent the patient, and the rows represent each gene, and the value here is represent actually the expression of these genes over this sample. The numbers here has no meaning, but the ratio or the relative values of this number, they have the meaning. For example, if you have these genes, for example, is expressed 0 here and very high in this healthy, that means this gene could be biomarker or the opposite direction. I see chat. Okay. So during my postdoc, I dealt with a lot of data analysis. And during that time, when I have the interaction with the biology, I start to look to the data in different ways. So when I provide, actually, this is something funny. When I present then my boss said, where's the genes? And I have here non-smoker. I will tell you the story. So then actually I changed the model. I start to give her the rank of the genes. And then we took these genes and moved to David to find the relation between these genes. I said, okay, instead of doing that, let's start to impede this information into the algorithm, because we know that correlated genes, they have the same function. So if the correlated genes have the same function, so let's put it together and start to rank it together. So put the similarity guys together and just deal with them together. Then actually the first idea that came is called recursive cluster elimination, which is actually associated with recursive feature elimination. And I start to deal with this project. Maybe I will answer the question later. Okay. So during the data analysis process, we're doing a lot of procedure, a lot of work. First of all, we do pre-processing to prepare the data to be ready for machine learning or for analysis. And there is a couple of approaches that you can perform. You can perform PDA, PAM, LDA, RFE. You can cluster the data. There is a lot of approaches and algorithms that can apply in this data. In the end, you would like to see a heat map that tell you these genes are very high, are very low in this NH, non-healthy, and they are somehow high in the other group, or something like that. Then actually we assume and we say this is the biomarker according to the gene expression. We can just distinguish the two groups. So the question is in machine learning, how we can take this huge data, I mean huge or big data in terms of number of genes, 30,000 genes or more, and then end up with something like that. Also, we would like to, during the data analysis, we try to find maybe the number of genes, over 10 genes, 20 genes, 100, and then we can say, okay, for my model, I would like to take this performance. So I will have 95% true positive, true negative, and average accuracy. Always we run the model 1,000 round or 100, and as you said, we can also, as I said before, we can give a score for each sample and then also plot this score to see the difference between these two groups. And also we can plot the data using PCA, multidimensional scaling to see how the data is behaving according to top 200 genes. So actually this picture gave you information about, visual information about your data. Okay. So now I will start to explain the merit or the generic approach that I have been developing with collaboration since 2007, I think. Yes, or 6. So actually, if we have data that consists from two classes, even with one class we can deal, but let's assume for simplicity that the data consists from two classes. The two class is two label, healthy, non-healthy, and is gene expression. So, and we assume that we have a grouping function, general grouping function. Now I'm talking about general grouping function. I will start to explain one by one. Then, for example, the grouping function could be clustering, to cluster the genes, not the sample. Could be microRNA target, could be kick pathway, could disease. That means a grouping function could be any biological function that you would like, that you can use it in order to put the genes together. Okay, then we have list of genes. Then G, you apply G, grouping component, that actually create a list of groups like this. So one group is actually consists from different or a set of genes. These genes, they are sharing some similarity. So in the end, we will have a set of gene groups like this. Then the question, how we can rank or predict if this group can distinguish the data that we have. Then actually we perform the ranking component. So ranking component could be, you can actually here and here you can put any idea that you would like. So the grouping component could be something that you would like to use biological information. And the ranking could be any approach that you would like. We decide to use the machine learning to split again the training data into, for example, cross-validation using support vector machine or random forest or whatever. But we present the data just according to this group. That means now we examine each group individually. We represent the two class, which is the training data, just according to the genes that belongs to this group number one. That means now our data just consists from these groups, these genes. Then we apply sort of machine learning in order to predict or to give a score or a rank saying these genes belongs to group number one, can distinguish the training data, for example, 90% accuracy. And then actually we give a score for each group. Then we can build a classifier from the top first group or the first 1 and 2, and the first 1 and 3 and such on. We choose to try it until 10. Then we apply the model in each group, the type group. We end the test data. Again, we always partition the data into training testing. All this process is happening just on the training data. We apply then the test part to see if we get some sort of good performance. This is the algorithm that we developed for ranking. So the ranking algorithm actually we call it R. You get X as a sub data. M is the group. F and R are the how many times you would like to perform cross validation or another sort of performance measurement. So then we actually go over each group. We remove all the genes from XT and XV, which are not in the group MI. That means the data now represented just by the genes belong to the specific group. And then we perform train classifier, for example, support vector machine, or as I said, another algorithm. Then we calculate and we give a score. So the score is saying this group alone, for example, group number 2, can distinguish the data, training data by 90%. So the 90%, 0.9 is the score that given to this group. Okay. So since 2007, actually, I started to develop with my colleague algorithm that we called SVMRC, support vector machine recursive cluster elimination. Then we apply the same approach, SVM RNE, recursive network elimination. Then I start to apply RCE, recursive cluster elimination with ensemble cluster. Here, actually, we cluster the data in different way. This actually was work with Dr. Loai, where he actually is expert in ensemble clustering. Then we just cluster the data in different way. In 2019, we start to work actually in combining microRNA with gene expression. Recently, we have updated SVM-RCE, and we add more functionality, and we also apply it in K9. There is the running project now. Cognate is now is under review, where actually we use PathFind-R, I will explain it later, to produce the significant enrichment pathway. Then we rank each pathway in the same way that we have done before. And also, just recently, we solved, I think, a very interesting approach where we have not the gene expression. All these tools actually deal with gene expression. This one actually deal with gene expression and the data from MERTAR base in order to group the genes. But this tool actually take as input microRNA gene expression and also the gene expression. Then the question is how we can combine and integrate these two data to figure out some useful information in terms of microRNA and target and gene expression. This one is actually is running project. We almost done. All the workflow is ready. I will be happy to discuss collaboration. And also this one is, as I said, is under review. So I go back to the first idea that we have applied. So we have data. We split the data into training testing. Let's assume 90%, 10%. Then we apply k-means in order to cluster the data. Now we have n clusters. According to k-means, we use the correlation matrix. That means these clusters contains genes that are correlated according to applying the k-means algorithm. K-means algorithm actually is a clustering algorithm. Then we have a set of clusters that k-means output. Then the question is how we can score it. Then the second approach is, the second component is the scoring or ranking. As I said before, according to this generic approach, we perform some kind of cross-validation, internal cross-validation on the training part when we apply it in each clusters. Then we give for each cluster a score saying if these clusters can distinguish the data very well. We sorted, we remove 10%, then we go back again and we cluster again. Then we go then we process the approach again. We remove lower clusters. In this approach, we don't remove as SVM-RFE couple of genes. We remove clusters together. So the weak clusters is removed. Actually, the question here, we dealt with individual cluster alone. The question that also we would like to explore, this is another project that also I have a student in Abdullah Gul that he's supposed to start this project, is what about considering different combination of clusters? That means we don't need to in this approach, we consider individual cluster alone. But what if we cluster number one, cluster number two, if we merge it together, we will have a higher performance. So in this way, we're losing some information. So actually, there is an idea and a running project that we as I said, with Abdullah Gul University, that we try to find the optimal combination of these clusters that can improve the performance of this approach. Then actually we move to SVM-RNE, where we cluster the data, not using k-means. There is a tool called GXNA that actually using the gene expression in order to suggest network between these genes. Then the network actually is a groups for me, like this, groups. So again, here the groups are clusters, here the group are actually the network. Again, we do perform similar approach and we redo that approach again and again. This two paper was published in PMC Bioinformatics. This one actually is the way how we can actually cluster the data in different way, what we call it ensemble clusters. I will not go deeply. The paper is ready to read, is published. This was collaboration with Dr. Lai. Recently, I did also this project with Professor Jens Almers, also a keynote maybe tomorrow, where we integrate information from microRNA and gene expression. This paper actually published in Bioinformatics Oxford Journal. This too, as I said, is just under consideration. This one is a running project. Okay. So if we have data, we apply G, the grouping, and we apply R in order to cluster. This is the concept that we did it. And also then we compare our approach SVM-RCE with SVM-RFE, with PDRFE. This paper, as I said, is published in BMC Bioinformatics. The output of our approach is consist from two parts, actually the number of clusters and the number of genes in these clusters. To match it, we look to SVM-RFE, we match 11 with 12, and we apply the output here. And we see in some cases we are outperform RFE, even in most of the cases here. This one actually we did bad, but the RFE did much better from us. So what is he saying here is not just only the significant list that we have, also the heat map, how it's looking. If you look here, you see we have clusters. We capture the clusters of genes that they are have some similarity. But here, actually, if I will not go deep with the difference between SVM-RFE and SVM-RC, you are invited to read this paper. But actually, we capture a cluster of genes rather than dealing with gene once at a time. As I said just recently, we submitted this project when I was visiting during the summer in the previous year with our institute. we convert the project as VMRC into K9. But additionally, we add a ranking. We add what we call it ranking function. So the traditional rank was using the accuracy, but during this version, actually, during this update, we let the user to specify the weight of the different measurement that we can calculate from our performance. For example, accuracy, sensitivity, specificity, F1, area under curve, precision. For example, for instance, you might be interested to have higher sensitivity, and you prefer to have higher sensitivity rather than higher specificity. So you will give a high weight to sensitivity and lower weight to specificity. And we try to different rank approach function. And actually, we see that different rank function can improve the performance even dramatically, like even 16% in some of the cases. As I said, the approach is also the K9 workflow also accessible. You can download it from my homepage or send the email. I can send it to you. The next actually question is to find the optimal weights that can give me the optimal performance. This is another project that is actually open for collaboration. Okay, 2009, actually, we wrote that as a conclusion in our papers as VMRCE, is that we suggest that gene interaction network or other biologically relevant matrix that group the genes based on functional parameter might also be useful. According to that, actually, we ran this approach. We use GXNA. That is a tool that can take as input gene expression and plot or create the networks. Now the networks, as I said, is groups, and then we rank each network individually. Again, we published this in BMC Bioinformatics. This work was a collaboration with Professor Luis, Professor Larry Minevich from Haifa University, and his student, Muhammad Kittani. Okay, next, the third project in this topic was using ensemble clustering in order to put the genes together, in order to group the genes together. This project was with Luai Abdullah, as we use his approach rather than using k-means. We use the ensemble clustering in order to group the genes together. Okay, so then we start a new project where we consider gene expression, microRNA grouping. As I said, I have experience also with microRNA expression, and machine learning. These two actually is the last project that we are dealing with now. This microRNA grouping with gene expression, now actually we did this tool. We call it MATI. We publish it in Bioinformatics. Okay, so what is MATI is? MATI is we have as input gene expression, but we don't use k-means to cluster the data. We use the information from MIRTAR base. MIRTAR base is actually a database that telling us have information about microRNA and its targets. So then we use this information in order to group the genes from this data. What does mean to group the genes? For each microRNA, there is a set of targets, validated target. So these genes are considered one group that associated with this one microRNA. But for our algorithms, this is one group, and this is the other group, and this is the third group, and so on and so on. So the question, we use this two knowledge, biological knowledge and gene expression, in order to integrate this information, biological information and gene expression information, to put the genes into groups and then perform the two-step component, the grouping using this and the ranking as we have done before. Actually, I remember we had tested here support vector machine and random forest, we found that random forest give us much better results in this term. Okay, the workflow is published in we publish it during our publication. Also, it's written in K9, somehow complicated. Recently, we just make it much easier in this way. That means we convert again the K9 into simple way that can be easy for interpretation. And if you look to the workflow, you see you have created the groups. This is meta node. You rank the group, then you run each one to remove the lower clusters, estimate in the test data, performance and test, then you collect the output. Actually, the workflow in K9 somehow describe this algorithm that we describe here, or the main algorithm that we describe here or here. So actually, in this way, it's easy to interact with the biology community and explain the algorithm that we are developing. This is a new version. This is also, we try to develop another stuff with this K9 approaches. We are open also for collaboration. We have different idea to improve and add more component to this workflow. Okay, this is actually the two table that describe the MAT algorithm. I will not go deeply to that, but you can read it over the manuscript. This is one output of the ranking approach, the ranking component. For example, if you see here, this is the microRNA. This is the accuracy. The accuracy is the rank. And the list is sorted by accuracy. That means all of these actually ignored from our analysis. We consider the top one or top two. And something interesting, you see, this is one actually consists from just one gene that belongs to this microRNA, and still have very good performance, just considering this one gene. And this one actually consists from 3 genes. The other project that also I start with the Professor Jens to see the influence of the size of the clusters or of the group. We assume this one is very giving higher accuracy from this one because the number of genes involved in this or belongs to this group. We start to deal with that to develop computational ideas. It's just in the first step to find a solution. The idea is to eliminate the size of the group in terms of ranking performance or ranking score. Again, we run it against SVMRC. This is the most similar approach that we have. We've tried to find similar approaches. We don't find anything that can compare to MATI. We compare it to SVMRC. We show that we get a very interesting result. But again, the idea here is not to get very high accuracy just, only to suggest different significant list of genes which is associated with microRNA. For example, if now the biology take this list, the top 2, and the top 2 are not, okay, they can distinguish the 2 class with very high performance or accuracy. The thing here that these genes are associated with microRNA. So the approach here actually predict the targets or the target involvement in the data that is under investigation without knowing any information about the gene expression of the microRNA itself. This is the unique of these tools. I suggest to you to read it over bioinformatics. This data actually is not published yet, where we group the genes according to pathway, KIG pathway. Now the grouping function is KIG pathway, or could be also we did some comparison, or another study is to put the genes disease. These 2 projects we still also considering, and we think about to make a general approach that the biology or the researcher can decide how to group the genes together. Okay. The two recent project is called the one called Cognet. This project was with Professor Cesar and his student Cesarman and his student Ege. They published a tool called Pathfind. This tool is R package. We're getting input a list of p-value that this p-value is extracted from gene expression. Then we feed this list to the Pathfind R to find the active subnetwork search and filtering. Then he suggests apply enrichment analysis in order to suggest the significant subnetwork. Okay, when I saw this tool, I have the idea to combine it with the idea of rank and the grouping. So now the grouping is Pathfind R here. And again, we perform similar ranking approach as we have done before, but we don't have recursive because we redo the process, we will have somehow slightly or maybe different, but the similar groups. So we create n data set for each pathway. We rank each pathway, and then we test it in top ranked, maybe 1, 2, 3, and so on. According to that, we can decide to which rank we stop and get the higher performance. As I said, this tool is under revision now. This is the result that we show, compare Cognet using random forest and the SVMRCE with MATI. And we see that Cognet getting very, very good results, even reaching 86% performance over the 13 dataset. This is the average. And the recent project that is still in running, we call it Mere Correlation Net. So this is the first time we feed our model with two kinds of data, the microRNA expression and the gene expression. And then the question, how we can integrate these two kinds of data in order to apply our approach of grouping and ranking. So we group the genes. First of all, we find the differential expressed microRNA. Again, we apply it in the test part. And also the differential expressed genes from the gene expression. Then when we apply correlation between these two matrices, we set a threshold 0.0 minus 0.6. That means we put all the genes that are negatively correlated with this specific microRNA into one group. So we hypothesize that these genes should be somehow related to this microRNA, could be the target, or they have some interaction with this microRNA. Because when this microRNA negatively correlated with these genes, should be some biological information between these two genes and the microRNA. So now we have a groups, we rank it, and we test it. As I said, this is in preparation. And also this project with Dr. Borjo from Abdullah Gul University, and Mitra and Christian is from Jefferson University. Actually, they are trying to find the biological validation of our outcome. And actually, Mitra did very nice analysis and find, when considering the top microRNAs, he found a correlation between our prediction and to what is known in the literature in terms of cancer and potentially in terms of cancer types. We still try to understand more deeply the data here and the outcome of the data. And again, we compare it with the MATI, with the Merck. This is our project, and RFE and RFE. This is the most similar approach that we find, and we try to compare it with them. Again, we wrote this in K9. So at the end, I would like to introduce you to my homepage. You can access my homepage and all of these tools. Actually, I'm also modifying this list as much as I can. And all of these tools can be downloaded and it's free for your usage and you can use it. And if you have any question, I'll be happy to answer and help in using these tools. Thank you for listening. And I would like to mention all our collaboration, Professor Jens, Larry, Mehmet Kittani is a student, Professor Louise, I still collaboration with her. Recently, we start with Rahman, the SVMRCR code, and with Abdullah Gul University. This is my picture when I was young. Dr. Luay Abdullah, we have applied many projects in ensemble clustering. And the Borjo group, Gokhan is responsible about the last project, Mir Core Net. And also Amhar is the guy gonna solve the problem of optimal optimization of the clusters. As I said, we have collaboration with Mitra and her boss to actually to find and to interrupt the outcome of our tool. Ugur and Ige, we did the project of Cognet, and also they published the Pathfinder tools. Thank you very much, and if you have any question, I will be happy to answer.
Yeah, thank you so much, Professor Yusuf. So there is one question from Anirudh. Does PCA or any other ML feature selection method help in feature selection in gene expression data?
Actually, if you look to the literature, there is a lot of review and a lot of study that compare different approaches of using different feature selection method. I didn't use PCA, but yes, I can tell from my reading that PCA has also influence in it could be considered as a feature selection. The problem with PCA that you don't actually have the gene itself. You have the component, the most component, the second or the first or the second or the third, but you would like to, in the end, to reverse this component to get the genes, because the biologist or the researcher in this biological domain, they would like to touch and to understand what is the name of the gene that they serve as biomarker.
Yeah, I think that's correct. So the next question is, how do you decide the value of K in KNN algorithm?
Yes, this is very interesting question. And also I forgot to tell another idea of this project. Actually, we try with K with 100, arbitrary. And when we eliminate 10% until we end with one cluster. But the question is very interesting because also we have another open project, how to determine the optimal number of clusters. And recently there is some clustering approach like DSP SAN that actually doing find the optimal number of clusters automatically as algorithm. We would like also to try this different kind of project.
Okay, thank you. So I think we can take maybe the last question from Prash. So could this 9-based mate approach you showed be used for long non-coding RNA target predictions?
Which one? Which tool?
Could this 9-based mate approach, sorry, MATE, can be used for long non-coding RNA target predictions?
Actually, I didn't present here my work for target or microRNA target prediction, but if you look to my publication, you will find other tools also from my homepage that we use just K-MER in order to distinguish microRNA and to find the target. From that, also I have a different K9 workflow that I will be happy to provide to you or for collaboration.
All right.
These tools actually deal with mostly with gene expression. If your data consists from gene expression and you have non-coding information that you would like to use it as a grouping, yes, it's easy to modify it.
Yeah, so I hope that answer Pras question of Pras. So yeah, maybe the last question. So how did you, from my side, how did you like, today is your birthday, how did you manage to be a speaker today? Is it because of the COVID-19 or it's the enthusiasm for research?
Say again?
So is it the situation that's allowing us to be a speaker on your birthday?
Actually, to be honest, I didn't notice that.
Yeah, the later one, yeah. It's a true
I did not
enthusiasm for
notice
the
that.
research. Yes.
And even my wife tell me happy birthday in the morning. I said, "Oh, really? This is because I was busy preparing also this talk, and it was very very nice thing that it came with my birthday. I am actually finishing 50 years old, which is very also nice number, and starting the 51. And I need to think now about my next stage in life.
Yeah, sure. So yeah, it was truly a valuable talk, and I'm confident that audience really enjoyed it. So again, we would like to thank again on the behalf of entire team. So we will definitely be in touch in the future. Abhishek is already
yeah. So basically, what I am saying that he always managed time for us, sometime even on Sundays. So thank you, and happy birthday once again.
Thank you very much.
And very interesting thing is that in my presentation, his daughter is also researcher and she is working with my team actually. So
yes.
2 researchers in the family is a great thing to have, and 2 generations, yeah.
Yeah.
So your parents and grandparents, but at least 2 generations. So happy birthday and have a nice time with the family. Yeah.
Thank you very much. Thank you. So I need to do what to
just stop your sharing.
Stop sharing. Okay. And you move the.
Yeah.
Okay. Do I need to give permission for anybody or what? Okay.
So moving to the next session, so this will be chaired by Dr. Dilpreet Kaur.
Hello.
So, are you now, Dr. Dilpreet? Is Asta around? Abhishek, has Asta already joined?
Yeah, I'm here.
Okay. Yeah, hi Asta.
Yeah, hi.
So yeah,
well, look like that it is saying that host disabled participant screen sharing.
Now you are host. Now you are hosting us.
All right. So Dr. Dilpreet, please, you are on the stage.
Thank you, Pankaj. The first speaker of this session is Dr. Asta Forsti, and she is Senior Research Scientist at German Cancer Research Center and University of Heidelberg, Germany. She has obtained her PhD in biochemistry and biotechnology from University of Kuopio, Finland. She has published over 200 research articles and research proceedings. Her area of research is breast cancer, colorectal cancer, genome-wide association studies, SNP array, and DNA sequencing. Today, she is giving a talk on germline genetics of cancer. Now, I welcome Dr. Asta Forsti. Over to you. Thank you.
Thank you, Dr. Kaur, for this kind introduction, and I'm happy to be part of this first international conference on data science in biology. And I also thank Abhishek for the kind invitation to this conference. So when I listened to this previous talk, so I noticed that my talk will be much more translational than the at least the previous talk, or I guess that most other talks in this this conference. But I hope that you will be able to follow me. So I will be talking today about germline genetics of cancer, and I will show you a couple of examples of our own studies on this field. And before coming to that, I would like to introduce you a few terms on this germline genetics. So the first one is hereditability. So which means or indicates how much of the cancer risk is explained by inherited genetic factors. And twin studies have been a valuable tool to evaluate heritability and also environmental components of cancer. And as you can see from this table, so the common cancers, prostate, breast, colorectal cancer, so they have a heritability something like 30–40%. If we then look at the proportion of cancer, which is familial cancer, and here by familial cancer, I mean that in the family, there are at least 2 cases, two first-degree individuals who have the same cancer. And the proportion of familial cancer of all cancer in prostate cancer is something like 20%, in breast and colorectal cancer, it is less than 15%. Then what we are knowing about the genes behind this familial cancer. So the proportion of familial cancer explained by the known gene is highest in breast cancer, where something like 50% of the genes are known, or the families get an information which gene could be the reason for their cancer. Then in prostate and colorectal cancer, so the proportion is around 30%. What are then these genes which are known to affect familial cancer risk? So here in the scale of variant allele frequency in the population and cancer risk size, so we have at one end the rare high penetrance cancer genes, which were identified mainly using linkage analysis in families. And here you can see the well-known breast cancer genes BRCA1 and BRCA2, and then also the known colorectal cancer genes ATC and mismatch repair genes. At the other end of the scale, we have a large number of common low penetrance alleles, which have been identified by genome-wide association studies. Individually, these variants cause a very low risk, but then in combination, they can have a substantial effect on cancer risk. And between these two loci, we have very few moderate risk alleles. And what we are doing now, so we are using the next generation sequencing techniques, whole genome and whole exome sequences to identify more high penetrance or moderate risk alleles. And we are also contributing to the identification of the low penetrance alleles, common variants, using the genome-wide association study approach. And then I start with the first example, and here we have coming back to the family-based studies. So we are trying to identify families with many cancer cases, and we are trying to also collect blood samples from as many cancer cases as possible, and then also from unaffected healthy individuals in each family. And we hope also to recruit some individuals from the older generation of the family. So by that, we try to guarantee that a single gene defect is the cause of the disease in each family. This is then the pipeline which we have developed in our division, and Abhishek Kumar has been a key person in developing this bioinformatic pipeline for this project. So we start here with identification of families together with our collaborators who have collected a large number of familial cases and families. And then we collect the DNA sample and isolate or blood sample and isolate the DNA, or our collaborators are often doing that also. When we then get the DNA sample, we submit it to our core facility at German Cancer Research Center, and they are doing the whole genome or whole exome sequencing. They are doing the variant calling and variant annotation. And here our main scientific collaborators have been Matthias Lesner and Nakarajan Paramasivam. After that, we get all the variants with variant allele frequency less than 0.1% to our lab, and we submit them to the pedigree-based variant segregation analysis. And only the variants which are segregating with the disease in the family, so they are then going further to this bioinformatics part of the pipeline. And here we classify the variants based on their location in the genome. We start with coding variants, which are the most likely to be the functional variants in the genome. And we used something like 10 tools to evaluate the deleteriousness of the variants. Then we used some 5 tools to evaluate the intolerance of the corresponding genes against the functional variants. And we used also 3 tools to evaluate the conservation of the variant position itself. Then for the non-coding parts, we have also divide that also in different parts like 5' UTR, 3' UTR, intergenic variants, and we use different bioinformatics tools to evaluate their influence of gene regulation. After that, we have something like 10-20 variants, which we then submit to a thorough literature search. And after that, we may have something like 5-6 variants, which we then go further with validating in additional families. And many times this is done by our collaborators and sometimes by ourselves. At the same time, we are also doing functional evaluation in our lab, and there Opul Pandapalli has been a key person behind these assays. And then Manda Pepingli, Sara, and Ayushi have been the persons working in the lab on this project. Oh. Somehow. Maybe I take this pointer out because it looks like that. Cannot move that. Okay. So here is then the overview of the family.
Pointer, can you check? Can you go back? Just
to the pointer.
Yeah, just pick the point, ledger. Oh, yeah.
Yeah. Okay. But if I had just problem to change the slide, so let's see how it works. Yeah. Okay. But here is then anyway, so that the families which we have sequenced so far, or now we have maybe somehow something like 300 families or 300 genomes sequenced, whole genome or whole exome sequenced. The largest number of these families are colorectal cancer families, but we have also a large collection of multiple myeloma families, and then we have some pancreatic cancer, thyroid cancer, Hodgkin's lymphoma, cervical cancer, and neuroendocrine tumors as well, which we have sequenced. And here I give the examples on the colorectal cancer family analysis, and here you see the 4 families where we identified mismatched gene mutation. So you can see here in these pedigrees that we have colorectal cancer cases which are indicated either with the black or red circles and squares, and so that we have colorectal cancer cases in 3 generations in most of the families. And the variants which we identified in these families was a brain shift mutation which caused a stop codon. Then we had a splice site variant which also caused a brain shift, and then immature protein translation. And then we had one large exonic duplication in one family, disturbing also the protein. And then in one case, we had a missense mutation, which was predicted to be deleterious. Then as an example how our pipeline is working in our family, so I take up this family where we have 3 colorectal cancer cases indicated by red and 2 polyp cases in the family. So these individuals have colorectal polyps and maybe an increased risk of getting colorectal cancer. Then we could also sequence one healthy individual who was unfortunately very young, 23 years old. And in this family, we identified a protein tyrosine kinase 7 missense variant. Here, here you can see also how our pipeline is working with the decreasing the number of variants. So this family was whole exome sequenced, and we identified altogether 10,500 variants with allele frequency less than 0.1%. 421 of them segregated with the disease in the family, 72 were exonic variants, and 47 non-synonymous variants. Then we used general deleteriousness prediction score, a kind of meta tool, to identify 32 variants which were among the top 10% of the deleterious variants in the human genome. 23 of these variants were located in the conserved region. 14 were located in the genes which were intolerant against functional mutation, and after we used the 10 deleteriousness tools, we ended up to 6 variants. And these variants you can see in this table, and we focused on to this protein tyrosine kinase variant because it is involved in Wnt signaling pathway, which is an important pathway in colorectal cancer, and it is involved in cell adhesion, migration, polarity, proliferation, and so on. So here is our original family again, and we confirmed the segregation in this family by using Sanger sequencing. So the variant was present in these 3 colorectal cancer cases, and it was present in one of the polyp cases. It was not present in this other polyp case and not in the healthy young individual. But then we screened this variant in an additional 1,700 familial colorectal cancer cases, and we identified another individual who carried this mutation, and the father of this individual and one of his half siblings also carried this mutation. So now we have 2 families where the same mutation was identified. This variant is located in the extracellular domain of the gene, so it is located in one of these IG loops, and there are some publications showing that the canonical Wnt ligands like Wnt3a bind to this extracellular domain of the PTK7 and initiate the degradation of the protein. So we hypothesized that this binding may be interfered by this variant and inhibit PTK7 degradation. Then we tested if this protein is more stable in the when it is carrying this mutation, and we introduced this mutation into the colorectal adenocarcinoma cell line HD29, and indeed, so we observed that there was an enhanced PTK7 protein expression compared to the in the wild type cell, in the mutated cells than in the wild type cells. We also observed that there was an increased cell proliferation in the mutated cells compared to the wild type cells, which are shown here, the mutated cell proliferation in the red line and the wild type in the blue line. Then we went on to look at the downstream targets of PTK7, and we observed that in the variant cells, the expression of the downstream targets of PTK7, CREP and P53 were decreased in the RNA level, And we also confirmed that in the protein level, as you can see here, for CREP and PTK7 in the mutated cells and then in the wild type cells. Then we also noticed that there was an increased phosphorylation of AKT in the mutated cells compared to the wild type cells. And that indicated that this mutation may interfere in the apoptosis or activation of the AKT signaling. Sorry for that. So I don't know why it is. Yeah. Okay, so as conclusion for this part, so we have developed a familiar cancer variant prioritization pipeline that can prioritize both coding and non-coding single nucleotide variants, small insertion deletions, and copy number variants in the whole genome sequencing data. And this data can be used then for further functional validation. In managing cancer risk, so it is important to recognize risk factors, identify high-risk patients, and implement appropriate surveillance. And we have contributed to this in this study by identifying a novel missense mutation in a protein tyrosine kinase PTK7 that potentially inhibits apoptosis pathways by downregulation of its downstream targets CREP and P53, and which activates AKT signaling by increasing AKT phosphorylation. As clinical importance, our study may help in genetic counseling and personalized therapy of the families. And it may also help in development and implementation of individualized treatment options. Then I move to the second part of my talk, where I explain or show you one example of our approaches on, or our studies on using whole genome association study approach. So here we collect a large number of cancer patients and a large number of healthy controls from the same panel population. Then we do the genotyping using the Illumina single nucleotide polymorphism sheets. And then we do quality control using both sample-based quality control and SNP-based quality control. And here we exclude all the samples where the SNP call rate is low, where the individuals are not of European ancestry, and from related individuals we only take one into our analysis. And in the SNP-based analysis also, so we exclude all variants with low call rate, with low minor allele frequency, and where the genotype distribution is not according to Hardy-Weinberg equilibrium. Then when we have this high quality data, so we impute all the variants which are not directly genotyped in these Illumina sheets. Then we do the association analysis, and here we use the genome-wide significance level p-value 5 × 10⁻⁸, and suggestive levels of significance p-value 1 × 10⁻⁵. Then in most cases, so we have either replication population for the best associations, or in many times also the whole genome association study from another population, and we pool these in the meta-analysis. Then we can also use the stratified analysis based on the subtypes of the cancer. And at the end, we of course have the most important part to interpret the results using both publicly available data and then also functional testing. So here the situation of the known risk alleles, of the known common risk alleles in cancer which have been identified by genome-wide association studies. And those ones which are underlined here, so that are the cancer types where we have contributed. And I will take up the myeloma as an example, because there we have basically identified all the so far known common low penetrance alleles. So multiple myeloma is a plasma cell disease which originates from the post-germinal center B cells. It goes first to the precursor stage monoclonal gammopathy of undetermined significance. Then it can stay in this stage, but then it can also develop further to multiple myeloma or AL amyloidosis. Multiple myeloma can then, in rare cases, also lead to plasma cell leukemia or AL amyloidosis. Here are then the diagnostic criteria of these different stages of the disease, and it is based on the M protein level. So this is an immunoglobulin which it is they are expressing. Then on the bone marrow plasma cell count and end organ damage. And here you can see that AL amyloidosis and multiple myeloma are the only ones where there is an end organ damage. For multiple myeloma, there is also a CRAP criteria, which means that calcium is elevated, there is a renal failure, anemia, and bone lesions. Then there are also a lot of genetic events during the course of the disease. So the primary genetic events involve usually the immunoglobulin heavy chain translocation together with known oncogene like cyclin D1 or fibroblast growth factor receptor 3. Or then there are also multiple myelomas with hyperdiploidy, and there are trisomies in the odd chromosomes or in the most odd chromosomes. Sometimes there is also a combination of both of these. Then there are also secondary genetic events which are often chromosomal regional deletions or gains. There are also some mutational events, most commonly NRAS, KRAS, and BRAF, and then also some new translocation, usually other translocations not involving the IGH. Then I will show you the results of our latest meta-analysis, which was published in 2018. So there we had some 8,000 cases and 240,000 controls with three genotypes from the UK, Germany, Iceland, USA, Norway, and Sweden, and the Netherlands. And we replicated this study in 1,700 cases and 6,000 controls from Germany, Denmark, and Sweden. Here are then the results in the Manhattan plot scale. So here on the x-axis, you can see the position of each SNP on the genome according to the chromosomes, and in the y-axis is the significance of the association as minus logarithm p value. And this red line here shows the genome-wide significance level, and the blue line the suggestive significance level. And I will show you later on an example of the importance of this most robust locus in this analysis. So in multiple myeloma, there are no high-risk genes known, and these 23 loci are the ones which have so far been identified as low penetrance risk alleles, and we have estimated that something like 60% of the heritable risk is attributable to all common variation in the genome. And because most of these variants identified only here in this study, so are located in the non-coding region, so there is it is important to functionally evaluate the reason for this association. So that we are doing using both publicly available data and then also doing by ourselves some experimental analysis. So we can evaluate the chromatin state of the associated SNP or linked SNP, so that like if it is located in active promoter or active enhancer. We have expression data available for the malignant plasma cells, so we can look at their EQTL effect, so that if they are affecting the expression of the nearby genes, then we can also look for if these variants are enriched in the locations where there are active histone marks and the histone is available for expression. And then we can also do promoter capture high-C analysis for long distance chromatin interactions, so that if the SNP locus is interacting with the promoter of a target gene. Then I come to the functional relevance of the 7015.3 locus. So this variant is located in this DNA H11 gene. But it maps to the IRF4 binding site, and IRF4 is an interferon regulatory factor 4, which is also known as myeloma oncogene 1. It is located in the strong enhancer element, and it is a strong EQTL for the nearby or neighboring gene CCDCA7L. This gene encodes a MYC interacting protein, and it is an important partner for this pre-B cell differentiation and B cell development, and MYC is of course an important gene in cancer development as well. So when we knock down this IRF4, so we notice that in addition to suppressing the expression of IRF4, also this target gene CCDCA7L was suppressed, and this suppression led to the decreased cell proliferation and induced apoptosis. Then we have also done stratified analysis using the cytogenetic data, which is available from Germany and UK for something like 1700 cases. And here we identified cyclin D1 splice site polymorphism as a risk factor for T1114 multiple myeloma. And this is the first time when a constitutive genetic factor was associated with the risk of a specific chromosomal translocation. This locus is very interesting in that sense, so that it has two transcripts. So the transcript A, where there is the G allele on the splicing site, is the full-length cyclin D1, and A allele causes an alternative splicing, and it leads to an alternative transcript. And it is interesting that the G allele is the risk allele for T1114, but then the A allele is the risk allele for cancer. In this B transcript, so we can conclude that it has a greater transforming capacity, but it lacks a DNA damage response part, which is associated for cyclin D1. So we hypothesized here that the G allele may facilitate clonal expansion after the translocation event and overexpression of cyclin D1. G allele may also affect the early evolution of T1114 multiple myeloma, because we observed a similar association in the precursor state of this disease. And most importantly, G allele is a major risk factor for AL amyloidosis, where T1114 is much more common than in multiple myeloma. So in AL amyloidosis, the frequency of T1114 translocation is over 50%, and in multiple myeloma it is about 20%. So that is also confirms the function of this variant in the T1114 type of myeloma and AL amyloidosis. So in this part, I have shown you an example how a germline variant may affect the MYC pathway, and I saw also another example how the germline variant affects cell cycle regulations. Other SNPs which we identified to be associated with multiple myeloma, so include autophagy and plasma cell biology, telomere biology, chromatin remodeling, and immunoglobulin production. So here are our collaborators for the colorectal cancer GWAS whole genome sequencing project. Professor Jan Lubinski from Poland is our main collaborator, so he has a large collection of familial cancer cases of 4 different cancers. Then in the multiple myeloma GWAS project, Hartmut Goldschmidt, Richard Houston, Martin Kaiser, Björn Nilsson, Karet J. Morgan, and Kari Stefansson and Peter Sonnefeld have been our main collaborators. Per Hoffman is an expert on this GWAS analysis and our longtime collaborator. Then the whole genome sequencing and data management was done in the core facility of our institute. Then Abhishek, Opul, Peping, Manda, Ayushi, and Lisen in our group were involved in this whole genome sequencing part and identification of the high-risk variants. Hauke, Mikuel, Polang, Supayan, and Kiara have been participating in this myeloma project. Kari Hemminki was the head of our division of molecular genetic epidemiology, which was closed last summer, and now he is a professor at Charles University in Czech Republic. Then in the bioinformatics part of our work, so as I told already, Matthias and Anaka have been our cooperation partners. So with that, I thank you, and I'm happy to take some questions.
Dilpreet, Professor Dilpreet will raise the questions, and I will check it in if we have question in YouTube. So if not many question, I will ask a question. So when the families are very rare, right? So earlier also publishing them without experiment or even with experiments were difficult in rarer mutations, yeah, rarest mutations, although they are very important. So how difficult it is now getting in, say, in 2020 or 2021?
To evaluate, you mean that to evaluate the function of the variants, or
if you find the mutation only in one family, suppose nowhere else, not identifiable in any other family of your data set or anybody in the world. So it's only
yeah, of course it is very difficult to convince people that it is a real contributor to the cancer risk, but it somehow looks, after our experience and also from other people who are working in the same field, so that the main cancer genes, the really high risk genes, so they have already been identified, like this BRCA1 and 2 in breast cancer, and then the mismatch repair genes in the colorectal cancer. And it may be that there is a large number of variants then which are individual for each family, so that each family has their own pattern of mutations, or that there is this one mutation which is the main mutation, but there may be then other mutations then also which then contribute to the risk.
Okay, so one important thing I forgot. Basically, by mistake, Neol has made you host of the meeting. Okay, so you have the full control.
Okay, so that's why it was there was some of these complications in between, but it worked anyway well, so that it's fine.
So basically, you can make Neol back as a co-host so that he can take over.
Okay.
Yeah,
so how do I do that?
right click. Can you see the participant ICDSB webinar 2020 on the right hand side? The participants?
Yeah.
Right hand side.
Yeah, let's share the screen and please help. I think that would be better.
Yeah, this one. Yeah.
Right click on it and promote to host. Right click.
Make right. Click.
Make host.
Make host.
Host. Yes. Thank you.
Yeah. Okay.
That's done.
Yeah.
So
thank you very much.
Yeah, Abhishek, can you hear me? I have a question.
Yeah, sure.
Yeah. So I'm Prashant, calling from Institute of Bioinformatics, Bangalore. It's a very interesting talk, and multiple myeloma is something which, we know that the mutational load is quite high, and what you have done, the meta-analysis from different Caucasian population, I would say, or different country. So what is something you predicted, or the mutational in terms of SNVs, is there some common, or is there some of those SNV which were very exclusive for even the population from different countries? So have you thought of it? This is the first question. And the second question is, have you seen the clonality? If we talk about the melanoma population, these are the different mutations. So did you go one step forward and see that what different clonal population which are common among different geographical region, or it may be very much, the similarity between the clonal population?
Yeah, so of course there are differences in different populations, but in those variants which we have identified, they are common for all Caucasian populations. And then so that in the Black population, there is a higher incidence of multiple myeloma, so there may be some additional variants or some private variants which then affect the cancer risk in that population, but there has not been so much studies on that yet. So that is one thing of these common variants, so that in the so it is always, nearly always, that one is studying the Caucasian population, and then one is identifying the variants which affect that population. Many of them are then also risk factors in other populations, but then there may be some where the allele frequencies are completely different, or they are very low in one population. So then, of course, they cannot affect so much of the cancer risk in the population level, but at the individual level, so that who happen to carry those variants, so that the risk is there. And what was your first question?
The first question was the mutational frequency, if you have seen through different geographical region. So how many of them were common, or is there any exclusively you would say that you found that prominently present in that particular population?
Actually, on this myeloma project, I cannot say anything. We have had in nasopharyngeal cancer, so that there are quite big differences between different parts of the world, so that in the Chinese population, the allele frequencies are different than in Moroccan population, where we have studied the same variants. And that then, of course, gives a different, like that one is now using the polygenic risk score, so then that one has to do for each population separately, so that one cannot use the Caucasian polygenic risk score for a population of India or China or Black population. So that
that would be interesting. I don't know if there is data from India because it's not that prevalent. But of course, the lung cancer is one of the examples we always speak, that the lung cancer, that EGFR mutation, it varies a lot between Caucasian and Indian and Chinese populations.
Yeah. And especially then also if there are some immune system related variants involved, so that they are differing a lot between different populations. So.
Yeah, and other questions. So I maybe missed it, and you might have explained, but I may not follow. So you picked 7p15.3 locus. So what was the rationale for you to pick that locus?
So it was just an example in this study. We have studied a couple of others, but then of course for this one, so that because its location, so that we using the publicly available data, we noticed that it is located in a region which is which should be a key regulator of this CDC, whatever gene it is. And then we continued with the functional confirmation that it is really so that there is this long distance interaction between this enhancer site and then this adjacent gene.
Okay, thank you very much. Really appreciate it.
You're welcome.
Thank you, Dr. Frosty, for answering those queries and wonderful talk. Any other questions from anyone?
I think there's no question, and we must, yeah, please proceed.
Okay. Thank you, Dr. Frosty, for your wonderful talk.
Yeah, thank you. And I wish you a nice continuation of the conference.
Okay, thank you.
Thank you, Asta.
Next speaker is Dr. Vihung Pan. Is he on call?
Yes, I'm here.
Okay, hi. The next speaker is Dr. Vihu Pan. He is Senior Data Scientist at Super and Super GmbH. He is responsible for various data type processing such as geographic sensor data processing, diagnosing with Python, R Shiny app, and in-house R package development, machine learning, model applications in agriculture data, neural network for multidimensional time series data prediction, to name a few. He has received many awards and honors for his work. He has obtained his PhD in bioinformatics from Kiel University, Germany. Now it is over to you, Dr. Vihung.
Yep. Hello. Yeah, thank you for this opportunity. So first, I would like to share my screen.
There's a green button.
Yes, I hope you can see my screen.
Yeah, it's visible.
Yeah, my topic is biology plus data science in real world. Thanks for inviting. Yeah, so my target audience might be the students who doesn't know what kind of data science or bioinformatics, what is a clear career for me to go. Yeah, but since I have heard from all of you and some talks, and since you are the audience who is online are very experienced researcher, so I'm not sure if that will benefit you for my talk, but I would really like to share some, especially some experience working in the industry. Yeah. So you can interrupt me anytime if you have any questions. Yes. So first, I would like to have some background understanding. Also, who are you? So if you could please do the pool, you can scan or go into this website, and there are some questions, and just for me to understand your academic background and yeah, how deep should I talk, something like that. I really appreciate if you do that. Yeah. Okay. Okay. It seems okay from computer science, bioinformatics, and the other science. Okay. Okay. Based on the data that I have seen, is most of you are from a science background. Yeah. Science background, biology, computer science. Okay, more. Yeah. Okay. Good. Okay. Yeah. Thank you. thank you for this feedback. Actually, it's quite awkward, like to talk to the computer whole day, but I would like to have some feedback. That's good. So, yeah, so I would like to introduce myself first of my experience. So I study math and statistics, math in my bachelor and statistics in my master. At that time, I do more ecological statistics, so like with biodiversity index in my master, and in PhD I go to bioinformatics. I focus on next generation sequencing, so which we have already spoke a lot. Yeah, that like gene expression, DNA methylation, and host microbiota interaction. So that's why that I know Pankaj there. We are in the same program, and we have kind of cooperation together. And after that, I go to I stay in Germany. Yeah, original from Taiwan. I stay in Germany, and then I go to pharmaceutical industry. So doing more biostatistics to compare the drug efficacy. If you're familiar with clinical trial, yeah. So what I'm doing is that I compare to the big cohort, and to see if the new developed drug is better than the old drug. And also I do the sample size calculation, and to say, okay, how many patients or how many healthy participants that we need. And the most important is that the more things I did every day is data management, which is I don't expect when I was in PhD. So it means that our data format should follow the structure of FDA. So we collect data from all different resources, like different clinic, different hospital, and when we want to merge together, it's a mess. So someone sent us a CSV file, someone sent us like SAS file, and what we need to do is that we develop some kind of UI designs like app. Then they can use a touchpad, and then we can collect all the data. So of course that need a lot of different new technique, I would say, or some like a how to say, data privacy things. Especially in Europe, this is quite important to have this. Yeah. So. And yeah, this is the company I'm working now. So it's a Supper and Supper. This is a consulting company. You can see that we are very diverse. This is a kind of a political correct in Germany. So we should have a different gender, different race, or different population, like Asian, Caucasian, Black, and that make our company more attractable. But in actually what we are doing, we are doing the consulting work. So it means our clients, they have a lot of data, but they don't know how to do it, or they don't have the human resource to do it. Then we go there, and then we help them to build their data pipeline and to answer their research question. Not only working for industry, but the other very big part that we work for government and university. So we have some project together with Freie Universität, so FU Berlin, Free University Berlin, and with so like a medical material, like AI and some computer vision project, we work together. So yeah, my responsibility. So my department is called Computational Life Science. So most something with life science is my job. So go into the outline. So bioinformatics versus data science, what I'm doing every day. So first, so bioinformatics, we can see bioinformatics is a very specific field. Yeah, this is very high level introduction. So might have like a omics study, like a transcriptomics, genomic, proteomics, metagenomics, and this is a field that I worked when I was in PhD. Yeah, like NGS data and how we make the gene expression, how we find the target gene, how we find the correlation between one another, how we find the loci for like a GWAS study. And yeah, this is called OMI study. And the other part is like a network system biology. So gene-gene interaction, protein-protein interaction. Yeah, and there's also it's structural biology, which means like a protein structure, so like a 3D structure. Another big one is like a database annotation, which is like I see like in the genome browser, and they build a database. They have a different name of the gene. They make that all together, and the people can search. And this is the bioinformatics field. What I understand, of course, there are some more fields which I'm not really good at. Yeah, maybe some audience here has a profession in different field. So my point is, when we say bioinformatics, this is very specific field. That means, for example, have experience in DNA methylation, epigenetic. I'm very deep with that, but if I talk about protein structure, I totally have no idea what does that mean. And also like have gene expression, but gene expression in certain disease type or in certain, I would say, yeah, gene expression in different setting, and it might be totally different, like in mouse, in human, yeah, in zebrafish, that might be very, very, very different. So all the mechanism, they are different. So I would say bioinformatics is you use some some informatic tools here, and then to deep into the research question of biology. So to be a good bioinformatician, you need to be very good at or not very good, but you need to spend some time to understand the biological background. But how about data science, which is another story? You can see here, of course, the domain knowledge is important. For example, like a text data, like a mobility data, like a transport. In Berlin, we have a shared bike and shared car, so you can use your phone. I think in India it should be as well. Like, and then how you collect the data, the transportation here and there, or this is different. And the finance data. Yeah, if you're in the bank, how you know that the people want to borrow money from the bank, from the debt, how I verify it. This is yeah, for the data science there should have some model. And like e-commerce. So e-commerce is like yeah, like Google, like Facebook. No, sorry. Yeah, not e-commerce. They are social media. Like e-commerce is like I don't know, eBay or Amazon. Yeah, they collect data how you buy, and they are not the only one to do that. Yeah, so they can collect a lot of data in very short time. And not only that, but also audio data is getting popular because nowadays people listen to podcasts, and also there are some identification. For example, if I go to IKEA, the customer service, and then they will ask you, okay, could you say your customer number or the items? And yeah, then I need to use like yeah, I usually do that to test my German level, because if I don't say that correctly, I say, please say it again in German, please say it again, and oh, maybe my tone is not correct, so I need to say it again. Yeah, this is audio data to identify these things. And we have like a geographic data. Geographic data is, yeah, it all merged together. For example, we have a project with a drone. So you have a drone, so drone go in on top of the field to see what happened in this field, and you have the geolocation where it is. So our project is we use the drone and to monitor the construction status of the highway. So we don't need to. So that means the supervisor, the unit, they don't need to go in person, but they have a drone go there, and then they can see, okay, every 2 days, every 5 days, what is going on there, what is going on here. and they can have a model of that. So it's, yeah, when we come all this together, actually, the interesting thing is the basic things behind that, the model that we can use to different data type. But of course, we need the domain knowledge. So for example, if I'm people with very good at text data, like, okay, Twitter, and I think after some time, I can be trained with mobility data. So the basic is more like the same, which is very different than bioinformatics, I would say. So let's zoom in biological data. So biological data, they're also very different. This is just my experience, like medical data, like in clinical, medical data, like their metadata, their background, are they smoker, do they smoke or not, male, female, we have epidemiologists, like this kind of medical data. Clinical trial is a different story. And the molecular or cell biology is like, we are talking about like gene expression, DNA methylation, and yeah, the thing. And also agricultural data is also a different story. Agricultural data, which I work quite often in this field. Yeah, it's like a geographic data plus biology data. So it's interesting. Yeah, you need to have some idea of an environment, which is soil composition and the weather, and to find the best combination. And ecological data, what I did in my master is like we need to count how many birds in this forest, like estimate how many, what is environmental change. So as the former bioinformatics students, and if you want to go to data science, so advantage is what the real data is, which means if you study, I don't know, pure statistics or mathematics, and you might not have an idea what the data is, which means you just get the data. You don't know data, how data to collect it, how data to process, how you lab, and what is the lab work it is. But as a bioinformatics, you should know that. You should have some idea about transcription, translation, transcription, and even you might have some chance to go into the lab to see how data be collected. This is also very important, and I think it's necessary as a bioinformatics student. And then you know the tools, like the machine learning, the statistic, even now it's getting popular, it's like a deep learning neural network. And then you know how to explain math to biologists, and which is also very important. This is an advantage. Yeah, because as my experience, I need to explain this to physicians. Say, oh, why this gene is important, what method I use, and how we see that, how we explain the result. And let's go to disadvantage. It's very hard to explain to non-bio people. I mean, I believe the audience here, most of them, we have a biology background, so to say, biology background. And it's okay as a gene expression. Everyone knows what is that. But it happens, I feel very frustrated when I try to go to industry and I try to explain my work to, for example, I have an interview with Lufthansa, aircraft company. I don't know how to explain. Yeah, there are 3 different layers. As one layer is gene expression, is genomic, and then the gene as a transcriptome, and then on top of that, there's the DNA methylation, epigenomic, and what is epigenetic? How that deal with the gene expression and how that change it. It takes like forever. I mean, even before I go into my real data analysis. So I need to explain the background. So yeah, this is a disadvantage if you want to step into the data science field, and you need to think how you explain and how you transfer yeah, the things. And yeah, as I say in the very beginning, bioinformatics is very, very specific. It's hard to transform this domain knowledge to the other. So I can give an example that for example, if I work for Uber, and then I can, after a certain of time, I know the domain knowledge, I know how it works, and then I can transfer my domain knowledge to, I can jump to the other company, which might have same service or similar service. Like in Germany, we have a Free Now, so the other one, or you can shift that to, okay, Foodora, like a food delivery. So you can, it's easier to transfer your knowledge from here to there, but it's not likely in the bioinformatic world. That's my opinion. So so if the students or the early stage of your career, you can think about you want to be a journalist or a specialist. So journalist is like okay, you got lost in forest, you need tools to cut, shelf, chain, just for surviving. Then you use this Swiss knife. But scenario 2, you are sitting in front of dining table and you just want to drink soup and you just need a spoon. And these are different training. So some people saying, it depends. In data science, we have these two different very big area. So some people like me, I'm a journalist, so I know that I have different experience in different fields. But if I go into specific field, I'm not so good. I mean, I'm good, but it's not so deep to understand how it works. And that's very good for consulting environment and this and there. For the other type is I only focus on one field. Like I only focus on how to analyze the text. Natural language processing, like Twitter, and even some scientific article, and like this. It's very, very specific. We can think about what is the best way for the career. So I still have some time. I want to share what is the differences working in the industry compared to in academic. So industry is from prototype to production and application business thinking. So the very important things I have learned in industry is that how we apply it, what is the market, how we change the human's life. And this is the first question, how we do that. And it's not only, in academia, we can think something which might not be really realistic, or even it is fail, it's fine. I did a lot of different projects in my PhD, but very few got published, maybe one or two. The other just, I don't know, put it somewhere in warehouse. It didn't work out. It didn't work out. But in industry, you need to think, okay, that should be used in human life. And also, like going to a production is a different things. For example, that I can write my code by myself and only me, myself understand what is this. When I was in PhD, this is how it is. No one will check my code. No one will go in there and then change. Very few cooperation opportunity to work on one project. But when I work in data science, I think that is totally impossible, because I need to communicate with like a app, how to people build the people who build the app, the people build the design, and also doing with together with software engineer and this and there. So that is very important. So not only we build a model and the accuracy 0.96, that we are fine, nothing else. No, we need to bring that into production. So how the front user can use that, that is very important. And also automation is another point which is very different than working in the industry. So automation means if you do this several times, you should automate this. I was so surprised when I come into the PhD program, and one of my supervisor, who is very experienced scientist. And they say, how you deal with the data? So I have a big Excel table, and then I cut here and then copy to here. And then, yeah, I change here and I mark here. And then I do it for different, so for many times, just get my things done. And also, he is a microbiologist. I'm so surprised about it. It won't work. I mean, in industry, what we need is automation. So you press a button and everything goes. And this I would say this is like a 60% or 70% of time are doing this job, because when data come in, we collect in different database, and how we go into the analysis, we need to automate the pipeline. And also like profit. You need to discuss with the cost. You need to discuss even the demand on the market, and we need a sweet daddy to like sponsor us. Like in our case, we need to like pitch. We have some idea and we need to go there and say, okay, our method is the best. So like profit, I would say more in the marketing your job. I can share one of my project. So one of the project is called automatic pipeline. So our client is it's a chemical production, so chemical company. They produce a chemical product for the plants, and they collect the different data. And the goal is they want the non-technical user, for example, their product manager, their business sales. They want them to the product manager or the sales. They want to know how their data look like. So before, what they did is they have a data. They have a specific product name ABC 123, and then they collect all the data and then put this data to the data science team in the company. And then after a certain of time, they create a 15 pages report, and then you can see and what happens in your product. But now, it's so every product manager, they have their own product, so it's kind of redundant work. So our idea is to how to automate this pipeline. So our challenge is that data is dirty. We have different systems. We have database system, and how we modeling and how we interpret the data. So what we do is we use the RShiny. So we have interface that can connect to database, and we use RStudio to do that. And the front end, of course, this back at the front end is a web browser. So you can open the web browser and you can type the information, for example, which field, and for example, which target, which crop. And then you can have an overview of this and what is percentage. And even more than that, we can go to, for example, here we can see in Africa, we have a different field for they make the experiment. And if you select this, and then you can see the temperature out of that. And also like the rainfall, it's a weather. So you can have the information, just you drag, and even you click this, you have actual information. And then you can have the modeling. So we have implemented random forest and X-ray boosting to different model. What we can do is we can see the important factors for product performance, like in what is the best temperature for this chemical product, and what kind of plants is more efficient for this chemical product. And then we can calculate the interact effect, multi-ingredient effect. Then we can say, oh, this crop stage is most important one, and compared to the other internal. So based on this, we can provide the information for the product owner, and they can have further deep go into what kind of adjustment for their product they need. I would like to stop here since I don't have much time. So you can go to the website. We have a different use case, graph database verification and optimization, and explainable AI for trial field analysis with management. And we have different use case. My department is computational life science. We have in the other department is geoscience and the manufacturers. Yeah, so mechanical engineer. So yeah, you can look at it. Yeah, so conclusion, choose your love and then you love your choose. So this is my LinkedIn. I'm very happy, and my email. I'm happy to have all the different kinds of cooperation, working in interesting project, and yeah, some discussion. Thank you very much.
Yeah, thank you so much, Wei Hong. So this was quite very informative, and you were actually talking to the people. So that's what I really was enjoying it actually. So
Thanks.
if you look at the comments, I read it for you. Very informative. I'm gladly listening to it. Very informative. Good explanation. Good motivation. So yeah, so actually people are enjoying it.
Okay.
It's generalized, but yeah, I believe it's going to help a lot of people choosing different fields that they want to go in future. So yeah, maybe can I go for a general question? Can I?
Maybe I ask for some question.
Sure.
Yeah.
So when did you realize when you're doing research that you either want to be general Swiss knife, or you want to be a spoon, and then when did you rebrand yourself? What time you started rebranding yourself? Because it's a question of we are taught in the learn Python, learn English, learn Russian or so, but we never taught how to brand ourselves, because unless you repackage yourself, it's difficult to sell. And so when did you realize, and when did you started preparing for it? How much time it took you to prepare from transforming from PhD to industry friendly person?
Yeah, so yeah, thanks. This is a very good question. Actually, I don't have an exact answer, but this depends on, so this all related to my background. So you can see my background, I was trained as a mathematician and then go to statistics and bioinformatics. So I have a very broad idea of how we deal with data. And and also I think, it all depends on how you start your career. I don't know, because I start my career with a consulting background. So first I say in the pharmaceutical industry, it's very general, pharmaceutical industry, but we need to learn different things. And now I'm in the very broad, I would say, consulting. So based on this, I would say I have to, because otherwise you will lose your business. So that's also a disadvantage for myself. I'm a journalist. So yeah, when we got what we don't have is we don't have the domain knowledge, very solid domain knowledge from the specific industry. But what we have as a journalist, we have the idea to deal with the data. And I think most of the industry, they don't need very deep model. I mean, very deep understanding of the model. They want the first product, the speed is most important. You can think, I mean, in the industry, the first one, like Uber, they have this product. Even the algorithm is not optimized yet, but they can occupy whole the market in the whole world. They only want to have a first product. So in this case, you don't need to very deep into the knowledge. But when they grow up, they have a competitor, they have this and that, then they need to, some people to go into very deep. And in that case, you need a specialist. Yeah, so I think that's my answer.
Thank you.
Okay, so are there any other questions? So yeah, thank you so much, Wei Hong, again, and I think we really enjoyed it. And also in future, we look forward to your talks when you visit India in person. That's
yes,
something we really look forward to. All right, so have a nice evening.
yeah, thank you.
Bye.
Bye.
Monish Wokan and Deok.
Danke auch.
Bye.
Bye.
So let's start with the next speaker. Professor Muthuraman is the next speaker.
Should I share my screen, Pankaj? Or?
Yeah, so we can start. Let me first introduce Professor Muthuraman. So Professor Muthuraman is holding a BE degree in Electronics and Communication Engineering from University of Madras in India in 2002, and then he he moved to Germany to do his MS in Digital Communication from Christian Albrechts University, Kiel, Germany in 2006. And then he also did his PhD degree in biomedical engineering from Technical Faculty and Department of Neurology of the same university in Kiel, Germany. Later in 2010, he joined the Department of Neurology, University of Kiel as a postdoc, and then in 2013 became a senior postdoc. Since December 2016, he has been with the Department of Neurology, Johannes Gutenberg University, Mainz in Germany. Now he is currently an assistant professor and also heading the Department of Biomedical Statistics and Multimodal Signal Processing Unit. His current research interests include the mathematical methods for time series analysis and source analysis on oscillatory signals, function of oscillatory activity in central motor systems, biomedical statistics, connectivity analysis, multimodal signal processing, and analysis of EEG, MEG, and MRI and EMG structural data, signaling data, and also network analysis on autonomical MRI and DTI sources. And function also he's doing work in imaging and machine learning and deep learning. So he's an expert. It's a great pleasure to have Professor Muthuraman here. So today he's going to talk about structure and functional network characterization of neurological disorders in patients. So welcome, Professor Muthuraman, and now please, stage is yours.
Thank you for the great introduction, Pankaj, and thank the organizers, both Pankaj and Abhishek, for inviting me to giving this talk. It would be, of course, very nice if it would have been in India, but of course, it's also a nice opportunity to sit here at home and talk. So can you see my slides?
Yes.
Okay. So I would give an overview of our research here, what we are doing in our group. That's why it's titled Structural and Functional Network Characterization for different neurological disorder patients. So first I give you some introductory slides and then go on with some real application of data, because what we do is that we try to analyze these data and then we go into the application of helping the clinicians basically to identify some markers for their clinical therapy. So speaking about clinical therapy, what we do in minds is that we call this therapy a deep brain stimulation. So here's a picture shown below that you have an electrode which is sitting inside the brain, and the person, for example, in this case, a Parkinson patient who has problems, for example, like tremor of the hand, they will be implanted with an electrode in the brain, and then it's being connected to this stimulator, which is then sitting under the shoulder bone, and they are then stimulated 24 hours, so that the good thing about this type of therapy is that they don't have then this tremor anymore. So this is done very often, and this is some DBS electrode. How does it look like? So you just give you an idea how big it is. It's about 7.5 millimeters are the contacts in this electrode. It can be in any way stimulated, and once you stimulate the brain, then you can also model how the stimulation field looks like. And there are, of course, additional developments. You have directional leads, meaning that you can project the current to a certain part of the brain, and non-directional, then you have a circular kind of current. And what we do is that when we have this active contact location, as we call here in the brain, then we look at different connectivity fingerprints. So we differentiate this connectivity into two: structural, meaning anatomical connectivity, which is present in the brain, and functional connectivity means that you do a certain task and then some regions light up and some regions don't show any activity. And then we can, of course, in the case of structural connectivity, we can isolate these running fibers from where we are stimulating to other regions in the brain. And we, using these fingerprints, we can either associate to the, let's say, to the voltage, how much we are stimulating, or any type of clinical score, how bad are they in this disease. We can either do an association or we do a prediction, to that at the end we give an individualized therapy to each of these patients. So I give you a few more introductory slides to give you an overlook of what kind of data we are having. So this is a slide where we show, for example, if some of you don't know about the regions in the brain. So this is the region in the brain which we are stimulating, which is very important for motor tasks, the hand or for the leg, and how it is connected to other regions in the brain on the top of the brain. And this is very deep in the brain. So we have several different modalities. Probably some of you have heard about it. So tractography means the diffusion tensor imaging. How does it, the tracks are running anatomically, or functional magnetic resonance imaging. We look at functional data. How is the bold, meaning how does the blood oxygen go up and low? Or we have positron emission tomography, and we do a lot of EEG, meaning electroencephalography. And you can look at these connections. Most of these modalities are able to find those connections, but there are very subtle differences in these connectivity. So the good thing about this type of operation is that we are putting an electrode inside the brain, which is commonly not possible. Usually it's only done in mice or other animals. But in this case, we have the capability to put in the human electrode, and then we are able to also record directly here. So basically neurons, you can record them directly. It can be single or multi-unit. Or you can also record, let's say, local field potentials, which is a summation of all the neuronal activity. Or we have the capability to have electrocartographic grid. What it means is we put a strip of electrodes on the top of the brain, and then we can also look at local field potentials. Or we can directly on the top of the scalp, we can put an EEG electrode and record the surface activity. So these are some of the methods what we use. We call them electrophysiological proxies. So just go into one or two of these measures. Let's say coherence, which simply means correlation in the time domain. Here in the frequency domain, we just want to know which frequency is present, let's say in the EEG or in the hand movement. We can also measure kinematic senses, what is the movement or muscle activity, and then we can look the coupling between this hand and the EEG electrodes. Or we can look at, let's say, phase amplitude coupling. This is also a type of coupling which relates the phase of one signal to the amplitude of another signal. And of course, probably other techniques are known to you, like power spectrum or looking at in the time frequency domain. So we first start with the structural domain. So this is an MRI data what we get from the patients. So this is done before the operation. And then what we do is we try to disentangle each of these regions and then try to estimate something called cortical thickness, which simply means that how thick is the cortex in this person. And using this parameter, we then develop correlation matrices and then use this for either group comparisons to see whether there is a group of people who have a very good outcome after this DBS, how is the thickness in that group compared to a group where there is very not that good outcome. We look at network topology and use this also for the comparison. And finally, feed this all into support vector machines and try to give us a good prediction so that we can use this to say to the clinician whether this particular patient will be able to profit from this type of therapy. I give a quick introductory slide about graph theoretical analysis, because in this study, we used this type of analysis. And here the brain is divided into nodes, and the connections, we call them edges. And we try to build the whole brain in a type of graph. And then using this graph based on the assumption what we have, either it's a single region or two regions or many regions, we then of course go to a multivariate analysis and then look at the network, how good is the network compared to a patient to a healthy control, for example. So we looked at two different group of patients. We call the superior outcome means they profited well with this deep brain stimulation. Moderate outcome means they are not very well profited. So we want to see the difference, what happens in the brain. And these blue regions are the regions which are then difference between the two groups. And some of the regions, if you don't know what about brain, then meaning here is the frontal part of the brain, they are responsible for a task. And for example, here the occipital part, meaning it's responsible for the eyes. So these are regions which are highlighting that there are differences in these two groups. And we use this to develop some network parameters. We call them clustering coefficient in this case. It's simple, it's very similar to the clustering algorithm, what most of you know. So this is a region in the brain, and if it had connections to all the neighbors, then you have a low. And if you have additional connections here, the clustering coefficient is high. And the same way as degree, low means less, high means higher degree. And we again look into these two groups and look at this degree and clustering and find out regions which are significantly different. Again, colored here, they have low degree, and the CN color here, for example, have high centrality or high degree. The same way is plotted for clustering. So once we identify these regions, then what we do is we fit that to support vector machines, and in the violet is shown for the network selected regions, and for the yellow or orange is shown for the volume at deeper in the brain. And we see how good we can classify these two groups. We achieve about 75% accuracy, and always in the clinical point of view, if you want to make the clinician believe, then you need to train your algorithm in one of the groups. That's why we call this discovery, and then test this algorithm in an independent group to show that this algorithm works. And there we, for example, achieve about 80%. And here are the receiver operating curves and the area under the curve. So each region, even if one single region we are able to take here, we can get an accuracy about 82%. So this is looking into the cortical thickness, or thickness of the brain. We also look in these patients how this tractography is what we call, we image the brain with a special sequence, and then we can have these nice pictures how good the tracks are going from a specific region to other regions in the brain. And then, as explained before, we again develop a connectivity matrix and then look at several regions in the brain, how good they are connected. So here again, we use this frontal part of the brain, these connections which are shown here, and these circles are regions. And then we use this for prediction. And again, you can see, for example, in the frontal region of the brain, you get a very good prediction of 82%, meaning we can use the patients even before they're going into the operation theater, how good they are able to profit from this type of treatment. So this is very helpful for the clinicians. Now I move my focus from the Parkinson's disease to multiple sclerosis. So these are patients who have lesions, we call them, in the brain, which is then not functioning really well. So these white spots here are called lesions, which means they are not doing the function what they're supposed to do. So what we want to find that is that this topography of these lesions, as shown here, do they go well with the clinical parameters, what we can measure, whether they are progressed in their disease or do they are in the beginning. Can we predict this disease progression using this lesion topography? Here we used independent component analysis as shown here. We use structural images and then look at the shared contribution between the lesion topography and we call the atrophy, which means again the cortical thickness. How does it change from one time point to another time point? And then we can look at different ICA or independent component analysis patterns. So we found 3 different patterns. This is a deeper region in the brain, and these are regions which are higher in the brain. And when we find these patterns, whether these patterns are useful to classify the disease progression. And here on the left side, we show again the receiver operating curves. So this is a pattern which is identified in the cerebellum, which is a region which is very important for any kind of action what we are doing, for example, the arm movement or the leg movement. And here we can see that the cerebellum is then giving us, as shown here in green, gives the best accuracy in predicting the disease progression. And here is a connectivity map. How does this pattern is related to different of these regions in the brain, shown in different colors. And what we did is we went one step further, taking this cerebral pattern and then fed that into convolutional neural network, and then try to find out whether we get good accuracy with this cerebral pattern. So again, here we show for the 3 patterns which I showed before, and here is the axis shows the prediction accuracy, and here are the number of subjects. So we used 50% of the subjects to do the model, and then use the other 50% to do the classification. And here, for example, again, the cerebral pattern, we get an accuracy of about 95%. And this is then predicting how progressed they are in this disease stage. And again, as I said before, we need to always show that in an independent cohort, so that we then used another cohort of patient and then did a similar analysis. There we found only 2 patterns, but still above 90% we were able to classify the accuracy, classify the patients who were progressed and who were not progressed using the cerebral pattern. So these are studies which I showed on the structural and the domain. And now I move on to the EEG analysis. What we do is we call this an inverse solution, because what we record in the brain is EEG or MEG data. So this is how the EEG electrodes looks like on the brain with these blue circles. And then the MEG coils are shown here. And what we get is we get time series of a certain amount of time, like say here 2 seconds. And then what we want to know is if we do a certain task, let's say if we ask them to move the finger, we want to know which of the brain regions are active. So for that, we just have this scalp data. We want to know from where this signal comes from. So that's why we call this inverse. So we have this data and we want to find this. And what we do is we call this beamformer analysis. And this beamformer analysis is similar in other domains, probably if you know, like for example, radio frequency domain, there also they use this beamforming, and then we try to locate this source. We always use the EMG, that's the muscle signal, as our reference to locate the source. And what we do is we trick the algorithm to say, first find the most activated source or the strongest, and then we tell the algorithm in an iterative way to do as a black box and then go further with the other sources. So in this way, we will be able to get the other sources, even deeper regions in the brain. The nice thing about this type of analysis is you can also get a signal out of this source as shown here, and then you can do also connectivity between these signals. Here I show the connectivity analysis, what we do generally in our data. We call this effective, meaning causal. So we want to know the causal information. Here I show a simple model simulation. So we have 3 time series from the brain. And what we show here is this time series X2 is giving information to X1, and the same happens with X3. And they have a time dynamics. So this connection, which is shown here, has a waxing sine wave, for example, or the X3 is a triangular wave. And we want to know this dynamics and also know the causal connectivity. In some algorithms, what we get is you get some very high values. For example, let's say Granger causality, then you get high values here and other values are lower, which means it just shows what amount of connectivity is being between these 2 signals. But we wanted to also look at the frequency at which is happening. Then we also get the frequency information. And what we finally do is we also look at the time domain. So at the end, we also get this waxing sine wave and this triangular wave, and all the other connections are 0. So there is no connectivity in these time series, but in these 2 time series, we get a very good connectivity estimate. So we use this type of analysis to again differentiate between patients and healthy controls. So as I showed the pipeline earlier, EEG, EMG, the coherence, and then we do the inverse solution, and then estimate that in a certain atlas, which gives you good connectivity maps. So here is Parkinson's disease, who have a problem with the hand, let's say tremor, and there is another tremor group, and here is the healthy controls, which means they imitate the tremor in these subjects. And then we find subtle differences in certain regions of the brain. And then we can also find, using this analysis of connectivity, to show which are the differences. So this is healthy control, and these are the patients. So you can see there are differences in the connectivity, because you can use this information then for stimulation of these patients. In another cohort of patients, as in another study recently, we were able to show that these are, for example, the electrodes which are sitting deep in the brain. And these are 2 groups of subjects where we stimulated in different frequencies. And here is again the electrode. And as I said before, how much of the tissue is activated can be modeled. And then we look at the correlation between this and regions in the brain. And then finally, we looked at cross-frequency coupling, meaning that how one frequency is dependent on another frequency. And then we were able to show that in different regions in the brain, so this is a certain oscillation. If some of you don't know, beta is around the frequency of 13 to 30 hertz, and gamma is from 30 to 100 hertz. And here is the stimulation, what is used generally in the clinics. And here is off, so we switch off the stimulation. That's really nice in these patients. You can just switch off and the stimulation. And we also use low, means we stimulated 20 hertz lower, and high means we stimulated 20 hertz higher. So you see this clear dynamics here in this group of patients. So there's a reduce in this beta and an increase in this gamma. So this we can find in most of these regions in the brain. And this region was used as a control region to see whether this doesn't happen. So it doesn't have any dynamics. So this is true what we are seeing in these other regions. And of course, what we are measuring is EEG on the top of the head. And I'm showing you regions which are very deep in the brain. So to validate this, we also were able to record directly from the deep of the brain and the EEG simultaneously. And here we show a time frequency graph. This is time, this is frequency. And you can see the dynamics of this coherence. So coupling between the brain and the muscle have a similar dynamics for the recorded signal and the reconstructed signal. So this holds true and shows you that the analysis what we are doing is validated by this type of analysis. And using the next 3 slides, I just show in our unit what we are doing. So this is what we are trying to do. We use all these different modalities. We bring that to a network perspective. And we also use, for example, mouse data. And then what we want to do is build predictive models and then think about critical time windows where a therapy would be useful. And now, as we've seen a lot today about other things like gene expression, proteins, so this is also one field we are touching in new, trying to find out also these kind of modalities to develop them as matrices and then develop as network parameters and then look into deep learning algorithm to see whether we are able to use this to also for clinical use. And finally, I think what we want to achieve is shown here. So we use all these parameters, we identify different network changes, and we use model approaches and deep learning approaches. And then finally, if we can give the clinician a therapeutic option, let's say either invasive or non-invasive, or even at some point we can say we could change lifestyle modification, let's say more sports or stop smoking, for example. So this kind of things can help the patients in a way that they can then try to go ahead in the disease what they have and not to progress. Yeah. So this is my final slide. So of course, all this work is not possible with a great group of people. So we have 5 PhD students, and they are 2 of them already left. They finished with us, and they are in US now. And we have clinicians and we have technicians. So it's a group of people very highly dynamic, and that it's possible to do such kind of analysis. And here are the collaborators, and here are my sponsors. Thank you for your time, and I thanks also for the organizers to giving me this opportunity. Thank you.
Yeah, thank you so much, Professor Muthuraman. So, yeah, questions. Time for questions. If you have questions, please write in the chat box. Okay, so maybe I can start with how challenging do you find in terms of integrating such kind of diversity of data, like you have imaging data, then you have signaling data, and also now you're planning to use also gene expression and other omics data. So as years of experience, what do you think, can you maybe comment on different sorts of challenges you find in
Sorry, the connection I think is really bad. I don't hear you. I think I hear part of the question. Of course, you want to know how the multimodal aspect plays a role in use it for prediction, right?
Yeah, in that line. So if you're planning to also include now different sorts of biological data in addition to imaging and signaling data. So what I want to know is, because you have now years of experience, so what sorts of challenges do you see in the coming years?
Yeah, I think the challenges what we have is definitely to have an expertise of all these domains would be very difficult. So of course, like you gave a good introduction, I have electrophysiological and imaging background, but of course these domains of biology or gene expression is very new to me. So those things I think we need specialized people who are then able to do to also understand the data. Of course, we can feed all this type of data into an algorithm and see what happens. But at the same time, we need to also know the background, like also explained earlier from earlier talk, that we cannot be a master of all of them. So I think we need to separate and expertise fields and then try to use this for a further analysis. We cannot, I think, be a master of all of them.
Yeah, I totally agree. I think that this is one of the challenges now in data science, facing having people with multidisciplinary knowledge. So there is another next question is from Brinda. She's asking that in CNN, what types of activation function and optimizer were used?
So in the case of convolutional neural networks, what we are doing is very simple algorithm. So we are trying to train the algorithm based on long short-term memory activation neural networks. And then because we want to at the end predict the clinical disease course or any kind of clinical parameter. So basically we know the output and we want to use this data what we have to predict. So if we're able to train the algorithm with these long short-term memory maps or neural networks, then I think we are having a very good prediction. But of course, as I said, this is now currently what we're using. So we are also in the learning curve in deep learning. I have some experience, of course, with machine learning, like support vector machines, but deep learning is hype now, and we're also trying to indulge ourselves, and we're trying to learning. So it's a learning curve. Probably there are much more algorithms which I don't know, which are performing better. But as we are trying to use it in an application-oriented design, so we try to use which is most performing better.
Okay, thank you. So I think we can take one more last question. The question is very interesting. So recently, Elon Musk recently unveiled Neuralink, as we know, it seems similar to your objectives. So how far from therapeutic use in your research?
I understand about the Neuralink, but the last part I didn't get. Sorry, I will just look into the chat function. Or
Okay, so I think the question is probably more in terms of how we can apply to it in therapeutical way, the kind of work that you are doing. Are there any scopes to apply in that direction?
yeah, definitely. I think, of course, what we do is we always try to replicate this in many different patients. And I think some of these algorithms could be useful, like they show in Neuralink. Of course, they are having that tested in pigs, but not in humans yet. But we have the capability to test it in humans. And they are looking at single neurons, which is actually highly detailed. And it looks very promising what they showed. But I think to get it to a big amount of success, they, of course, need to do it in humans. And the algorithms, what we show here is tested in different cohorts of patients so that it can be, of course, used as an additional parameter to what they already have.
All right. So I hope that answers that question. So maybe then we move to the next speaker. I would like to thank again Professor Rahman for joining us. It was really wonderful to have you.
Thank you again.
Okay, so let me take a look at the program. So the next talk is from Professor Mohammed Asif Khan. And now the next coming talks will be chaired by Dr. Neha Jain, Assistant Department of Science and Bioengineering, IIT
Hi, Pankaj, can you hear me?
Yes.
Yes, I'm here.
So maybe you can
Yes, but sorry, Pankaj, your voice is not clear, but do we have the speaker added yet?
Leon?
Hello?
Yeah, I cannot see Mohammed Asif.
Yes, yes. Yeah, he has not joined yet.
Yeah, he's not joined yet.
Yeah.
Dr. Abhishek? Okay, so we can take a break for 5 minutes and then we try to connect back.
Sure.
Okay, so basically, Dr. Khan is busy, has some confusion about he's in Turkey, so basically time zone difference. So I will be speaking initially, and he will join later on. Later on, so in 5 minutes, I will join. Let me see my presentation.
All right.
Okay.
So I will be joining in 5 minutes. Let's have a small break, and then I'm almost ready actually.
Then Abhishek, shall we start at 5?
Yeah, we can start at 5. So
Okay.
because he needs their time, little bit time. So
Sure,
that
sure.
have a bio break for 5 minutes.
Yeah, yeah.
Coffee break maybe.
Okay, Neon, I am back. Neol, sorry.
Yes, we can start.
Yeah. So I can share the screen.
Are you able to see my slides?
Yes, yes, we can see.
Okay.
Hey, can we start now?
Yeah.
Okay, so just 1 second. Hi. So welcome in the evening session. We have Dr. Abhishek Kumar from Institute of Bioinformatics with us. Dr. Kumar has received his PhD in bioinformatics from the University of Bielefeld in Germany. Followed that, he had a postdoc stint. Dr. Kumar has, to his credit, sequencing of largest number of marine fungal genomes in the public domain. He has been a recipient of prestigious Ramalinga Swamy Fellowship awarded by DBT, and currently is working at the Institute of Bioinformatics, where he is employing multiomics approaches for studying cancer and neurological disorders and developing computational pipelines for detailed analysis of genomes. Today, he will be talking to us about deep learning applications in bioinformatics and genomics. Over to you, Dr. Abhishek. Welcome.
Thank you, madam. And it's my pleasure to have go live in front of audience. And thanks for joining. And so I'm working at the Institute of Bioinformatics, which is a non-profit organization, and it's a premier institute in area of translational bioinformatics. It started in 2002, and it is a major proteomics center in India, and it has produced many good researchers and many uh, uh, employees who are working in different proteomics lab throughout the world, including in India. The staff scientist or trained in proteomics, and then as everything evolves, the institute evolves. There are new faculties. There are a lot of other faculties. They do a lot of other areas of research, and you can go to our profile ibioinformatics.org and check there are different faculties. And so we have about 10 to 12 faculties, and we have also centers across different hospitals in Bangalore, like Nimhans, which is Neurological National Center. We have laboratory there as well. So basically we are a translational research, and if you look in our profile, you understand, in my opinion, it is one of the leading translational research started in just after human genome sequencing. So there I'm working, and I'm faculty at Mahe, which is the orange logo is there, and I call my lab as a computational genome biology, and which in COVID-19 I started working with viruses. So we have slightly changed the logo with virus like look. And today, what I will do is that I will talk to you about deep learning and its application in bioinformatics and genomics. I'll give you two examples. So this is how my talk is organized. I'll discuss about, since a lot of students are there and a lot of people who understand deep learning, and they want to see what kind of examples we employ in our laboratory, or want to employ in our laboratory. Okay, so I will discuss briefly about deep learning and what is ensemble deep learning, and then what I call mere deep, and image deep. So two examples I will give. On partly we were working on MIRDL, and then we want to work on image analysis. So but we are in a period of transition from sequence to data, and now we have taken a good example of image and sequencing data merged together on a same analysis. So I will show you an example there, and then I will introduce you to the slightly who did the work and who all are in the team, how we are growing up, and what are our infrastructure. So my team is called Computational Genome Biology, and which I have named, and based on my experience and my understanding. So what we do is we analyze the genomes of eukaryotes, and so marine we have analyzed, insect we have analyzed, fish we have analyzed, and now we have received grant for 2 new species. So in 2020 we received 2 grants to analyze 2 different eukaryotic genomes which are not listed here. So that will come to the end, and then we are when you are analyzing genome, actually you are having a large amount of opportunity to play around. If you want to look in evolutionary genomics, the genome is a platform where you can easily look into evolutionary genomics. So we have in the past we have studied intron evolutions, serine protease inhibitor phylogeny, and GPCR phylogeny. And at the same time, when we are doing the marine fungal genome analysis, we are looking into future drug-like compounds and natural product discovery. And there we are doing fungal genomics, marine fungal, and even now we are working on lichens. Then at the other end, we are doing clinical genomics with human genome, cancer genome, and trying to find out the one potential marker, genetic marker that will lead into a certain disease. Overall, when you see that what we all do is that basically I want to understand how much genetic and chemical diversity exists on our planet, and that's the ultimate goal for which I work. And slow progresses in each direction is pushing me to that direction, and we do generate a lot of data, and we do analyze a lot of data. So basically we call ourselves genome data scientist or genome data analyst, and depending on what skill an individual person is possessing. So during COVID-19, as I told you, that we have set up a team which is analyzing SARS-CoV genome. So that is viral genomics coming into the picture. But today's talk is on data science. So basically, biological data is very heterogeneous, and it comes from DNA, it comes from methylation, it comes from RNA-seq, it comes from tumor genomes, it comes from various different arena, and data is actually doubling. If I only count on this scale, initially drawn by Stephen et al., and I have modified it as our requirement. You see that data is very rapidly increasing, 6 to 7 months is doubling. But if it's like every 2 years 1 month is taken out, so data is doubling almost now in 6 months, and that is just genomic data. I'm saying other omics, if you add genomic, proteomics, metabolomics, that data is massive. So it's becoming bigger and bigger. So that's when biology is entering into big data. But big or small, data is data, and if you can make sense of the data, then only it is useful. So just imagine you have an USB stick of 5,000 songs. If you don't know what is inside it, it's garbage. And so if you have the knowledge what is inside it, how you can utilize it, and even better if it is musical data, how you can combine and make sense and make a new music out of it, it will be much better. And so the data is the money, and data who will be able to utilize will be the richer person. So Google is rich just because of deep data. And so rich in the knowledge and rich in the money, both are important. And so most of you are aware the deep learning is deriving from human brain. And the last speaker, Dr. Muthuraman has already shown you those examples, but I'm just giving a simple example. You give some 4 or 5 inputs, neurons will process it, give you in a hidden layer in the brain, and comes out as output. And that's how the deep learning is inspired. And there are very good literatures. Oh, sorry, the title is different, but very good literatures are available about deep learning primers in genomics. And I will take some of the example from here. Why I'm putting these slides, because I want readers to read about it when they want to learn about it. So what is the deep learning approach is doing in the genomics or in sequence field? You have a sequence parameter, you label them 0 and 1, only 2 terminology, yes or no, Boolean way, and you generate a training data, you generate a test data, and you generate a validation data, and then you apply your machine learning architecture. You can apply CNN, you can apply RNN. Depending on what you apply, you will evaluate in terms of how many true positive you have, how many false positive you have, how many false negative, and how many true negative you have, so that you can calculate your accuracy in terms of precision and recall. And then you make a finally try to understand what is actually happening with the data. And that's the broad spectrum, and you can read more into Zhao et al. In Nature Genetics. And then this is the same one. And then I went back, I guess. Yeah, so then there is a new paper just now came, and many people have understood also. And it's today morning also somebody had discussed that now you have a deep learning and you apply ensemble method to it. And that's when Perry Young from Australia, who is a pioneer in deep learning and ensemble learning, has provided information about a very big review. And what he's saying, I have marked it, that let's come as a community, come together, machine learning researcher, bioinformatician, and biologists, to develop a better understanding of ensemble based deep learning, and develop unachievable bioinformatics methods which will surpass the traditional problems. And that's when I have a marked in very light color. Yeah. So okay, so this with this we have a background.
Okay.
What is ensemble?
Thank you.
So you have one area. Bioinformatics is a very big area, and artificial intelligence is a bigger area which is merging together. As you see, green and reddish here. Artificial intelligence has sub area machine learning, deep learning, and within the deep learning, now you can combine ensemble learning. I'll explain you how ensemble learning is working, and then that ensemble learning together with deep learning is now called ensemble deep learning. And then this is modified from this paper. And what is happening in ensemble learning? You have data, you can beg to get a board function, you can boot you do boosting, or you can do data perturbation. Three different kinds of ways you can do the ensemble algorithm. So basically you have separate ensemble algorithm, you apply it to deep learning with the deep learning algorithm, and then that's how you will get ensemble method popped up into deep learning, and that will solve your problems, like if your data is very small and if there is some imputation required. So all these problems can be resolved with the deep learning, and this is getting fastly resolving your problems. And so there are different ways of supervised and unsupervised learning. I will not go much in detail. What is doing that you can combine yeah. So am I visible? Yeah. Not sorry. Am I audible? Okay. Yes. By mistake it happened actually. So you can have different input layer, different output layer, and that layers, combination of layers will define So in the C case, there are two hidden layers. So that combination will define when you're defining your strategy, what is your purpose. You can actually define those which layer you want and how you want. There are 6 different models, and one, if you are from computer science and algorithmic area, or want to understand, you go and read those paper. And then and obviously it's, again, I said, let's community come together. If I'm not understanding something, I have to ask help from others. So so I often go to a computer scientist and ask, okay, I'm doing this, how can I do? I am unable to do it. And that's when you grow. And so you want to see how much deep learning has imbibed into bioinformatics. This is an example, but I can say there are at least 500 papers on sequence analysis and genome analysis itself. So here are some sequence analysis. It is used in a sequence analysis, gene analysis, genome analysis, sorry, and gene expression of structural bioinformatics, proteomics, systems biology, multiomics, and bioimage analysis. I will give you an example of sequence analysis and bioimage analysis today. And then different combination of ensemble method and different architecture of deep learning is used. And so I will give you 2 examples which are not listed here. And so let me go. So basically I covered 2 portions already. And now I'm coming to MIR-DL, which is discussing about microRNA target prediction using machine learning, especially deep learning method. So what happens in the microRNA is that you have microRNA is biosynthesis I'm showing here, which we have reviewed in recently in RNomics with Malik Yusuf, and it is in the press. And so there you want to this is processing of a microRNA. Once microRNA is generated, microRNA had to hit to a target gene, and the target prediction is experimentally, all target prediction in human genome is a time-consuming process. And why microRNAs are important? MicroRNAs are important because if you are not knowing, it's involved in cellular proliferation, differentiation, angiogenesis, apoptosis, and that means it is involved in different diseases as well. And then there are 15 different tools over the period of 15 years. Almost 1 tool per year is created, and still this is the problem is not resolved, or still there is scope for newer development. So these are 15 tools and references we have given, and this year and the for species it is working, and the species and the features which is reviving, and then the approach. So you see many of them are using machine learning approach and SVM, but deep learning is hardly used yet. And now there are some examples, and we want to see what happens when you use deep learning approach to see this problem. And so we had a review also about it along with Pankaj. Pankaj and Alisha was working most of the time in this work, so her name will come again and again. And machine learning method we have characterized is we have wrote the review, and we were analyzing the data. The review is published, but the main paper is still due. So there are different tools. If you are aware, ProMIR, MIR predict. So there are prediction tools. There are tools which are MBA star, TARP MIR. They are for target prediction, and there are tools for microRNA annotation or microRNA functional annotation, you can say like this. So these are listed tools, and these are using different methods. So as you see, deep target is only one which is using deep learning until now, and then so there is scope for others to try, not just to us. So we have tried our a method, and this method. So whenever you want to method, you need to have a feature selection, right? So in microRNA, what can be the feature? MicroRNA sequence can be feature. As you see, the microRNA region is given there with the bulge and seed region, and microRNA mRNA binding is given. So structural RNA structure can be one feature, thermodynamics can be one feature, microRNA to mRNA binding can be one feature, and then feature will have subset of the feature. So what we did was we had 95 features selected, so I cannot put 95 of them, so I just pull it like a large table where you can have it. And what we did was we have features like bulge present in the seed with length 1 or length 6, and those were extracted using a source code developed by Alicia in Perl. And then sequence features, other sequence features, she wrote JavaScript for that, Java in Java, and then other features she extracted from different tools which is pre-existing. So we do not need to redo the, we do not need always to draw the wheel, because if the wheel is there, just use it. So the tools are there and giving us the input output, we just took it. So that these are the feature type of feature if you want to understand. And every time we go with zero knowledge and we try to build up a strategy of what can be the features. So if you are dealing with economy, you have different features, then you sit and note down what can be the features. Yeah. So so in our process, you also do hyperparameter optimization. So what that means, the maximum margin. So one which is a class, two classes are there, those classes will stay out if they are not falling in optimal linear line, if you try to understand. And that's in our tool we had 9 layers, and then these layers has values, and those were calculated. And then different tools, this tool which we call HDR is a new tool, and then we compared with the other tools and their hyperparameterization. And that's what and initially she thought how she will do it, but she built up everything, read the papers, and she could do it. And so anybody who want to do it actually just need to focus on it. So what I'm going to give an example. If you want to train with me, I always tell you that you have to train with me, then only I can hire you, because I don't know what is your skill. Education is not a set of the, as a person from super told you have to rebrand yourself, and you're not rebranding and saying it will not work. You simply asking for a job will not work. Everybody needs job, but if you can do machine learning or deep learning for me, I am ready to invest on you. But then you have to prove it that you will work and you will not run away. So why we developed this tool? Actually, we developed this tool to as I told you that it needed a more appropriate method for the putative target prediction. And so these are the tools, machine learning tools, and scanning based, sequence based tools. And then there's a limited deep learning tool. So therefore we needed to test it and to build it. So how we build it, we had divided our training data set, and this is how training data set is built up from Vienna package. And then we use scikit-learn. So I'm putting you here, if you're a student, you want to understand how the microRNA features will be extracted. These are the tools which you require, and you need to train yourself into these sequences. But much detail I'm coming into it. So what we did was we have you have mRNA, and in mRNA microRNA will be there, right, in a human genome. So you take transcripts, and you take the original transcript and identify the original microRNA by matching with the microRNA database. Okay, once you identify your microRNA database, you go to another side and generate negative data set. We just mutated 95% of the transcript randomly. So if there was a microRNA anywhere of 15-20, almost all positions are mutated. So it will lose microRNA features. And then we did interaction prediction. And then if something is positive is positive. Even from the negative, if we are hitting in good one, we removed it actually. And by this we bring entire data set of positive interaction and negative interaction, and generated a data set. And then from that data set we selected the feature, did a training, and that training data set, our method is following deep feed forward network. And that what deep feed forward network means, that you have one inner layer, two or more hidden layers. So you are going from one input to hidden, and hidden to back and forth, and then you make a your output will come. So these during our training we selected 19 features, and I will show you how out of 95 features. So when we run our different algorithms, you see that some algorithms have, like chi-square has a 10 features, and random logistic has a very less than 10 features. So what you need to do is this: you cannot have very high features, and you cannot have very low features. So optimal features were required, and we selected Lasso because it is picking up 19 features, and these are the features which is from 95 big table. Lasso selected 19 features, and those features are actually imbibed into further analysis. And so this is this is where this analysis was used, and Relo was used as a function. So this is the output prediction, and this is accuracy of ADHR. If you see here, it is close to 1 in all aspect, and these methods are okay to be your machine in comparison to other algorithm. It's not doing bad. And then you have if you collected the it collected the performance accuracy. It was 98 or 99% accuracy in different scales, and 95 Matthew correlation coefficient. So that shows we were on a correct path, and maybe if we need to improve something, we will think about it. And then it has to be AUC values has to be between 0 to 1, and closer to 1 the better it is. And then you see that AHDR is outperforming most of the tools which we have used. And then if you just talk about numbers and accuracy, many times it's confusing if you are not understanding a statistic. So what we did was we take the microRNA interaction with the pan-cancer driver genes. So pan-cancer driver gene was published from Ding Lab. Matthew Bailey was the first author in 2018, and it is one of the largest study of that time. So today, last February came another pan-cancer data. So before that, this was the largest study. And then we took all the important genes which were cancer genes from this data set, for an example, to test how all the methods are working in picking the mutation. So what we see is that TP53 is the most critical gene in cancer, and there are other genes as well. So you see that target scan is predicting 12 prediction interaction, and ADHR could pick 17 microRNA target sites in flanking to this gene. And these are red colors, and the color in the microRNA are showing these microRNAs are picked up by different picked up in between the different genes as well. And microRNAs are known to bind to different because it's at a small stretch, it's known to bind to different targets. So we wanted to compute in this how many targets are actually picked up, and how many of that are matching with other targets. So with this, we have developed a method. We are improvising it, and we see whether this can be used for on other small a small molecule microRNAs or not. So yeah, so this if you want to play with data, and if you want to play with data, you can invite, because the system was very easy. The codes are available in the GitHub. If you go to my GitHub, you will find there in deep learning microRNA. Shortly we are calling MIR-DL and naming. And then you can also see on the right side, it was primarily developed in Python and Jupyter Notebook, Java and Perl. So the percentage of development, how it was, so it was 90% Python. So Python is predominant tool. And so with this, I'm coming to a very short presentation, thing that which we want to develop a strategy. So in last talk, you hear images can be imbibed to brain analysis, and it is very well established. But there is a recent paper which has fascinated me. I'm fascinated about, and I think how we can implement it. That what they have done here, they authors, what they have done is that they predicted microRNA expression data by just scanning whole slide images and then matched it. So I will explain you how it is happening. So you have the images collected from Otking laboratory, is an image repository for cancer data, and then merged with they computed how by just simply seeing the visual specialization of the colors here, that how expression will look like, and then they correlated this with TCGA data, and if it's matching, they have counted. So basically they have done for 38 cancers types, and it's a very large study because the data is available in TCGA, and also the imaging data is available in aukinlab.org. So basically it's a hematoxylin auxin strain, and therefore called HE2RNA algorithm. And HE2RNA algorithm is really fascinating, because if you I'm a person who's deal with the sequences. I'm sick guy, so sick guy will understand image when you can correlate with the image with the image. So cell biologists will understand sequencing only when you correlate with the cells as cell biology images, right? So this can be a very comprehensive implication in biology of such, because there's just take the images, analyze the images, and then went to RNA seq and said, okay, there the expression is up or down. So you see the different colors in image, and actually once you have data, actually you can go to one hospital to different hospital and only need image then, because you already have your from image you can a library of image. So it's like a uh, DNA fingerprinting, or you can say cell fingerprinting. So this is very interesting, and I was thinking how we will work on it, and that's where a lot of students are asking they want to work. And if you can invite, if you are 50% prepared to analyze the images using machine learning, we will train you 50%, and then there is a way in 3-4 months you show that you work. And otherwise, a lot of people ask me, tomorrow I want to join to your lab, you pay me. That's not possible. That's not science is working, because the money is always limited. Nowhere it works. You cannot go to IT company and say, tomorrow I want to work. You have to show something which is sellable, that makes you better than others. And yeah, so over the COVID-19 and last 1 year, when I started my laboratory, I have some build up a team, and many of them are students, and some of them have finished their master thesis, some of them joined as a PhD. Primarily the work on deep MIR was done by Dr. Alisha Parveen, and she is now scientist in Germany, and she was working with my group from last 2 years in a transition period as well. And so several projects he developed, and then I want to mention the names of Gaurang and Vineet, who have done substantial work during COVID-19, and they were actually my COVID-19 volunteers. And once you prove it there that you can do as small projects, very good one. I'm very, very excited to not lose the person who is delivering very good results. And then we have a lot of students who have these the front row students were MSc student 6 months back. They joined and they really have performed well. And the last row also, so I'll tell you who has done what. Mayar is working as a doctor in Freiburg, and she has joined my laboratory to work on cancer genetics and genomics for a year, and she is developing her project. Then there are a lot of students who have worked on, if they have worked really well, they will get authorship. And then Dr. Mitra has worked on biostatistics of coronavirus, and with Yusuf Malik and me, we are trying to develop a strategy. Mitra is an associated member of my group currently, and he has also a faculty position in, he was looking in India, it was difficult, so he now has a position in China, but he's a very excellent scientist. So with that, these are the people. And then if you want to join, you can join. This smiley ones are one who want to join, and then that is possible. Only thing is that you have to tell me what you want to do and align with what our team's requirements. Some of the requirements I have shown, and some of these. So the 4 students have finished their master thesis written. Then their theses were submitted to different university. So notably, Madhu, Mita, and Namrata has done lichen genome analysis and try to find biosynthetic gene clusters. And they are now currently writing. They finished their master thesis. They were brilliant, and they are now writing their paper for a genomic journal. We will communicate. And then Rakesh has. Rakesh came to my laboratory to join as a master's student from Bihar. And initially, I could not understand what he was saying because probably English was the barrier. So I asked him to talk to me in Hindi, and I can understand. And then he said, I can work. Only thing is, the problem is my language. So I said, no, language is never a barrier. So let's do it. And he had done a SMAT4 variant analysis in different cancer type, and he's writing his paper. And Soumya has done work, but Soumya, after doing work, she has not contacted whether she want to write, but she had done good job. And 2 other fellows are actually stuck due to COVID-19. Their project, their university is not yet started. But yeah, Vinith was MSc student as well, but Vinith performed so well that before his master degree, I said, I'll pay you. I don't want to lose you. That's it. I am paying for him because he showed his brilliance. And then why should I lose a person who has a brilliance, who is hardworking and doing the task? Yeah, so that's what I'm saying. You prove me, we will try to find budget. Yeah, and then we have a Steffi who is now working with the Campegoda Hospital and joined PhD student with my lab and Campegoda Hospital. So with this, I will also like to thank Department of Biotechnology that they funded my own project on disease genomics platform development. And also in 1 year time, I received 1 grant from them on genomics and transcriptomics of Asian green mussel. And then ICMR, one of our grants were failed in February in ICMR, and suddenly ICMR had decided to fund it. So Indian medical research is has just accepted the grant. And then Institute of Bioinformatics for wonderful support, and it's a wonderful place to be, and wonderful infrastructure. The infrastructure I needed was there, and therefore, and Mahe for their support, and Mahe is also funding our human resource and whatever we need. So they, and omics box specifically, because they have partnered with me for the analysis, and they have provided their entire package to me for a year time. And also I'd like to thank you and like to thank eDot Solution, Cassid, Tribos Data Science, eDot and Cassid for their wonderful support. Whatever you seeing, the videos are running, whatever the eDot and Cassid joined together are helping us and using their employees and helping us during COVID-19. So they're very nice of them. And I'll ask them to, whenever there's a time, just give a small pinch what you do. So they are actually software companies. And Tribro Data Science is a one new company which want to train on data science and run data science in future in Bangalore. So I told why not to support them. And then the In Campus is an organization running events, and they advertise for marketing for the event. So they, in return of I talk for them for some minutes and give them a space. They are doing advertisement. And BioClue is a very good organization. Already we know, and it's a wonderful organization to be for networking. And International Journal of Computational Biology, the journal of the journal of that organization, BioClue's organization, and they will publish. I'm editor as well, and they will publish all the proceedings of the meeting there. And if you want to become part, just drop me a message. Look what I require in India Biosciences GitHub or Job Archive or ResearchGate, but look very carefully. Just do not send the CV without any email message. That will not work. An email, website of our institution, my email, my mobile phone number is given here. So please contact us when you need. Yeah, with this, I just finished my talk actually. I hope I'm on time.
Thanks, Dr. Kumar. Can we take a couple of questions?
Yeah, sure.
Yes, I have a question.
One question that,
yeah.
Sure, please go ahead.
Okay, so Abhishka, it was very nice talk. You have presented this study that using structure for macroeconomic prediction and also sequence features and so on. Did you try to figure out which most important features from this set or kind? You have 3 types of features, right?
Yeah.
Which is the most important type that actually is, becomes significant to build your own model? Or you just combine all the features together? Because I have another question.
Yeah, no. We have combined all 19 features. I will go back and tell you which one was predominant. I think sequence and structure was predominant features.
Because if you remember, I have done a couple of works. One of the study I did using structure and sequence, but recently we show that using just k-mer, that means the sequence information is enough to do the performance, to get the classification. Okay, so I wonder if it's also it would be interesting to use the same data and you just apply it in k-mer.
So let me see which sequence, which feature was used actually by us. 19 features, yeah. So there was energy. So basically, it's not sequence feature. 3' minimum energy, microRNA binding site actually, total GC length. So these are all sequence features. Yeah. So there are some features, but primarily it is sequence feature. Yeah, as you see here, I will give you exact detail of this each of these separately. Let's share the whole list, but there is some mixture we have. So we can still try with this data your algorithm. And I must tell you that we are working with Professor Yusuf Malik on the machine learning and microRNA. He has the expertise, and yes, definitely we'll look into it. So my today's presentation is only on machine learning and machine learning based things, not on the general things which we are doing in genomics, so that it is fitting to our requirement. And if you want to hear on genomics and all, we will be having a separate meeting later on, maybe 3 months later, or maybe next year.
Okay, that will be interesting. Also, the idea of mature microRNA. It's very interesting to try to figure out just using the sequence features from the k-mer features.
Yeah. So basically, we will sit together and build up. I was also reading about it, and then as you see that if I am able to convey some of the students that they can also do it. And so when I showed you the image analysis or whatever Python analysis is there, so in my team it is now must to have knowledge of Python. And if the person has shown other skills much better, and Python is not Python is a problem, I tell him to learn from a teacher on 90 working days, continuously practice for 2 hours, and then you will be good at it. So for analyzing data, Python is a critical tool, Python and R also. And so they are doing, otherwise I'm asking them not to join my lab, and if they make a commitment, then only they join. So once we have this strategy working, we need more and more people to join, and the data is not a limit. I totally agree with you, and we can try different algorithms. And we have some experience that 1 or 2 students started and then they run away with you. So I do not want to have a bad impression, right? And
Yeah.
on project side. So therefore, when a new student has come, I have told you to just keep eye on him, and then let him work, because the commitment is not there from. If I am not committed to work, I will be never able to work. Every day will have something else to do it. And then I do see a lot of people working in software and all. When they go office, they just go office, so that they can't do anything else, because otherwise they lose a job. And in research also, that self belief has to be there, that I'm in office, why should I pick up the call? And or why should I go somewhere else? So sometimes it's lacking, and I'm not telling all students do it. A lot of students do much better. So in my team now, new members are there. They're really wonderful, and I hope that they will sustain this for throughout. The better you do, the better your career. So if I am programming better, my career will be better. So
Yeah.
same applies to anybody
Yeah.
in any field.
Okay, thank you very much.
Thank you. So if there is a question in YouTube, no? Okay. So thank you.
Yes, Dr. Kumar, there is a question in chat box.
Yes. Did you try supervised machine classifier and come up with a SVM, or did you also try neural networks, sir? No, we tried deep learning in this data set actually, and we can discuss it. So data set is there. So if you want to try, so we have seen the SVM limitation of SVM by others. So therefore, we have not tried ourselves. So and primarily also the focus was on deep learning. So for that is the reason. Prash is from gene expression. Yeah. Yeah. So Prash is saying that non-coding RNA is also from expression. Yeah, sure. And that could be also picked up from image analysis, right? Thank you, Prash. So with this, we will just wind up, and you can raise question on Facebook, whichever social media, because now otherwise the speakers will be late. Yeah, we are already running late. So thank you, and I will stop my voice, yeah, and video also.
Thank you, Dr. Kumar. Thanks for a wonderful talk. Is Professor Puneet Goyal with us?
Yes, so Puneet Goyal is there.
Is my screen visible?
Yes. I'll just quickly introduce, and then the stage will be yours. Can everyone hear me?
Yes, we can hear you.
Okay. So we have our next speaker from Indian Institute of Technology, Ropar, Professor Puneet Goyal. He has received his PhD in electrical and computer engineering from Purdue University, USA. After a short stint in California, he returned back to India, and currently he is serving as an assistant professor. His main research interests include image processing, computer vision, deep learning, machine learning, security analytics, and assistive technologies. And today, Professor Goyal will be telling us something about deep learning-based efficient methods for segmentation and classification of burn images. Thanks for joining us, and the stage is all yours.
Okay, thanks for the opportunity. So I'll be, it's a privileged opportunity to present in front of you, and I will share about some of the works that we have been doing for the burn injuries and burn patients using the deep learning. And so I am a faculty member in the Department of Computer Science and Engineering here at IIT Ropar, and I'm also serving as an associate faculty with the Department for Biomedical Engineering here. So I'm just making it the full screen. So I will be going through the problem motivation and the introduction, following that with the dataset description and the other categories of the problems that we have catered to, the burn images body part classification, why that is important, and burn severity estimation and the burn region segmentation. Then another work which is not related to the deep learning based, but it is for the particular problem of the burns. So we call our project, this project that we are doing, it's self-motivated project. It's we call this project as burns. We want to reduce the number of burn injuries and get the people treated as soon as possible. So that's why we named the project as the burns. So the motivation. So burns is a very serious injury affecting large number of people, and even as per WHO, 180,000 people just lose their life, and many more become crippled forever. And you know that the suffering that they experience while living is going to be really torturous for them. And is this a problem for countries like India or developing countries? Not exactly. Even in UK, USA, there are a large number of burn-related deaths every year. So, of course, in our country, where we also there is a lot of people, the number of people per unit of area is very high. The density is very high. Population density is high, and we use a lot of the oils and other kinds of things, which makes it also make the people more prone to the burn injuries. So, number of injuries are quite large. So before proceeding further, let me tell you briefly about the skin anatomy. So there are 3 layers of the skin: epidermis, dermis, and hypodermis. So accordingly, the burns that affect, they may affect the different level of layers. And if they affect the innermost layer, the hypodermis layer, then it becomes a very severe burn. And burns is a special category of wounds. So it requires a special different treatment in terms of the treatment, also in terms of the deep learning applications also. So that's why we have just highlighted here that there are different kinds of wounds. Within the wounds also, we are just focusing on the burn injuries here. So these are some different causes of burns. So thermal burns, which we mostly encounter. And the thermal burns is a serious number in terms of the total number of burn injuries because of fire, because of hot liquids and something like that. Then there are sometimes friction burns, chemical burns, electrical burns, and the radiation burns as well. The number of cases for the radiation burns and the chemical burns are generally less, but there are sometimes, like people who construct roads, they go through the chemical burns and something like that. The sportsmen and other people sometimes go through the friction burns. And of course, the electrical related people, they face the electrical burns also because of shock and other treatments and so on. In terms of burn severity or the burn degree, there are 3 different types of burn severity. We label them first degree, second degree, third degree, and we can also give them the severity: low burn, moderate burn, and severe burn. As such, severity is a function of both the area, how much is the area of the burn body is burned, and how much is what is the degree of the burn. And now coming into the issue. So burns treatment, burn diagnosis require one to identify also the total burn body surface area as well, which we call TBSA. And for that particular thing, we need to perform the burn segmentation as well. And secondly, burn segmentation is also important for the degree classification. What is burn segmentation? Burn segmentation is basically given a total image of a particular person, including one skin and the area of the wounded region where the burn injury is occurring. We want to identify that region only, as you see here, which is actually burnt, only that portion. We want to identify how much this pixel, this area is contributing. Okay. So, and secondly, if we want to classify the different level of burns in different categories, whether one is low burn, moderate burn, or severe burn, then also it is presumed that the segmentation can help improve the quality of the burn severity and burn degree estimation methods also. So that brings us the need for the burn segmentation methods. So this is the current method that has been followed by the doctors in estimating the total body surface area which is burnt. So earlier, the long time back, the Palmer method used to be used, that 1% of your palm is basically, if the body of the person that is burnt is this much covered, there is 1%. So accordingly, we will compute, if you see in this slide here, that there is a map that the doctors are using in PGI here nearby. And it is a similar thing that is used in a lot of burns hospital. So the doctors will mark the markings and estimate that how much area of the person or the body is burnt, because the treatment requires giving the fluid to the patient, and then the how much fluid to be given to the patient is dependent upon the total body surface area. So that's why we need to know how much the body surface area is burnt. There is a total fluid requirement formula, Parkland formula, which given an input to the TBSA is given as an input. It gives you the values, how much the fluid requirement for the person is. But in order to estimate TBSA, the current methods that are used, they are very, very traditional methods. It's just hypothetical methods. There is not much progress. These are very old methods. If you see from 2004 onwards, these are the methods. The best method that has been used, which is considered nowadays the benchmark, is a Lund and Browder chart, which the doctors use. So there the body parts have different portions of the surface area percentage. So if your chest is one little bit, they count it as a 13%. Some doctors customize it by themselves, but more or less Lund Browder chart is a driving force for them. So there is no clear, accurate model that given a body patient image automatically can compute the burn surface area percentage. Even we are in the progress of that work, it is still a big challenge. So as I mentioned to you already, the reason for working on this burns problem and what are the different types of burns and what are the stages of the burn diagnosis involved. So now what is our motivation? There has been a work earlier recognizing the burn degrees and burn other aspects of the burn injuries using the color images. But that was a time when the machine learning and deep learning technologies were not advanced. Also at earlier time, not the good quality camera images were available. So earlier the laser Doppler imaging based modalities were considered good, but that have another challenge. Laser Doppler imaging infrastructure is very, very heavy. It's not easily portable. It's very highly costly. So even in USA, many hospitals cannot afford to have the LDI based imaging modality to classify the burns objectively. It's just subjective assessment. Doctors make a visual perception about it and they make some estimation and give you the treatment and so on. But as we understand that the subjective treatments have their own accuracy issues, so it is reported that only those subjective estimates are only 50 to 70% accurate. But now, given the advancement in the technology, in terms of the camera models that are available to the people in very easily, every phone has nowadays very good camera models within them embedded. And secondly, a lot of recent progress in the machine learning and data sciences, as this conference is all about. So we wanted to also explore that how these deep learning approaches can be efficiently utilized for this model, for this particular problem. So that was our motivation to explore. So there are some objectives that we identified in our problem. So I will now just briefly describe the data set of our problem, and that is a specific case for every other problems as well. That we prepared a data set from we have some collaboration with the doctors, but the complete IRB approvals is yet to be taken. We got duly supported with one of the expert surgeon who had more than 30 years of experience, and he has been working as a ex-HOD also in the Safdarjung Burns Hospital. So that really helped us a lot in this particular work. and we collected our data set of images. Because of the ethical issues and other issues, we did not want to delay the work. We collected from the internet manually and then segmented it in consultation with the doctors and categorize our data. So we have a large scale of data, which is around 604 images through the internet collections, and we gave them the different body parts also we identified. I will tell you why we came to the body parts. But the problem that we want to solve is that what is a burn degree, or you can say burn severity. And secondly, identifying the burn segmentation so that we can make automated burn diagnosis models. So these are the different body part classes, and these are realistic data, as these images are collected from internet, so they have the different illumination conditions and everything. So the burn images body part classification. So we wanted to just prepare a model which will help us identify whether it is a hand body part or other face body part or inner forearm or back, because these are the 4 body part areas that are most prone to the burn injuries. Why we are doing it? First of all, body part classification, because that is nothing to do with the burn diagnosis. The reason is our hypothesis that we explored, and we will show that we want to develop the efficient methods for burn images, body part burn images classification. The reason we identify that in general, the body skin properties are different at different body parts. We all know our skin property at the face, at the chin, at the chest part, at the stomach part, and other body parts is different. It's not exactly same, right? So the skin characteristics, the depth of the skin quality is completely different body parts. So different burn injuries reflect differently at different body parts. So their characteristics, the output when we capture through the color image or something like that, that will be slightly different for the different body parts, even though their severity level may be similar. Second thing is this, that when we are giving these deep learning models the images, okay, for a particular anything for classification, they also, if they are not customized, if they are not made specific to the different body parts, then try to understand that we have a limited data set. We don't have a tens of thousands of images or tens of thousands of data with us. So the challenge that it becomes is that the model has to generalize against a body part and able to make the prediction for the burn degree classification or the segmentation, which is very difficult in general. So the hand image is completely different looking compared to the face looking image or the chest looking image, and so on. If similarly, the hand with the fingers is very differently looking. So instead of generalizing a particular model that performs the burn degree classification, despite having the images of different types, we wanted to develop a burn images body part classification first, and accordingly perform the segmentation models. Accordingly have the segmentation models. Okay, so for this particular reason, we understood that we want to go for the customized deep learning models. Customized deep learning models is body part specific models. So our first task would be that given a burn image, make a automated classification for the burn. So we tried the existing simple deep learning models. There are very good literature in that particular sense. ResNet, VGG16, VGG19, these are the standard benchmark deep learning models for the classification that has been used. So we also tried that. So there we proposed another approach also. Now these already trained models, they have been trained on a different kind of a data set, all world images. So they do not again generalize for the body part problem. So, and our data set of the burn images was limited, even though we have a good number of 600 images, but still it is generally less compared to the number of images required for a deep learning model at times. So we enhance the improvement in our model using the known burn images. We took the known burn images and collected from the computer vision different problems. Known burn images data sets were available. We use that to train our model for body part classification. Then we fine tune our model. Deep learning models are fine tuned later for the burn images only. And we will see that what the accuracy of these models are. So these are some images, and burn images and the known burn images. So this is just the simple description of our data set, that how much images we have in the data set. Earlier, then we were performing the burn images body part classification. That time we had only very limited data set, only 150 images only in total. And we therefore enrich our data set using the non-burn images. So first, in our final approved model, we will train our model using the non-burn images and then fine tune it using the burn images, as you can see from this particular architecture. And the base model could be the any standard architecture that we can consider about ResNet, VG16, VG19, and so on. And then we perform the classification using softmax and so. So we also compared this accuracy with the standard machine learning models. Compared to the machine learning models, we find that the ResNet 50 based BI-BPC method was performing better. If we are just thinking about BI-BPC right now, and this is the class-wise accuracy for the different deep learning models, ResNet 50, VG16, VG19. Class-wise accuracy-wise, for some classes VG16 performed better, and for some classes the ResNet 50 based model performed better. But consistently, this is observed that deep learning models, despite these are the pre-trained, these are the fine-tuning, or you can say the transfer learning approach that we are using. So transfer learning approach along with the deep learning, in that sense, is better than the machine learning approaches straight away. And we further improvise the accuracy up to 90% using the additional data set of non-burn images. So please, I want to give you all also the insight. Many times a realistic problem in biology or any other area that you would be working, the deep learning, the data sciences are very much crippled by the fact of the limited data set. So in any case, sometimes in your problem, the data set is limited, but we also highlight from our example also, if you can enrich your model performance by using some similar data set of other domain, that also can help the improving the performance of your method. And we can see here also, so using the non-burn images improved the accuracy by almost 10% overall. So this is the class-wise accuracy of these models. For some particular cases, we did almost a threefold validation. So for the 3 different folds, we got this kind of accuracy. Now coming to the main problem part 1, which is burn severity assessment. So referring to the literature in a particular domain, this particular domain, there has been very limited work in exploring the deep learning approaches or machine learning approaches, which are burn images based. As I told you, the visual images based modality considered in the burn images, but it has not been that successful. So there has been very limited work in this particular domain, unfortunately. So earlier images were using just standard image processing based methods and using some psychophysical experiments with the consultation with the doctors and making some multidimensional scanning analysis, but they were tested again on very small limited data set. If you see, all these initial works were tested on around 100 images only. And in this particular work, they have tested, but they also used very standard simple models only and applied SVM and simple CNN ResNet only. And then recently there was another work, but they tested using only 23 burn images only, very limited data set again. So compared to these literatures, the performance that our models have performed is quite a bit. Our data set, it's not an exact formal comparison because different approaches are not easy to implement and repeat because they were psychophysical experiment based. But overall, if you see here, the data set, the number of the images in the different data set that has been considered and the accuracy values are been reported here. Now let me tell you the model approach that we are taking. As I mentioned, we are taking a 2-stage approach. First, we are performing the burn images body part classification. Then the second approach is burn severity estimation, or we can say burn severity assessment model. So first body part classification, then the customized deep learning model will be activated on that particular image to make a prediction. So given this hand image, so body part classification network predicted fortunately correctly that it is the hand. So accordingly, then the next time the burn severity assessment model of the hand only got activated, and that worked on this particular image and predicted that it is a moderate one. And this is also rightly the case. So again, we use the transfer learning approach only here. We have the pre-trained network, which we used and then fine-tuned it using the deep learning. But what we did was we did not just use the complete end-to-end, the softmax layer. we use the feature extraction extracted through the deep learning model, but applied SVM after that. We found that the results of the SVM compared to the softmax were better. So that's what we are doing it here in this particular. But the features were not handcrafted features. Features were coming through the deep learning model. So that was a slight difference. So first perform the body part classification here. The overall architecture is this. First perform the body part classification. After you get the classified image, then you feed it into the base model, which could be ResNet 50, VG16, VG19. Let us see how their performance is. And then that the output is 1000 crores because we are taking the pre-final layer output. So 1000 crores one size feature, and that feature vector is been then tuned to the, that been provided to the SVM classifier for the final prediction out of the severity, whether it is a low severe burn, mid moderate severe burn, or the severe burn. Just a minute. Okay, so this is a data set description of these particular experiments that we have performed. So we also compared these our customized BSAM. They are going to have the less number of images within the model itself. But the generic BSAM, although it is generic, but it has a leverage that is having a larger data set. So we also compare and present the results that even if a deep learning model which has been trained with a large number of images compared to the customized models which were trained using the less number of images, how does the performance compare for these particular models? So what we observe from this here, that the generic model we found, we can, I think, summarize the results here better. Okay, so the generic models was not performing that good compared to the body part specific models, and within the body part specific models also, if you consistently observe that the ResNet 50 BSAM ResNet 50 has been performing really the best consistently. If we can observe that the blue curves are here better for different metrics, either we take the accuracy, F1 measure, or the recall. We also prepared another data set that we collected later using, we purchased some images from the iStock. That's a website. From there we purchased some images. It costed quite a bit of amount of money, but finally we procured some images and tested the performance of our model against this additional set of images. And there also we consistently observed that the BP BSAM model with ResNet 50 is consistently performing the best given the different measures. So this is the overall approach again highlighted here, and we compare that the performance is comparatively better compared to the actual image. So these are the few examples that has been mentioned here, and these are comparatively results compared with the existing image processing based handcrafted feature based approaches applied with SVM. So ours compared to the standard image processing modalities, those Herlich features or local binary pattern or histogram or gradient, these are some of the feature extraction methods for the known to the image processing community. Compared to that, our model is around 20% better, which is deep learning based. The features are extracted using the deep learning, and the model accuracy was comparatively overall much better consistently on the different data sets. Here the results are on UBA data set. So these are the overall results that we are mentioning. So this work led to some publication. I am thankful for my PhD scholars who have contributed. Particularly Juhi has been leading this work, and the surgeon from the Southern Burns Hospital who has been assisting us a lot. So this led to some good publication in this particular domain. Then coming to the challenging the problem, the last part of this our talk, the segmentation. So as I told you, why we perform the segmentation, because it is important for the burn severity assessment and also for the primarily TBSA estimation. In TBSA estimation, we have to have the burn segmentation. We want to know how much area of the body is burned. So we want to develop the automated models for the TBSA estimation. So that's why it will require that just click an image. Can we compute the TBSA? So that will require first task is segmentation. And in medical imaging, the image segmentation is a very rich studied problem. But most of those works have been done on CT images or X-ray images and so on. So they cannot be directly used here. We have to think about the good segmentation approaches which have been used in the color images domain. So these are some existing methods that has been explored for the segmentation. And again, we find the similar limitation that earlier works have been really old and their accuracy is also limited. They tested on a very limited data set. Only recently some work have tested on the larger data set, and they propose very basic segmentation network model, SegNet, which is a very known 2017 work. SegNet, the latest work in this particular domain in the Bern community reports the SegNet to be the best performing model. And these are the models which have been used in the color imaging community and the computer vision community. You can say that they are considered different. So if you can see, this SegNet is very highly cited work, around more than 5,000 citations. But along with that, recently a lot of our progress have been made, which we wanted to explore and examine that how effective they are for the Bern imaging problem that we are studying, how this problem, Bern images, can be addressed using the computer vision knowledge. So we compared and explored these different models. So I'm just mentioning these different models, and they are really published in a very top conference in publication. That's why this ACCV, CVPR we are mentioning here. By the very number, the number of papers citing it, that tells that these are quite known and popular methods. Before proceeding further, I think I have to make some assumption here with the audience that they are familiar with the convolution. Okay, so because this concept, if you're not familiar with the convolution, you can ignore this. But in general, if you were to work in the deep learning or applied data sciences, particularly deep learning methods in your domain, then I suggest that everyone have to have the good understanding of the convolutions, because that is the vital step in the any deep learning model. Now, based on the simple, against the simple convolutions, the model that we tried was the Arcturus convolution base. What is Arcturus convolution? We gave it here some neighborhood gap. So here, if we have the consider that we have a here the Arcturus rate is something let's say 1. So in that particular Arcturus rate is let's say 6 or something. It can be varying. So here if we see Arcturus the Arcturus rate is 2. In that particular case, what I'm saying is R-1 neighbors I'm going to ignore before considering applying the convolution. So this is a 3x3 filter. It is not exactly applied for the 3x3 neighborhood image. Okay, we left the gap of R-1 neighbors before considering the another pixel. So if you see the domain, the view, the perception that I am capturing when I'm using the Arcturus rate 2 is actually 5x5. Okay. The window that I'm considering is 5x5 window before making some derivation. So higher the Arcturus rate, larger will be my scope, larger will be my field. So Arcturus convolutions have been reported to be really performing good in many deep learning applications. So we also explored the use of Arcturus convolutions. The deep learning based method, the Deep Lab, which is from the Google, if you see here, this from the Google Deep Lab based method, they're also based on Arcturus. They have considered the 6, 12, 18, but we consider the higher Arcturus rates as well in our model, and we found it to be performing really better. So this is the description of our model. Okay, so there are some technical details here, which for some of the audience maybe right now not that obvious, but it's okay. More the people get familiarized with these terms and the approaches, more it will be supportive in their professional career. That's why I will just go over a bit for this particular architecture. So again, as I mentioned, given the input image, I'm going to consider the standard pre-trained based model, which is having the multiple stages. So I will be combining the features that are obtained after stage 1 and 2, because what my assumption is that for the image, not the high level features only makes a difference, but the low level and mid level features also makes a difference. So I'm considering also the low level and mid level features, and I'm combining them, applying the convolution 1x1 convolution finally to get particular feature map. Along with it, I am also combining and fusing the final output of these features along with the stage 3 output, applying the Arcturus rate at different things. As I mentioned, Arcturus convolution, applying the Arcturus convolution different rates, 6, 12, 18, and 24. And whatever the feature output I'm going to get, I'm going to also merge them, combine them together and make another feature map, which is out2. And finally, I concatenate both the feature maps, out1 and out2, because that's how I'm going to get the high level semantic features also. And also I will be giving importance to the mid-level and the low-level features, which are important for my details. I combined them and applied some simple convolutional filters followed with the ReLU activation function, and then finally upsample it to perform my output prediction, which is my segmented output, whether the pixel is burnt or that particular pixel is of burn image, or that particular pixel is not a burn image body part. There is another method that we considered, which is not based on RTOS as such, but which is combining the spatial stream network and the contextual stream network, the low level features and the high level features in a different manner. So here we have a simple convolutional network, which is going to give me the spatial features. And again, I'm using the ResNet 101 backbone network as a backbone network and getting the weights of that particular different stages, out 1 and out 2. And then after some upsampling to get them the merge them together, followed with the fusion of this particular feature map obtained through this with the feature map obtained through this. And then finally, I have to, of course, apply the upsampling. Here, I'm using the global averaging pooling in this particular way. And here I am using finally the sigmoid function before I finally make the concatenation followed with the upsampling. Here you see the upsampling rate is very, very high. So I have to, because I am getting the 512 cross 512 image, and I have the features which are been reduced to 64 cross 64, I have to apply the high level upsampling in order to get the output prediction. So what we observed here is these are some mathematical aspects that we are considering the two losses, spatial loss and contextual loss. We are combining them together. And this is the description about the experimental result, that what kind of network data set we have considered in our experimental network. And these are the output ground truth data that we generated again, thankfully with the support from the surgeon. And these are some evaluation metrics based on which I will be presenting my results. So again, we observed that for our method, both method 1 and method 2 outperformed the standard state-of-the-art computer vision methods which are there for the color images. Most of these methods have been tried and tested for the other data sets. We will show the results for other data sets also, how we performed compared to that. But we found for the burn images data sets, certainly our performance was very huge, like almost 5% accuracy improvement you can observe here. And almost the segmentation approaches are more evaluated using the Jaccard index and mean, that is mean IOU also. So in that also we found a 70 to 12% improvement compared to the next best state-of-the-art method. So both the methods that we explored, of course, method 2 outperformed method 1, but both the methods outperformed the standard known methods. And these are some qualitative results. You can observe here, given this input image, our method is able to make the prediction that this is a burn area. The other methods were having a high recall because they were considering a lot of false positive area as well also in their assessment. Consistently you can observe these images are of different quality and very complicated nature, but our method consistently performed better in predictions. Of course, there are some tough cases, like this particular image is a very tough case. So our accuracy is still limited to 85%, 86%. But again, in general, our accuracy was around 90% or much more than the 90% in terms of the matrix correlation coefficient, which is one of the good benchmark assessment for binary classification problems. So these are some difficult cases. We can see here, this is very difficult case, the original ground truth, we are not able to much replicate. So there is some issue. This is the false positive coming, but you can understand this image illumination condition and the wrinkles here and wrinkles here and the illumination conditions makes it very difficult. Secondly, again, as I told you, face body part is very challenging, and the accuracy in the face body part is very limited for all the methods. The performance of other methods was even worse. It was less than 50%. Compared to that, our models was brighter, but still it is not that good, I would say. So in order to validate our model accuracy, we have also explored performed some ablation study, and we found that all the networks and different modalities are very much important. If we just choose a spatial stream network, our accuracy is limited to just 64% compared to 75.8% of the final network of the burns net. If we just consider just stage 4 or stage 3 and stage 4 only, still our accuracy is not that good. It's 72% at best. But if we combine the special stream network and also the multiple features that we are deriving from the base method, the multiple stages of the base method, both stage 3 and stage 4, then we are getting the better accuracy, as I shown you here. That here we are combining two stages. Here the stage 3 also and stage 4 also. Both the stages network is combined, stage 3 and stage 4, with the special stream network. The fusion has been done here, final fusion. Then that's why we are getting the best network performance. And it is to tell you the basic study that all these different networks played an important role in coming out with better results. So we also compared it with the standard computer vision segmentation data
Sorry
sets.
to interrupt. Sorry,
Yeah.
we are running out of time, so it would be just a polite.
Yeah. Okay. Sure. We started also quite late, I think, but it's okay. I will just wrap up with this particular topic. Okay. Is it okay? May I have 2 minutes?
Yeah. Sure. Thanks.
Okay. So we tried this performance of the model on the standard computer vision dataset that we considered. Again, we found the performance of our model is better. So you can see here these. So as you have been hearing about autonomous cars and so on, so that has been the driving force of the computer vision to perform the segmentation there. So mostly it's the scene images that they have performed. Okay. Now, why this burns? This is important. Okay. So I have to compute the total body surface area, which is the one wounded area divided by total body surface area. And I have no clue, given the image of the person, and particularly the total body surface area of the person. So this work we have been starting doing in collaboration with the National University of Singapore. There has been our collaborator there. So we are exploring there that how we can find the completely automation task for this particular work. So it's quite a lot of stages are there in this particular work. Because see, different people can take the images from different scale. So sometimes the image may have large number of your hand body part, but it's not necessary that that is your 100% of the image. So we have to have the scale estimation performed. And secondly, the lateral view, the 2D images do not consider the lateral views. So we have to prepare some automated app, which we can move around the body, the burn patient, so that we can get the better accuracy. So we are trying certain models with that particular approach. And now for the our model, the burn segmentation model, is thereby here going to make some contribution in finally getting the numerator term. For the denominator term, the total body surface area, there is a famous 3D scanner, and there is a very high quality 3D scanner in NUS with some particular industry where we got some good collaboration. So our task is, again, when the burn patient is going to come to a hospital, we understand that we may not have that 3D scanner with us. Okay. And these are not easily deployable also. Hardly there are any 3D body scanners, complete 3D body scanners in India. So what the approach that we are using is, again, we are using some data science approach of regression, basically. So we will be giving the input to the model as the person's face image, person's information, height, weight, palm image, gender, age. And for the limited data set, we prepare the complete model using the 3D modeling. And then the regression model for any particular patient will help us estimate the total body surface area as much as accurate we can. So that's what I would stop here. I think even there are a lot of applications as well. I will not go in the interest of the time. We have made some progress in the application as well also. So I will really like to thank the Juhi, who has been in the forefront in this particular, driving this project forefront. And as I mentioned, my surgeon collaborator from the Southern Burns Hospital and my departments for providing necessary resources. So any questions or comments?
Thank you, Professor Goyal. And in the interest of time, we'll skip the questions, and the students and others may ask over the email. And thanks again for a wonderful talk.
That's fine.
And I would like to thank all the speakers of this session. Here we end this session, and the next session will be chaired by Dr. Priyanka Singh.
Yes. Hello, everyone. So I welcome you all to the last session of the day. So the first speaker for this session is Professor Arijit Samal. He is a faculty member in the Computational Biology Group at the Institute of Mathematical Science, Chennai, India. He obtained PhD under Professor Sanjay Jain and Professor Shobhit Mahajan at the Department of Physics, University of Delhi, India. He works in the area of complex networks and computational system biology. His main focus has been the study of structure, dynamics, and evolution of metabolic and gene regulatory networks. He uses constraint-based metabolic modeling, Boolean modeling, and graph theoretic approaches to investigate genotype-phenotype relationship in metabolic and gene regulatory networks. He has also used Monte Carlo techniques to develop novel methods to sample networks with desired properties. He has also worked in the area of theoretical population genetics. So with this, I welcome Professor Arijit Samal, and I'm looking forward to your talk. So to you, Dr. Samal.
Am I audible, ma'am?
Yes, you were audible.
Um, so, so I request, ma'am, because things are running late. Dr. Singh, if you could just alert me when I am past 15 minutes.
Sure. I'll keep a track. No problem. Yeah.
Very much, ma'am. So let me just start by thanking the organizers, specifically Abhishek, for this kind invitation to present our work at this nice meeting. I've been following this meeting via YouTube actually since morning. Um, I also thank all the participants and everybody on the YouTube who has stayed late to hear me today. So the idea of today's talk is to basically present two pieces of work which I have done in my group. Uh, we do several things, but I would just like to present two stories today. Uh, I would try to present the first story as much as possible completely, and the second story, if I have time, I would try to present it as much as possible. So with that, I'll talk about some chemical spaces, and there is no machine learning or anything which is going to be in my talk, but we'll talk about some data curation and generation and analysis of highly curated data sets which are relevant for biology. So specifically, I would present in the first part of the talk on a chemical space, which is the phytochemical space of Indian medicinal plants, which is potentially beneficial to humankind. And then the second part of the talk, I don't know how much time will permit, I will talk about our work on a potentially harmful chemical space for humankind, which is the space of endocrine disrupting chemicals. So with that, let me just first go to the first part of the talk. As you know, plants use phytochemicals as chemical defense mechanisms against their competitors. We also know that plant extracts containing phytochemicals have been used to treat various human ailments for centuries. Also, many present-day drugs were initially isolated from plants. Prominent examples include the anti-malarial drug artemisinin and the anti-cancer drug paclitaxel. So, as we are aware, various systems of traditional medicine, including Indian and Chinese, use several of these medicinal plants in their formulations. So, if you look at traditional Indian medicine, they use several plants in their formulations to treat various ailments, but such information, including the information on the phytochemicals, which is the centerpiece of this work, is largely buried within books. So when we started this work, I argued that the non-digital nature of this information on the phytoconstituents of Indian medicinal plants limits its effective use towards modern drug discovery. I would also like to point out that in contrast to Indian herbs, there were many more comprehensive digital databases such as TCMID and TCM Mesh, which existed on the phytochemicals of Chinese herbs. So in short, what we did is that this is a work which we published in 2018, which took about a couple of years to accomplish. We have built the largest digital database, IMPACT, which spans 1,742 Indian medicinal plants, 9,596 phytochemicals, and almost 1,100 plus therapeutic uses. And we expect this platform and the database to facilitate computational approaches towards traditional knowledge-based drug discovery. IMPACT is accessible using any of the three domains which are mentioned below. I would take only 2 slides to walk you through the tedious 2-year process of generating the data set that we have as a database now in Impact. So the first step was to get a curated list of Indian medicinal plants. This is not simple because the plant names in the literature are quite synonymous. So we gathered a list of Indian medicinal plants from reports and books from Ministry of Ayush, CMAP, the CSR organizations reports, FRLHT organization in Bangalore, and several other resources. And we tried to build first a non-redundant list of Indian medicinal plants. And for this work, when we started, we had about roughly 5,000 Indian medicinal plants names for which we could search for phytochemicals. So this seems tedious, but this was not that difficult. The step 2 was the main step, which is we wanted to gather the information on the phytoconstituents of Indian medicinal plants. So without going into the detail, the tedious process involved sourcing the data from 3 sources. One, the books, which is highly non-digital in nature, so we had to digitize them. Second, through natural language processing. So we did text mining of PubMed. Specifically, the data was collected from more than 7,000 abstracts. And there were few existing databases in this area. So I should mention that my colleague Ganesh Baglar, who is now at IIIT Delhi, he had built a database of 963 phytochemicals for only 5 Indian medicinal plants called Phytochemica that existed before our work. And there was another work by Polur et al., which captured 1829 phytochemicals of 295 Ayurvedic Indian medicinal plants. Unfortunately, Polur et al. Never published a database, and the work did not contain any supplementary material. So we had to extract this information out from the NutriChem database, which claimed to have collected the data from Polur et al. So as you see, before our work, it was basically limited to around 1,000 or 2,000 chemicals, phytochemicals, and roughly less than 300 plants. So, however, one of the important, you know, centerpiece of our work is that we wanted to have chemicals or phytochemicals in our database with structures only, not just names. And when you go to the books or any resources, the chemical names or the phytochemical names are reported in a very synonymous fashion. So we did a very manually tedious job of removing this redundancy. That is, take the names which are reported in the literature and map them to standard chemical identifiers, including stereochemistry, etc. So, of course, if we found a name which could not be mapped to a chemical identifier, we don't report it on database. To give you an example, Phytochemica lists 963 phytochemicals, but only half of them, or slightly less than that, have a structure associated with it. So in our database, Phytochemica's data will be only half of it. So in this process, what we were able to do in our first round, which is available openly, is some 15,000 common names, which were mapped to 9596 standard chemical identifiers. So essentially, we have in our data space information for more than 1,700 Indian medicinal plants, which involves phytoconstituents that are 9596 in number. So once you have these 9596 phytochemicals with their 2D structures available, you can do a lot of chem informatics with it. You can gather the 2D and 3D structures. You can compute a lot of physicochemical properties. You can compute the drug likeness. You can predict the admit properties. You can also predict human target proteins using various resources. And all that information was gathered and put into our database. Furthermore, in addition to the information on the phytoconstituents of plants, we also focused on gathering the therapeutic uses of these Indian medicinal plants. As you know, the ethnobotanical or ethnopharmacology of these Indian medicinal plants is a controversial subject. So we didn't do any text mining here. We only resorted to gathering information from very standard books from Ayush Ministry. Again, this was collected from books which are recommended by Ayush Ministry, and we did some minor innovation here as well. Again, the reported therapeutic use can be very synonymous, so we wanted to remove the redundancy, and we mapped the therapeutic uses to disease ontology, mesh terms, and OMIM terms. So we finally have a non-redundant set of therapeutic uses for these plants as well. Finally, on the pilot scale, we also have data on the usage of these plants in different traditional medicinal formulations in Ayurveda, Siddha, etc. Here, we didn't want to reinvent the wheel. There is a large project by CSIR called the Traditional Knowledge Digital Library, where they have claimed to have digitized more than 2 lakh formulations, but only 1,200 of their formulations are openly available. So we just use these 1200 formulations from TKDL and link them to our plants if the formulations which are openly available in TKDL use any of the plants in our database. Again, we didn't want to reinvent the wheel, so we didn't link to formulations. And our primary interest was to gather the information on the phytochemicals of Indian medicinal plants. So what we have in our database finally is more than 1700 Indian medicinal plants, more than 9500 phytoconstituents, more than 27,000 plant phytochemical associations, more than 1100 therapeutic uses, more than 11,500 plant therapeutic use associations, 974 open formulations from TKDL, more than 5,000 plant formulation associations, and a number of phytochemical human protein associations from Stitch. So, okay, we created this data set, and what can we learn from this? This is just not a database paper, but we have extensively analyzed the data subsequently. The first question I asked is that what could be the potential drug-like space within impact? So I am not a chem informatician, but my student is trained in chem informatics. So I asked, right, let's take the wisdom of crowds approach. Let's put in several filters which are used in initial drug discovery pipeline to figure out which are potentially drug-like. So we use 6 different filters. Some of them are used by even pharma companies. And I asked, which are the molecules which pass all the filters that we have evaluated? So we found a set of 960 phytochemicals, which is roughly 10% of the phytochemicals in our database, to be potentially drug-like. What is interesting is that only 32 of these 960 are already FDA-approved drugs. And if you ask if any of these 960 phytochemicals have significant similarity, and the similarity is sort of evaluated based on the Tanimoto coefficient, which is a metric used to compute chemical similarity. So we use a similarity greater than or equal to 0.5 with any FDA-approved drug to say that if a phytochemical, which is drug-like, is similar to FDA-approved drug or not. We find that only 40% roughly of these 960 have any structural similarity to any FDA-approved drug. So another way to look at this drug-like space within impact would be to view it as a network. In this picture, each dot or each node in the network is actually a chemical, one of the 960. And we connect two chemicals if they are similar based on Tanimoto coefficient with a similarity greater than 0.5. So they are structurally similar. The color coding is as follows. A red phytochemical is one, which is similar to any FDA-approved drug. A green node is one, which is not similar to any of the FDA-approved drug. So you can see there are some disconnected clusters. So the space is highly chemically diverse. There are some clusters with many reds. So they have similarity to FDA-approved drugs, but there are also clusters which are purely green. So these are maybe potentially new diverse chemical spaces to explore. We are in subsequently analyzing this space and in a much more thorough fashion, and we will be coming out with more work on this analysis. A second question which is obvious is, and people asked, is that how does our chemical space compare with the chemical space of Chinese herbs? So for that, we took a TCM mesh database because, as you know, to do the comparison, you also need to map the chemical to structures to figure out which are the common chemicals and which are the ones which differ. So we took the TCM mesh database. Luckily, they had given some mapping to the chemicals to identifiers. So it again had roughly 10,000 plus phytochemicals covering more than 6,000 Chinese herbs. They had not done and evaluated the drug-like subspace. So we did the analysis, again using the same filters, wisdom of crowds, and we found that roughly again 10%, slightly less than 10% of the phytochemicals passed all the drug-like filters. So, so again, of the 972 in this database, which passed all the drug-like filters, we found that only 19 were FDA-approved drugs already. And if we compare the complete space of 10,000 chemicals plus chemicals here with our impact, we found that there is only a 25% overlap between the two, the phytochemicals in our database and the phytochemicals in the database on Chinese herbs. And similarly, if you compared also the drug-like space, again, the overlap is roughly 25%. So in summary, we find that the phytochemical space of both Chinese and Indian herbs provide extensive opportunity for drug discovery. The final analysis, I don't have time to go in detail, is basically a very involved analysis, but I request you to consult our paper. But I'll try to give you the main message through it because of the lack of time. So this is inspired by work by Stuart Schreiber, who is one of the main leaders of the Broad Institute's drug discovery program. What they have done in a back-to-back PNAS paper is analyze physicochemical properties of three compound sets they have collected. One is a set of commercial compounds, roughly 6,000 molecules, which are used again by pharma industry in the initial drug discovery pipeline. A second set, which is the diversity-oriented synthesis compounds, again roughly 6,000 compounds, and a selective set of natural products, which is roughly 2,500 molecules. For each of these molecules in their sets, they have computed two measures. One, which is known as stereochemical complexity, which is basically the fraction of carbon atoms in any molecule which is stereogenic. Second, which is the shape complexity, which is the fraction of carbon atoms that is sp3 hybridized. Note that these two measures are size independent. So they are defined in a way that they don't depend on the size of the molecules. So what they did is that they looked at the distribution of stereochemical complexity and shape complexity for these three sets: commercial compounds, diversity-oriented synthesis compounds, and natural products. And what they found is that both the stereochemical complexity and the shape complexity is higher in general for natural products in comparison to commercial compounds or diversity-oriented synthesis compounds. But they didn't end here, but they went ahead and did something very interesting. They tested each of these compounds with respect to a random assay of 100 proteins to check how many proteins can these compounds bind to typically. And what they find, and I'll try to summarize in one line, is that they find that natural products typically are very specific binders to protein targets. That is, they find that a natural product will bind to only one protein or two at best, but commercial compounds and diversity-oriented synthesis compounds can be promiscuous and can bind to many proteins. What this means is that if you design a drug molecule potentially around a natural product, it's going to be likely to be highly specific, and it won't have side effects because it will bind to the intended target, but will not bind to other targets, which may lead to side effects. So, yeah.
Dr. Shamil, 15 minutes are over.
Yeah, sure. I will speed up. I am also done with my first part of the talk.
Sure.
So, so what we did was that we basically cannot do the experiments, but we tried to compare these distribution of these measures for our library, the Chinese library of phytochemicals, and compared it with the result of Stuart Schreiber. So basically, I took these plots and plotted them as a box plot here as a reproduction. As you can see, both stereochemical complexity and the shape complexity of the natural products in our database, which have hardly any overlap with the natural products analyzed by them, is very similar. And if I was allowed to extrapolate based on the results by Stuart Schreiber's group, this may suggest that the phytochemicals in both IMPAT and also for the Chinese herbs are potentially likely to be also very specific, in their targeting of proteins and unlikely to be promiscuous. So this may have some advantage if you go for these phytochemicals to design drugs, lead molecules. So in summary, in the first part, what we have done is that we have three networks. We have medicinal plants and their phytochemicals, that is our centerpiece. Subsequently, we have made a lot of analysis of these phytochemicals. We find that there's a large drug-like space and also a large fraction of this drug-like space, which is not similar to any of the approved drug. There is only a 25% overlap between our set of phytochemicals and phytochemicals in the Chinese herb database. So there is a unique space of phytochemicals potentially for Indian herbs. And finally, with this analysis, this suggests that the phytochemicals of Indian medicinal plants or medicinal plants in general are likely to be very specific pointers of human proteins. We also have a data set of medicinal plants and their therapeutic uses and medicinal plants and their use in traditional Indian medicine formulations. But if you see, we have plant phytochemicals, we have plant therapeutic effect, but what we would like to do basically is to go from phytochemicals to target proteins to therapeutic effect. If we can link that, maybe that can basically give a mechanistic explanation of if potentially a plant or its plant extract could lead to therapeutic effect. So we expect that our platform, or we hope to provide a platform based on computational biology tools, which will aid in actually traditional Indian knowledge-based modern drug discovery. So with that, I would like to shift from the first piece of work to a second piece of work, from the potentially beneficial to potentially harmful, which is on endocrine disrupting chemicals. I'll try to speed up here because I won't have time for this extensive piece of work. But briefly, if you look at human body, roughly it has 100 trillion cells. And one of the challenges for the human body is to keep communicating between these different cells in the body. And broadly, at least, there are two systems through which the human cell communicates between these different cells. One, the nervous system. Second, the endocrine system. And this work concerns the endocrine system. If you look at the endocrine system, it is basically a system of chemical messengers or hormones enabling communication between different parts of the body. It basically consists of glands which secrete hormones into the circulatory system and thereby control distant organs in the body. So I have basically listed glands, hormones, and their effects in the body in this table. So the work concerns class of chemicals in our environment, which are known as endocrine disrupting chemicals or EDCs, which are defined by the World Health Organization as follows. An endocrine disruptor is an exogenous substance or a mixture that alters the functions of the endocrine system and consequently causes adverse effects in an intact organism or its progeny or subpopulations. These are dubbed as chemicals of emerging concern according to the World Health Organization. These EDCs are actually omnipresent in our environment. They are in air, soil, and water. Few of these EDCs are from natural sources, for example, in plant-based foods, while some of these EDCs are, of course, synthetic chemicals used in human man-made products, e.g. in pesticides, cosmetics, etc. So it is important to understand what is the mode of action by which these EDCs cause their effect. Basically, these EDCs interfere with the endocrine system in at least two different ways. They can either mimic the natural hormones and compete with them in binding to hormonal receptors, or they could indirectly change hormonal levels in our body by affecting the hormone metabolism. So if you are somehow able to disrupt the hormone secreted by these different glands, you can imagine that you can cause through these chemicals developmental disorders to fertility problems. So it is important to realize that there is a huge burden that the world is facing due to these EDCs. In the study in a Lancet Diabetes and Endocrinology last year, they have estimated that the disease burden, impact on healthcare, and loss of productivity due to EDCs is more than $300 billion in US annually and more than 160 billion euros in EU annually. So there was a report which was also commissioned last year and was published by the European Parliament, which found gaps in the definition of EDCs and the risk assessment. And the report actually led to a call by the European Union, which calls for a comprehensive framework to define EDCs and limit their use in various products used by the humankind in the next years. So actually this work which I'm briefly presenting is in the area of computational toxicology. If you look at toxicology, it is basically an international disciplinary field which studies adverse effects of chemical substances on living organisms. Traditionally, of course, toxicologists have used animal testing to determine the adverse effects, but due to ethical issues, animal testing and there are calls for alternative methods. So if you look at the EDCs, if you look at consumer products, there are at least 100,000 chemicals which are in commercial use. And most of them have not gone any toxicity testing. And computational toxicology is basically an emerging area which aims to build mathematical models to predict the adverse effects of these chemicals on living organisms. Just a second. Sorry. Sorry for the disruption. Basically, computational toxicology is an emerging area which aims to build mathematical models to predict the adverse effects of chemicals on living organisms. So there are 100,000 chemicals, but if you look at US EPA, it has only tested about 8,000 chemicals to date. So we need mathematical models to predict the adverse effects of various chemicals. So our basic goal is to build network-based predictive models for these endocrine disrupting chemicals. But before we could build such models, we need highly curated data sets for EDCs. So one thing to remember is that if your model is based on trash data, the results of the predictions are going to be even more trash. So you need a highly curated data set. So there were basically three features we wanted in a highly curated data set for EDCs, which we started building at. One, we needed a supporting evidence in humans or rodents for these EDCs, because there are some resources which exist, but they combine information from fish studies to environmental monitoring studies or computational predictions. But we wanted a data set which is exclusively based on experimental evidence in humans or rodents. Secondly, we wanted to compile the adverse effect of EDCs, and there is hardly any resource. Actually, there's no resource to date which compiles the system level perturbations caused by these EDCs in humans, etc. Finally, what is even more important is to also compile the dosage levels at which these EDCs cause their adverse effects upon exposure. So these second and third feature were not there. With these short-term goals, we decided to design a 4-stage workflow to identify EDCs with supporting evidence in humans or rodents. So I won't go into the detail. Again, work for 3 years. I will completely skip this in lack of time. We started with about 18,000 articles, which could potentially contain information on EDCs. A couple of my students read all the abstracts, figured out there are 14,000 papers out of the 18,000 which could contain information on EDCs. In the second stage, we read the papers to see which papers could have evidence only for humans and rodents, led to 3,300 articles. Subsequently, we retrieved the chemicals which were tested for endocrine disruption in humans or rodents in at least one of these articles, and we arrived at 1,626 chemicals which were tested in at least one of the papers. This was still, if that sounded daunting, but that was not that daunting. What we went ahead was we wanted to basically evaluate these experiments, published experiments, to see if they had really evidence to our satisfaction to call them as EDCs. So I would like to mention this consensus statement by Soleki et al., which was published subsequent to a big conference organized by the German Federal Institute in 2016, which was published here. They call for the weight of evidence evaluation for both adversity and mode of action to define these EDCs. So we evaluated that by reading each experiment and evaluating them. And I don't have details to show here due to lack of time, but we finally find that we find sufficient evidence for 686 chemicals, which we call as EDCs, based on 1,796 published studies. Since we have the structure and all, we could compile diverse information on each of these chemicals. So a question which emerges whenever I present this work is that can't these be done automated in some machine learning approach? So this is an example of a very simple paper which we have curated. So as you see, my student picks the chemical, the system, the test concentrations, the observed adverse effects to a controlled vocabulary, reproductive perturbations, the dosage at which the lowest dosage at which the effects are observed, and so on. So, and then we get a graph like this. So this requires really full text mining, understanding, interpreting the experiments. It cannot be done those so easily. So what we have finally is these 686 EDCs. We of course have classified them based on their environmental source, based on 7 broad categories, 48 subcategories. You see consumer products capture most of these EDCs. Second, we have a division of these or classification of these chemicals based on the type of the evidence we have. Is it only in vivo human, in vivo rodent, in vitro rodent, and such classification. Thirdly, we have done a chemical classification as well. We find that many of these are benzenoids basically. And fourthly, we have also categorized the EDCs based on their adverse effects, which is one of the important things we wanted to do. For example, the effects observed have been classified from, for example, developmental, neurological to reproductive to endocrine mediated cancers. So what we see is that in our data set, most of these chemicals have reproductive effects. So they can cause fertility issues, for example. So, okay, this is all captured in a data set which is accessible online. But after building this, we have done some analysis, which I want to quickly summarize. One, we have analyzed, for example, list of what are chemicals reported by US EPA as safer chemicals, and also a set which is FDA inactive ingredients. These are, for example, colorants used in medicine, which are considered to be safe. What we find is that 10 chemicals in the safer list are EDCs, 44 are in the FDA ingredient lists are EDCs. We also analyzed a set of 1328 chemicals which are banned in EU and used for use in cosmetics. We find 101 of them are EDCs. So I will, in the interest of time, quickly summarize by saying a couple of things. I don't have time to see this network analysis. We have also made a network of chemical similarity network of these EDCs. What we find is that there is a large cluster of EDCs which cluster together, and the color coding of each of these nodes, which is a EDC, is based on the number of benzene rings there are. So as you see, there is a large cluster which can be classified as follows, that they have at least one benzene ring, these chemicals. But there are also some isolated clusters, and there are also isolated nodes, which are not chemically similar to any of these other ADCs. This shows that the space is highly diverse, and we can actually tease out some interesting features. For example, why this cluster doesn't cluster with this, although they are both containing a single benzene ring. We find that this is basically chlorinated benzene ring, this is brominated benzene ring. So similarly, we have also looked at the network of EDCs and the gene or protein targets, and we have teased out this information from in vitro assays, which are published by the US EPA ToxCast experiments, and we have been able to make a network for 383 EDCs. So basically, you join two EDCs if they share common protein targets using the metric called Jaccard coefficient, and the network looks like this. So if you see, there is again a big large cluster. There are some isolated clusters and there are some isolated nodes. And here the nodes are the size of the node is based on the degree of it, and the two hubs are basically DDT, which is in pesticides, and octylphenol, which is a well-known carcinogen. But finally, we wanted to see if similarity of two chemicals in their structure leads to similarity in their targets proteins. And what we find is that there is hardly any correlation, and this actually projects a big challenge for the community, uh, which wants to design predictive models for ADCs based on QSAR, or quantitative structure activity relationship, which rely on chemical structure similarity for making predictions. So I don't have time, but I wanted to say that we have also evaluated using our positive data set various ADMET predictive tools for toxicity, and we find their predictions is not good based on our data set. In summary, we have done this work. I talk about a harmful space and a chemical space. Both works have received a lot of luckily press coverage in international and television media. But in the end, I just want to thank the people who did the work. The three people who led the work on phytochemicals, Vivek, Karthik, and both Kartiks, and the people who, Janani and BS Karthikeyan, who led the work on our endocrine disruptors. Both projects have received no funding, but my lab is well funded, and I'm grateful for that. I'm sorry I went over time, and if there is any time, if the chair permits, I will be happy to answer any questions. Thank you very much.
Thank you, Dr. Samal. Thank you for a nice talk. So there is time for a few quick questions. So kindly post your questions in the chat box.
So should I look at the chat box?
No, I'll read out those questions. So I think in the meantime, I have a very quick question. So you mentioned about Chinese herbs and Indian Ayurvedic plants. So there, I was wondering, like, there should be an overlap between these, the kind of like Chinese herbs and Indian medicinal plants. So there should be overlap between the two categories also, right?
Yeah, so the problem is that we have to curate their list of herbs, because the problem is that they don't give the herb names in a standard format.
Okay.
So that's the problem is of standardizing the data in such a way that you can easily compare these things. So that is one of the large scale efforts we are doing.
Okay.
People just list names as strings, but they don't. The same herb could be. There are even synonymous scientific names. That doesn't allow ready comparison. That's a big problem.
Okay, thank you. So if there are no questions, then we'll invite our next speaker.
Thank you.
Thank you, Samal. So the next talk is from our keynote speaker, Dr. Jessica A. Lesky-Sue. So Dr. Lesky-Sue is an associate professor in medicine at Brigham and Women's Hospital and Harvard Medical School, Boston, USA. She earned her doctoral degree in genetic epidemiology from Harvard School of Public Health and has spent the last 19 years focusing on the identification of genetic, genomic, and metabolomic determinants for complex diseases. The accumulation of these efforts has resulted in over 100 peer-reviewed original research manuscripts. Dr. Lesky-Sue's more recent work has focused on analytic and network approaches to integrate metabolomics and other omics data types with the end goal of making stride towards the precision medicine. She's currently the principal investigator and co-investigator on many grants focused on the integration of metabolomics and other omics data types for several diseases, including asthma, allergies, preeclampsia, muscular degeneration, cancers, and other complex diseases. Dr. Lesky-Sue currently serves in leadership capacities in a variety of consortiums, including acting as a chairman of the Consortium of Metabolomic Studies. So with this, I welcome Dr. Jessica, and I'm looking forward to your talk.
Thank you so much. I just want to check that you can hear me and see my screen.
Yes, the screen is visible and you're also audible.
Great. So yeah, today I'd like to talk to you specifically about metabolomics, what it is, and how we can utilize that with other multi-omics to understand disease, and particularly with the underlying idea of epidemiological study design. But to give us a little bit of context, I want us to go back in time. The year is 2003, and I want us to just think about how much has happened when it comes to multi-omics in the last 25 years or so. In 2003, we had Eric Lander and others, we had the Human Genome Project, and it took over 10 years and $3 billion to sequence just one human genome. And I remember sitting there in April, listening to him lecture, and this was certainly a key point in time in this field. It wasn't that much later that James Watson, who was the one who discovered the double helix, was able to get his entire genome sequenced for just $2 million. At that time, in 2007, the vision of where we were with genetics specifically can be best summarized in this phrase. That was in Technology Review in 2007. Scientists ultimately hope to bring down the cost of whole genome sequencing to less than $10,000, a target price that many people believe will be the turning point in genomic medicine. At that price, many could afford to have their genome sequenced, and doctors could use that data to give their patients more personalized medical advice. And this really encapsulated the idea of precision medicine and what it was in the year 2007, with really just the idea of genetics behind it. I want us to fast forward until today, when we can get our entire human genome sequenced with a little spit of saliva or a drop of blood, and we're now in the range of doing this for hundreds or low thousands of dollars for just one human genome. Along with that, what has developed when we think of genetics throughout that period of time, a lot of different individuals and researchers went to study human disease and identify genetic variants associated with human disease. And I like to say that throughout that 20 years, we developed what I call the omics standard. And that is, what do we use to actually identify and validate and substantiate a genetic effect? And through these genetic studies, we identified that we need large sample size with replication in independent cohorts that is robust to multiple comparison correction. And then we actually need to have further biological interrogation of our finding to actually see if the association with the specific genetic variant is causal or not, and where the causal variant is. So indeed now, when we look at where we are, and when we look at what's happened in this past 20 years, we are no longer thinking just in the context of genetics, but we're thinking in the context of the whole central dogma, that is, whether it be DNA, RNA, protein, or small molecules. And that is really where metabolomics now fits. Metabolomics is the study of small molecules in a specific biological fluid. And I really want to talk to you today about how we can utilize metabolomics, sometimes by itself and sometimes in conjunction with other omics, to really translate what we see with omic variants to a precision medicine framework. Now, why do we think about this with metabolomics, and how can that help us with underlying other omics? I think it's important for us to realize that the omics happen through a cascade of events that we learned about, early in our biology career, where we know DNA goes to RNA, goes to protein, goes to small molecules. And while we initially focused very much so on genetics, there are a few things I think that are important to realize as we go from the genome to the metabolome. And one is, while the genome is fixed, as we go down this cascade, we slowly enable environmental influence to be a part of the equation. So it is now not just our genome, but it is the combination of our genome and the environment together that is dictating what either our proteome or metabolome may look like. So when we think of precision medicine in 2020 versus what we thought about in 2007, we really think of this in a distinctly different way. Rather than just being genetics, we now have a much broader framework where we look at multiple omic variants in combination with clinical data and lifestyle/environment to generate more precise precision disease phenotypes. And what exactly do I mean by that? Well, initially, if you look on the slide all the way to the left, we would have a bunch of people who were diagnosed with a disease, let's say hypertension. And of those people, we would give them medicine. And some of those people would have a great effect from the medicine. Some may not have that great of an effect, and some may actually have a bad effect. We transitioned from that to what we call stratified medicine, where basically we created different subtypes, usually based on clinical data or underlying risk profiles, maybe one biomarker, but really not a lot. And then, utilized treatment options that could be specific to those generalized class of subgroups. In precision medicine, we look at a much more comprehensive framework. That is, we look at all of some of the underlying omics in combination with all these clinical data and potentially biomarkers to then inform the optimal treatment for each individual. Now, when I talk about some of the studies and what we've done with metabolomics and other omics, I also want to point and emphasize the importance of study design and how that can impact the inferences that we make from any given study. Especially the concept of large population-based studies and the idea that we can utilize multiple studies together to inform precision medicine. Primarily because that capitalizes on many things, from study design to multiple visits to a larger number of biological specimens and omic data and multiple cohorts that enables us to assess things like generalization. So in what I'm talking to you about today, I just want to give you an idea of what we have assessed and grouped together, even for what I'm showing you today, for specifically metabolomic data. And when we look at this, when we utilize multiple studies, multiple study types, multiple studies, multiple individuals, multiple time points, multiple types of biospecimens, the total number of samples that we're looking at is much larger, right? We go from any individual study that may only have 750 samples to 25,000 samples. So just by doing this, where we group cohorts together, we have the ability to much more robustly assess different associations. And in fact, when we look at these different types of study designs, different study designs enable us to infer different things. So the combination of multiple study designs together may enable us to look at things, and I will be speaking about asthma today, that may start from underlying asthma etiology to looking at the pharmacometabolomic impact, and finally to be able to translate that into clinical practice. So with that, I'm going to go over two general groups of studies that we've looked at that talk about different aspects of this. The first part of this is going to talk about a few cohorts and the idea of using longitudinal multi-omic data. Now, while the focus of this is on metabolomics, we also in this study and in my examples, I will illustrate how we've incorporated other omics, whether it be genetics, microbiomics, or outside exposures. The second part is going to really take utilizing that and looking at it in a framework where we can actually take metabolomics all the way from the discovery population through to clinical trials and finally to biobanks and precision medicine recommendations. So in starting with the first part of this study, where I'm going to be talking about really utilizing the multi-omic data, I'm going to be talking about a study called the Vitamin D Antenatal Asthma Reduction Trial. And this was a study that was born by my mentor, Scott Weiss. And I actually remember this vividly. We were in, we were at a wedding, a colleague's wedding, and he looked at me and he said, "Jessica, I really think that giving women vitamin D during pregnancy can reduce the risk of asthma in their offspring. And I want to do a study on this. And when we do it, I want to look at all sorts of things, multi-omics, environmental exposures, etc." Little did I know that that conversation would then progress to now what is 13 years of really exciting research. And why did he think this? Well, just to give a little bit of background, one of the things that's important to understand is vitamin D plays this critical role in the immune response that may reduce inflammation, which develops during asthma. And particularly the prenatal period was a time that seemed to be very susceptible to this, as that's when lungs grow. And so the influence during that time and making sure the supplementation was correct, he believed was key. So I'm going to give you a little overview of this study. This study started with women early in pregnancy, and they were randomized to either the standard dose of vitamin D or the standard dose plus 4,000 IU. And we've now followed those individuals. Those kids are now around 10 years old, and we've looked at the development of asthma over that time. Now, just to give you a summary, this is the initial publication of this study. And we actually had a sister cohort who did something very similar in Denmark. And when we looked at these studies, sure enough, what we do see is that there's a protective risk of vitamin D that reduces asthma by approximately 23%. And while in each one of these studies, they were quite different in several regards, especially ethnic makeup, socioeconomic class, this reduction in risk was profoundly similar. So what else did we do in this study? Well, one of the things we did was we generated a lot of multi-omic data throughout the course of pregnancy and the children. And I'm going to be focusing specifically on metabolomics. So I want to give you an idea of what I mean when I say we have generated comprehensive metabolomic data. These indicators down here show you the time points that we have generated either plasma metabolomics or stool metabolomics. You can see they've been generated in the mothers and in the children throughout the course of their life. The other thing we did was we partnered with our COPSAC friends in Denmark and actually looked at both of our studies together and asked the question, can we actually standardize time points that we generate these multi-omic data so that we can actually look at things together and replicate and validate results, which really is a key, I think, when it comes to omics and validating our initial findings. So I now want to talk to you about metabolomics and multi-omics. And when I say that, the first multi-omic I'm going to look at is multiple metabolomes. And that idea is this question: can we look at multiple metabolomes in the mother during pregnancy and understand anything about asthma risk in their children? So we had 2 time points, early in pregnancy and late in pregnancy, when we generated metabolomic data on the mothers. And we assessed asthma at age 3. Now these were done at age 3 because at that time, the kids were younger. So that's why the age is a little bit younger there. So I'm now going to show you what we saw. So the results for the metabolome, both in early and late pregnancy, we saw a bunch of different associations. Now, what you see here is this is the odds ratio of 1. Anything above is an increased risk. Anything below is a protective risk. And one thing I do want to point out is during pregnancy, we understand that embryogenesis has different influences at different times. So it isn't necessarily unusual to see different influences early in pregnancy and late in pregnancy. So for example, what we see is later in pregnancy, we see a protective effect for both cortisol and cortisone and some sphingolipids, which didn't surprise us because we know later in pregnancy is when the lungs develop. And we saw this increase in sphingomyelins, which are also related to these sphingolipids. We were able to replicate that in COPSAC. As I said, we try to replicate results. But the other thing we found that was quite interesting and seemed to have a consistent negative effect or protective effect on asthma was this one group of metabolites. And this particularly struck me because one of these metabolites was called theophylline. Now, for people who are in the field of asthma research, we know theophylline. We've heard of theophylline. And why is that? Well, for me, this struck a particular chord because I have asthma. And if I think back to my own life, back in the summer of 1984, this is a picture of me in the hospital where I was hospitalized several times for severe asthma. And what medicine was I taking at that time? Well, I was actually taking theophylline for asthma. Unfortunately, theophylline wasn't really cutting it, and I was in the hospital, as you see here, in an oxygen tent. Having said that, though, when I saw theophylline, and I saw that increased amounts of theophylline in these pregnant women were associated with reductions in risk of asthma, I was particularly intrigued. So, as I said, what do we know about theophylline? As something that has been known to treat asthma, it is a known bronchodilator. It increases the size of the bronchioles. It also has anti-inflammatory effects. But the other thing that was particularly intriguing about this is theophylline is not commonly used today. When you see that picture of me from 1984, it was used all the time then. But now this is really not something that is commonly used. So why are we seeing higher amounts of theophylline in pregnant women associated with reductions in risk of asthma? Well, when we looked at this further, we found something very interesting. And that is theophylline is a primary derivative of caffeine. So pregnant mothers, looking through this further, the question is, are they increasing their caffeine intake? Are they drinking coffee and having chocolate? And is that actually increasing these levels of theophylline? And then the other question was, why didn't we see caffeine? If we see theophylline in the metabolites, why aren't we seeing caffeine? So we identified all of the particular caffeine metabolites that we had in our data. And sure enough, we see them here. There's in various aspects from what we see in the liver to the gastrointestinal tract to what's in actually ground coffee. And sure enough, we see in this that several of our metabolites highlighted in red here all had a reduction in risk of asthma. Actually, it was these three, apologies. But then the question is, why was caffeine missing? Well, caffeine was actually missing because in our QC step, we took out metabolites that had a lot of missing data. And when you look at pregnant mothers, one of the recommendations is to not drink a lot of coffee. So this was missing in many of the pregnant mothers, and it was actually dropped from the analysis. However, when we looked at it again, when we brought it back in, we did see, again, this protection of risk of asthma of caffeine as well. In looking at this further, we actually see that this protection against asthma for coffee, it has been observed before. So in this study, I just want to highlight that looking at the multiple metabolomes, we can see how things change over time. And we can also then look at our biological hypothesis in the context of changing time. We also replicated these results in other cohorts, which fits in with our concept of generating robust associations. I'm now going to just very briefly give you an example of a few other multi-omics studies that we have done. Unfortunately, I can't go into the details because of time, but just to give you an idea of the other types of things we can look at. And in this study, one of my colleagues and mentees, Kathy Lee Sarwar, actually looked at multiple metabolomes. She looked at the plasma metabolome and the stool metabolome in conjunction with a microbiome, dietary intake, and looked at all of these together for risk of asthma. Using co-inertia analysis, what she discovered was that when we look at any of these things, food, the microbiota, diet, and plasma metabolites, a large portion of their effect was being mediated through the intestinal metabolome to cause asthma. And the mediation percentage was about 25%. And that, in fact, things that were highly influenced in this pathway was dietary intake, the intestinal microbiota impacting the intestinal metabolome, things like reduction of lack of breastfeeding associated with increased asthma, also impacting intestinal metabolites to cause asthma. So highlighting this interplay between other omics on one side, the intestinal metabolome as a mediator prior to disease. Another example of multi-omic data that followed very logically from this study was this question: can we look at genetics in conjunction with the metabolome in the context of this VDART study, where we also had a known exposure of vitamin D, and look at all three things with risk for asthma? And why was this particularly compelling? Well, it was particularly compelling because one of the underlying, the strongest gene actually for risk of childhood asthma is a gene called ORMDL3. And that is an ORMDL sphingolipid biosynthesis regulator 3. And this was particularly important because there had also been work implicating sphingolipids in the generation of asthma. Now, this gene sits at the very beginning of the sphingolipid pathway. So the question is, can we understand something about how this gene or genetic variants in this gene that are already associated with asthma impacts the subsequent metabolism of sphingolipids that also have been known to influence asthma? Furthermore, in our study, since we have vitamin D, can we infer or see and understand anything about how vitamin D may impact this? So looking at this in a sequential way, one of the first questions we asked is, can we look at the ORMDL3 risk genotype in association with sphingolipids? And in this case, this just exemplifies what we see over and over again, is that increasing numbers of the gene, of the allele, are associated with a decrease, in this case, in the sphingomyelins. There are other specific metabolites in this pathway, and you'll see that are associated with a slow increase. And that is actually due to the metabolic flux of the pathway. The other thing we saw very distinctly, and note in these results, we did this not only in our cohort, but also in our COPSAC friends cohort, who were able to replicate various aspects of this study, was this very pronounced interaction between this genetic variant and vitamin D. And that is with increasing numbers of the risk allele, when we see the effect of vitamin D on the genotype, we see that increased vitamin D and in combination with the non-risk alleles reduces the overall risk of asthma. So this then necessarily followed up with, can we actually understand the mechanism? And in looking at this further, what we were able to see is ORMDL3, as I said, sits at the beginning of this pathway and is responsible for potentially shutting off the whole pathway. What we see and what we're able to understand is if you don't have 2 copies of the risk variant, vitamin D supplementation will work and go through this pathway, increase sphingolipids, the good ones, and decrease asthma. However, if you had 2 copies of the risk allele, it didn't matter how much vitamin D you were given. This pathway, the ORMDL3 variant, would still shut off this pathway and really rendered any effect from vitamin D useless. We were also able to replicate this using human airway epithelial cells, where in this case, what we see is when we had increased amount of vitamin D, without the risk variant, we see that sphingolipid production goes up, which is what's responsible for the reduction in risk of asthma. However, in the overexpression of the bad allele, what we see is as vitamin D levels increase, we see a much more tempered relationship in the sphingolipid production, thereby further validating our result with more functional data. So in summary of this part, I want you to understand that we can utilize metabolomics in conjunction with these other omics and understand that sitting right before phenotype, at that critical stage, by linking the prior omics with this, it really enables us to incorporate outside influences like the environment in conjunction with prior omics to understand disease pathogenesis. And in all cases, we try to do this in a robust way where we are replicating results across multiple cohorts. So again, in this case, we see that metabolomics really can be a key identifier. These longitudinal measurements, identifying exposures, and replication are all key. So the next part of this study is going to take a shift and really ask this question: what can we utilize and understand about metabolomics and prevalent asthma in the context of potential precision medicine? And this study was done by a lot of people. Here's a bunch of members of my research team, where we basically utilized multiple studies in a discovery phase and then further studies in a translational precision medicine phase to study these data. The first step was taking 2 cohorts, one very large population-based cohort with almost 11,000 people, and look at associations with prevalent asthma. We then replicated that in a cohort that we actually generated within the Partners Healthcare Biobank. So we had a lot of comprehensive longitudinal data on these individuals, on their intake of different medicines over time, et cetera. So what did we see with that initial association with prevalent asthma? Well, this is just a negative log P plot of this. And what we actually see is this super high tail of highly significant results. And these results were significant in the discovery. And then also we saw some of these results in partners as well. But what could we actually see that was overlapping between the two, and how robust were those associations? So here you actually see the odds ratio in the discovery cohort and the odds ratio in the Partners Biobank. In the discovery cohort, we certainly see that these are tremendously robust associations with highly, highly significant p-values. These are all meeting Bonferroni adjustment. In the biobank, we also see significant associations, and these all were the FDR corrected associations, so showing replication in both. But I want to talk about this. When I looked at these data, there's a problem I noticed that really was striking. And that is when we looked at the direction of effect of these different steroids, it actually went contrary to what we thought. And that is, if you look at all these odds ratios, the odds ratios for all of these steroid metabolites were associated with a reduction in risk of asthma. Now, this struck me particularly because, if anything, in this study, you would think individuals who are asthmatic are taking steroids. So if anything, we'd expect to see an increase in these steroids due to taking oral steroids or inhaled steroids. So why was it that we were seeing this very consistent reduction in steroids in all these individuals? So we asked the question, is this a potential effect of oral steroids? And is this a potential effect of inhaled steroids? Now, looking across here, we see these are all different steroid metabolites, and this is stratified by controls, asthmatics off steroids, and asthmatics on steroids. Now, these are the inhaled steroids. We found really zero association with oral steroids, but when we looked at inhaled steroids, as you see here, those on inhaled steroids had these dramatic reductions in all the various steroid levels. So this begged the question, what could potentially be happening here? And one of the questions is, with inhaled steroid use, are we actually getting some level of subclinical adrenal suppression? And what do I mean by that? Often with inhaled steroids, the research to date has really said that for most often, the effect is primarily in the lung. But are there more systemic effects than we realize? That is, are these going into the system and being absorbed and causing the adrenals to reduce their production of many steroids? And the primary steroid that really encapsulates a lot of adrenal function is cortisol. Now, one issue with this is, if we think of it, inhaled steroids really didn't become common until the late 1980s or 1990s. So in many ways, the long-term effects of these inhaled steroids haven't been studied. So what would we need to do to study this? And this really got me thinking about this idea of steps towards clinical translation. How do we go from a discovery all the way through to a clinical recommendation? And that is, we look at a cohort, we find something, we validate it, we identify the cause. Then typically good science looks at this in the context of a randomized controlled trial. We quantify the clinical effects, evaluate the clinical impact, and then give a clinical recommendation. So thinking of this in that framework, we've already done the first two steps. The next question is, could we actually interrogate this in the context of a clinical trial? Fortunately for us, we had probably the best clinical trial in-house to look at this. And this was a clinical trial that randomized asthmatics to inhaled steroids and followed them for 4 years after randomization. Fortunate for us, we actually did have measures of cortisol and cortisone in this cohort. When we looked at these, one of the issues with this cohort is it was over the pubertal period. So we had to account for that. But sure enough, accounting for that, we see this dramatic reduction in both of these steroid levels throughout the course of the trial. Now, one of the issues here is that while all these data were generated using metabolomics, metabolomic data are not quantitative. That is, they are generated in a framework that the results are relative in any given study in which they're generated. So in order to really quantitate these in a robust way, we would have to utilize a better source for quantitation. And this brought us to this next step, which is this idea of, can we quantify this effect with clinical standards? And fortunately for us, we actually asked this question of, well, what is the clinical standard for looking at adrenal suppression? And in fact, the clinical standard for that is measuring cortisol in the context of an ACTH stim test. And we actually, in thinking about it, had these data in our biobank and electronic medical record system. So that is, we utilize the Partners Healthcare Biobank data and the electronic medical record data that has over 100,000 individuals and asked these questions. We asked, can we identify individuals who have taken that or had that cortisol stim test? And can we look at that by asthma status and by inhaled steroid use? So this requires collecting a lot of data, and it requires tremendously querying the electronic medical record, cleaning the different data, generating phenotypic algorithms, harmonizing the electronic medical record data. So this is certainly quite a complex process. And in the case of what we did here, we were able to then go from the qualitative measures we had before to quantitative measures. We were able to query the EMR for adrenal suppression tests with the cortisol stim test. We were able to harmonize those cortisol levels in about 2,200 people. Now, this is important because when we think about a lot of these tests, something like the cortisol stim test, it is often incredibly difficult to get large numbers of people who've had this at one time because it's not that common of a test. But when we're able to utilize electronic medical records and biobanks, it really enables us to do this in a large scope way. When we have over 100,000 individuals, 2% results in 2,000 tests, which is what we had here. Again, we were then stratified by asthma status and inhaled steroid. And we asked the question, can we observe clinically important differences between those not on inhaled steroids and those on inhaled steroids, and those particularly who were asthmatics on inhaled steroids compared to other groups. Now, one other thing that is important to understand is cortisol by nature is something that has a diurnal variation. That is, at different times of day, cortisol levels peak and reduce. So when looking at cortisol levels, it is incredibly important to take this into account. The good thing about what we had in the biobank is because they recognize this, every cortisol measure that is ever done, the timing is very precisely calculated. So what we were able to do is to look at these cortisol levels for controls with no ICS, controls with ICS, asthmatics with no ICS, and asthmatics on ICS. And what we see here is that the most profound reduction, stronger than any others, is this joint combination of asthmatics on ICS. Looking into this further, what we do see is that asthmatics as a group already have a reduction in these steroids, which is one of the things that makes them susceptible to being asthmatic. When we then put inhaled steroids on top of that, it makes the suppression even worse because there's already issues with steroid production in these individuals as a whole. And you see that really reflected here. The other thing that was really notable is during the time frame, and this is 4 a.m. Throughout the day to 4 a.m. Throughout this time period, the lowest measure or the lowest levels of these cortisol measures are actually during the time period when individuals are most susceptible to asthma attacks, making this even more important and imperative to address. So is this a clinically important drop? Yes, absolutely. So in looking at these steps towards clinical translation, can we evaluate the clinical impact? And the answer to that is yes. We also went further on and were able to understand through back calculating using the EMR that it turns out of asthmatics who are on inhaled steroids, and it really doesn't even matter how robust you are in that assessment, whether you say, oh, they've just been on inhaled steroids a shorter amount of time, or you're much more conservative and say, oh no, they had to be on steroids for 5 to 10 years. That pretty much a third of individuals who've been on steroids for really any period of length of time, who are also asthmatic, around 6 months or more, a third of those individuals go on to have clinically diagnosed amounts of adrenal suppression, making this certainly something that is relevant and important to be watching in more detail. So are we able to make a clinical recommendation? And I think that that clinical recommendation is certainly, and that clinical recommendation would be monitoring steroid levels if you're putting them on them, reducing the dosage or quantity as much as we can, and looking for signs of adrenal suppression before it gets to a point where it's irreversible. And another thing I want to point out when we look at these different groups, when we see this adrenal suppression, the other aspect that is very difficult about that is actually when the adrenal suppression becomes significant, it also makes these individuals at an increased risk of asthma exacerbations, highlighting another important point of why we would need to make this clinical recommendation. So in this case, I feel like we've gone through all of the steps from discovery to clinical recommendation. And to me, really, this is what I would identify as the steps and the translational omics standard that we need to be doing in all of our omics research. I want to end with just reflecting on a few things. You know, I showed you this picture earlier of me in 1984. 1987 was a game changer for myself. That was when I was put on inhaled steroids. And since 1987, I have been on inhaled steroids literally almost every day of my life. So while talking about the dangers of inhaled steroids, I want us to also recognize that there's impact and that this has had a dramatic improvement on overall health of asthmatics and changing lifestyle. However, I want us to think of this in two ways. While I most certainly have some level of clinical adrenal suppression that is probably irreversible, I've now actually personally myself reduced the amount I'm on as much as I can. But this actually is a picture of my son who actually turns 10 on Monday. And what you see here is he unfortunately has followed in my footsteps, and very early on, you see he had severe eczema, and to this day has significant amounts of asthma. Now, what stopped this incredibly bad eczema he had as a kid? And that was steroid cream. And what does help his lifestyle now? And one of the things is inhaled steroids that he does need to be on. But the question I have for you is, while my generation, we may not be able to reverse some type of damage that could have occurred, can we think of alternate strategies in treatment where we're reducing these inhaled steroids so that lifestyle and quality of life is optimized while minimizing the impact for long-term adrenal suppression? So just to summarize, I want us to just think about when we think of multi-omics, just the potential that metabolomics has to bringing us at the forefront of omics in translational and precision medicine research. You know, in this presentation, I've tried to show you how we can capitalize on things like study design, longitudinal data, generating quality metabolomic data, looking at multiple independent cohorts to maximize inference, linking this to biobanks and electronic medical records. Applying different novel approaches, or maybe approaches where we're just looking very focused, like we did in the sphingolipids, on one aspect of the multi-omics. And of course, working together through initiatives and consortiums. I'd like to end just by saying, you know, showing this slide, one of my 4 children is a rock climber, and we watched this movie Free Solo, which is about a rock climber climbing up El Capitan with no support. And you know, one bad movement and he would fall and die. I suggest to you today that in research, we need to take the exact opposite approach. And it's the combination of collaborating together, good statisticians, doctors, biochemists, postdocs, collaborations, and the next generation that is really going to make us successful. So I would just say in the context of successful research and multi-omic research, there is no such thing as no free solo. So with that, I'd just like to thank all the people who were involved in this research and the various sources of funding. Thank you.
Thank you, Dr. Jessica. It was an amazing talk. I think what better than you're inspired by your personal experience in science. So thank you so much. I'm very much inspired by your talk. So the talk is now open for questions. Kindly post your questions. So there is a question from Manas. He's asking, is there any side effect of long-term usage of inhalers or oral steroids?
So this is a good thing. So I think the key thing here, and we're actually just submitting this now to be published, there is definitely a very bad long-term impact of inhaled steroids that is largely gone unrecognized. And I think it's because a lot of these studies have only been done over very short periods of time. Whereas I think this effect is much more profound when you look over the period of years versus just a few months. Oral steroids, there absolutely are effects, and they're likely larger. However, the key difference in clinical treatment is that oral steroids in the context of asthma are most often given just in short bursts of 10 days, and then they're taken off that. And the effect of those 10 days of being on oral steroids cumulative over life is quite minimal compared to the potential ramifications of taking inhaled steroids, for example, in my case, of over 30 years. So that is, I think, the difference we see.
Okay. So actually, I have a very quick question about this theophylline. So you found it in the metabolomics of the pregnant women. So I was also quite puzzled because usually in pregnancy, you are not advised to have so much caffeine or chocolates or anything with. So is it like it's present in such a huge amount that it shows up significantly in the metabolomics?
So this is a very interesting point. And it is actually why initially caffeine was dropped from the analysis, because it was missing in a lot of the pregnant mothers. And actually one of the comical parts about this is our discovery cohort, our replication cohort, COPSAC, didn't have enough mothers who had taken caffeine to actually look at the result, because as you said, it is recommended to not take them.
Yes.
The VDAR cohort was a little bit more rebellious, and I think that's why there was more caffeine. But it certainly does beg the question of, is caffeine really as dangerous as what we may think in pregnancy if we're seeing these notably protective effects in development of asthma? And it certainly is an area of research that I am super interested and am following up, in further studies.
Yes, so basically a follow-up question onto that. So you also mentioned that it was recommended and that it was kind of dropped after a certain time window. So I was just wondering, so what would be your clinical recommendation basically based on this kind of analysis during pregnancy?
Yeah, so I think we need to be very careful about a clinical recommendation without more rigorous and well-designed studies on the topic. So I would say we still need to do something that is a much more robust design of a study to really assess it. Because the one thing I will say is we do know that coffee intake can be associated with, during pregnancy, can be associated with slightly higher rates of obesity. And so when interrogating the health benefits, we need to be looking at this in a much more holistic way. And so I think that has yet to be done. However, I will say, and one of the things I've realized is I absolutely think that a lot of asthmatics, even without knowing it, are self-medicating with caffeine and coffee. And they are not even cognizant that they're doing it to help them with breathing. We are actually seeing that very consistently in our research of individuals with asthma as well.
Okay, thank you, Dr. Jessica. So if there are no further questions, then I thank you again for your time. So
thanks.
Yes, so now Dr. Abhishek is going to take over the session and he's going to introduce next speakers. Hello, Dr. Abhishek.
Okay, yeah, thank you, Dr. Priyanka. So
yes,
let me introduce Dr. Sushant. It seems Abhishek is not around. So we have Professor Sushant Patel from University of North Carolina, Chapels, USA. Dr. Sushant is an assistant professor, and he has previously worked at Lingenberg Comprehensive Cancer Center and at University of Chicago Medical Center. He is a graduate from University of North Carolina at Charlotte with PhD in nanoscale science, biotechnology and bioinformatics track. His theme and MS in bioinformatics. Earlier in India, he pursued a Bachelor of Pharmaceutical Sciences from University of Mumbai. Dr. Patil's research interests range from bioinformatics, statistics, predictive analytics to cancer genetics, molecular pathology, and personalized diagnostics. So very vast areas of interest in the areas of intersection of these disciplines. His current projects are focused on development, evaluation, and application of genomics and related technologies for clinical and translational cancer research. So today he's going to talk about his recent work on So I'm going to talk about the NGS technologies in microsatellite instability detection. So and we look forward to his talk. So Professor Sushant, now stage is yours.
Thank you, Dr. Yadav. Can you hear me and can you see my screen?
Yeah, please.
Okay, good. So today I'll be talking about very specific yet very promising emerging area within the cancer genomics, which is microsatellite instability. Being working in the clinical laboratory, I have been leading the development efforts for the algorithm in the past few years, and that's what the focus of my talk will be today. So here is the brief outline of the talk. Initially, I will provide a background about what microsatellites are and why do we care about those. Then next, I will walk through the algorithm development efforts within the clinical laboratories that I have worked and the validation. And lastly, as an example, I'll share the results about studying MSI progression in endometrial carcinoma specimens. Okay, so just to familiarize ourselves with the vocabulary here, microsatellites are short repeating motifs. They could be mononucleotide, dinucleotide, or trinucleotide, or widely dispersed throughout the genome. And microsatellite instability basically means the somatic polymorphisms of microsatellite lengths caused by uncorrected slippage of DNA fragments during DNA replication in cell division. So I have shown here the gel image of two specimens. On the right hand side, we have microsatellite stable specimen, and on the left hand side, we have microsatellite unstable specimen. For each of the in each of the gel images, there are 5 microsatellite loci that are there, and for each of the loci, the samples of normal, N stands for normal and T stands for tumor, were run. So as you can see in the microsatellite stable sample on the right, there is hardly any change in the length of these 5 loci, whereas on the left hand sample you can see that the length is changing. I would like to also note here that even though in this example the length of the microsatellites in the tumor sample is decreasing, suggesting deletions, that may not be the case always. So MSI encompasses both insertions and deletions, and many times mixture of both. So perhaps the most important word in the MSI definition that I shown you in the previous slide is the uncorrected damage. So replication errors occur all the time, but microsatellite instability occurs when those errors go uncorrected. And the machinery which oversees the repair process is called as DNA mismatch repair gene, MMR gene. So this is composed of basically 4 proteins: MLH1, MSH2, MSH6, and PMS2. And whether a somatic or a epigenetic changes to any of those 4 proteins would disrupt the machinery, and that would ultimately lead to MSI, and which will ultimately lead to tumorigenesis. So microsatellite instability prevalence varies across the tumor type. On the bottom, you have I have shown you the percentage of MSI high cases across the various TCGA tumor types. It is highly prevalent in endometrial carcinoma, colorectal cancers, gastric cancer, and not so much so in these types of cancers. So the important question is, why do we care about microsatellite instability? So in May 2017, Keytruda, the chemical name is pembrolizumab, commonly known as Pembro, received an accelerated approval from the FDA for use in any unresectable or metastatic solid tumor with MSI. So that is in a way a historic event, because it became the first biomarker to be used for any solid tumor without a specific genetic feature. And MSI positive tumors are more susceptible to immune enhancing therapies. So ever since this, the interest in the MSI and the algorithms and the treatment options have certainly peaked. So as you can see, even though I have shown the data till 2018 over here, there is a sharp increase in the number of MSI related publications. Okay, so far, there are 2 gold standard ways of MSI detection in clinical laboratories. The first one being immunohistochemistry, or IHC. Here you perform IHC on the 4 proteins that I mentioned earlier, and majority of MSI high tumors will show loss of expression in at least one of the 4 MMR genes. But a word of caution over here is that the IHC results may sometimes be misleading, in the sense that IHC alone may not detect cases of Lynch syndrome because of the mutations that result in full-length yet dysfunctional protein. The second gold standard method for MSI detection is PCR-based fragment analysis. So the commercial kits use either 5 or 7 well-characterized loci. They amplify the loci with PCR, and then they analyze those amplified PCR products with capillary electrophoresis. So as an example, I have shown you capillary electrophoresis results of one of the tumor samples. So left column is normal, right column is tumor, upper row is MSI sample, and lower row is MSS sample. So as you can see in the lower row, in microsatellite stable sample, bearing this last loci, there is hardly any change between the normal and tumor samples in terms of length variations of the 5 loci that this assay is looking at. Whereas in case of MSI, you can see there is a peak splitting in about 3 of the 5 loci, and the percent threshold of calling MSI by this kit is 2 out of 5 loci. So that's why the upper sample is classified as microsatellite instable, whereas the lower one is called as microsatellite stable. Okay, but conceptually speaking, this kind of length polymorphism can also be detected by next generation sequencing. So here I have shown you IGV view of one of such homopolymer locus. In the normal control, you can see, yes, there is some variation in the normal sample in terms of length, but in the tumor sample, you can see this broad distribution of lengths for this particular loci. So basically, that's what most of the algorithms, most of the NGS based MSI calling algorithms attempt to do. They try to quantify these differences between normal sample and your tumor sample, and the way different algorithms do that makes them different. So these are some of the popular and well-cited algorithms for NGS-based MSI calling. I have divided into two categories: those that work on paired tumor normal samples and those that work on tumor-only samples. The problem of MSI calling using paired or matched tumor normal sample is relatively easy. And this list is very long. MSI sensor Mantis are some of the well cited ones. The MSI calling using tumor only samples is somewhat challenging. There exist few algorithms. For example, MSI sensor offers tumor only mode as well. There is MSyncs, which is one of the first algorithms published, and there is MSI colon care. In our internal validation and also in some of the published manuscripts, it has been shown that the results of these in tumor-only mode are somewhat unreliable. For example, this is a table that was published in the Mantis paper, and as you can see, for MSyncs, which was basically developed on colorectal cancer, the accuracy is reasonably good, but when they tested it on a endometrial cancer, the accuracy drops away sharply. So these are the things which prompted us to develop our own tumor-only MSI calling algorithm, because our lab at that time in University of Chicago used to have tumor-only based cancer sequencing panels, and many academic labs only use tumor-only sequencing rather than matched tumor normal samples just because of the cost effectiveness. So developing a reliable tumor-only MSI calling algorithm is important. So here are the results. So here are the results of MSI sensors tumor normal algorithm. MSI sensors algorithm in tumor normal mode. Here I have shown you the samples from TCG EC cohort, which is endometrial carcinoma. On y-axis, you have percentage of unstable loci, and on x-axis, there are box plots of 4 types of annotated samples: indeterminate, MSS, microsatellite low, L stands for low, and microsatellite high. For the purpose of comparison, we will be comparing microsatellite high samples versus microsatellite stable samples. This in-between category is somewhat challenging, and it's really beyond the scope of this talk to explain what's going on here. So as you can see, the performance is pretty acceptable for MSI sensors tumor normal mode. This is the recommended cutoff, which is 3.5% unstable loci. So barring these samples, the sensitivity is pretty good. Whereas the same algorithm in tumor-only mode, as you can see, even though it has a good specificity, but the sensitivity is not something that you desire in clinical diagnostic lab. So from now on, considering the shortcomings of the already published algorithms for tumor-only NGS-based MSI calling, I will discuss how did we develop our algorithm, and later I'll talk about the applications of it. So here, today I'm going to show you data from the TCGA UCEC cohort, which has around 500 cases, and the gold standard here is again the MSI status determined by capillary electrophoresis method. So 514 samples have that. Okay, so our in-house name for the algorithm is Atomic, which is another tumor-only microsatellite instability caller. We couldn't come up with a more catchy name, so suggestions are welcome. So basically, first we create a baseline using pool of normals. The thing to note here is that to use pool of normals sequenced on the same sequencing platform and also prepared using the same library preparation kit, because we have noticed that differences in the library preparation kit and the differences in the different sequencing platform lead to different profiles of errors. So to avoid that noise, ideally your pool of normal should be sequenced on the same platform and using the same library. So basically you cannot use all the homopolymer loci that are in the genome as your candidate loci for your MSI calling. So the main step of the algorithm is loci selection, and there are 3 steps in the loci selection. First is length-based filtering, second is depth-based filtering, and third is stability-based filtering. I'll talk in detail about each of these. Then once you have a candidate set of loci, you will compare at each loci the profile of your tumor sample against the pool of normal samples, and you will categorize each locus as stable and unstable. And from that, you will calculate the percentage of unstable loci for a particular tumor sample. And once you have that, and once you have the gold standard data, you determine the percentage of unstable loci threshold for MSI calling at a sample level. This is happening at a locus level, and this is happening at a sample level. So here is the first step of loci filtering, which is a length-based filtering. So as the length of microsatellite locus gets longer and longer, so does the probability of having it mutated. So here is a figure from this paper. As you can see, after about length 10 or so, the mutation frequency saturates. So that's why we have decided to work with loci of at least 10 bases, 10 nucleotides long or longer. So that leaves us around these many loci. Then the next step is a depth-based filtering. So you have to make sure that the loci that you are working with are sequenced with acceptable dates routinely. So we have used DP30 of greater than or equal to 20 at this microsatellite loci position in at least 90% of the sample. So that drastically reduces our number to 696 loci. And the last step of the loci filtering is stability-based filtering. Before I talk about that, this is the image from a paper published in 2016, which analyzed which loci are mutated, which microsatellite loci are limited across the different tumor types. And what they have observed that is that the microsatellite loci that are frequently mutated changes across the cancer type. So it is important to optimize your loci set for each of the cancer type that you are working with. So the question would be, how do you quantify the stability of a locus? And for that, we came up with what is called as D statistic. D here stands for distance. So here, for example, for a particular locus, here is the read length distribution or a homopolymer length distribution in your normal sample. And this is the homopolymer length distribution in your tumor sample. So basically what you do is at each of the different homopolymer length, you take the absolute differences in the normalized read counts between the two, and then you sum up all those differences. So because these are normalized, if the two distributions, here the blue represents normal and the orange represents the tumor sample, if there is absolutely no overlap between the two, then your D will be 2. And as the overlap increases, your D will tend to be 0. So D of 2 means highly unstable locus, and D of 0 means a stable locus. So coming back to our loci filtering for pool of normal, see, in a pool of normal, you don't want to, because this is going to serve as a reference for our tumor samples, you don't want to include loci which are hypervariable, meaning that you want to just retain the stable loci in normals. So here I have plotted for the 696 loci that we obtained from the previous step, the D scores. And on the x-axis we have mean D, and on the y-axis we have standard deviation of the D. So we have decided to only include the loci which are in this lower red box over here, with a mean D of less than 0.1 and standard deviation of D less than 0.075, because these are highly variable loci in the normal samples themselves, and so they won't serve as a good reference when comparing against the tumor samples. So that leaves us with 259 final set of loci. So at this stage, you have 259 loci, and using the pool of normal, you know for each of those 259 loci how the distribution looks like, meaning that you know for each of the 259 loci the mean D and the standard deviation of D. So at this point, you analyze your tumor sample the same way, and you calculate the D score for each locus at the in the tumor sample, and you compare the D score of your tumor sample, which I represented here in an example, to the mean D and the standard deviation of D in the baseline. And here the rule is really simple. If your D of a tumor is outside mean plus 3 standard deviation, then flag that locus as a stable, or if it is not, then flag it as an unstable. And so you do this kind of a thing for all the 259 loci that we have selected so far. And at the end, you calculate the percentage of unstable loci. So this is how our results look like for the same cohort that I have shown, for the same data set that I have shown before. So as you can see, compared to MSI census tumor only mode, this is much better with 100% specificity and nice sensitivity. So if you plot the ROC curve, this is what it looks like. So in the blue, you have MSI census tumor normal mode with accuracy of 0.97. These are the optimized thresholds, not their recommended thresholds. So we are comparing the best case scenario of each algorithm. MSI census tumor only mode is in the green, and atomic tumor only mode is in the red. So as you can see, the accuracy of our algorithm is compared to, or even better than the MSI census accuracy using tumor normal samples, and the accuracy numbers are consistent in our internal cross validation experiments. So When I developed this algorithm first during my previous tenure at University of Chicago, this algorithm has been incorporated in their clinical pipeline. And apart from just a clinical research, here I'm showing an example of the algorithm that goes beyond just the MSI calling. So a typical endometrial hyperplasia and endometrial intraepithelial neoplasia, AH or EIN, is a histopathologically distinct precursor to the endometrial carcinoma. So EIN is basically precursor of EC, and EIN gradually progresses to EC. Even though that is well known, the mechanistic details of this tumorogenesis process are not clear. So within this EIN EC cohort, we decided to work with a specific subset of samples which are MSI hypermutated. Okay, and the reason being that we believe that because you have paired samples, you can call it like early stage tumor and the actual tumor of MSI. This kind of a model provides a base system for studying MSI progression and the clonal evaluation and the relationship between the two during the neoplasm evolution. And these findings have been published in the Modern Pathology journal in the last year. So here are the results of not only MSI, but also the variant calling. So on the top, the bar plot represents the MSI calling results from atomic algorithm. For a subset of sample, we decided to validate our results with a classical gold standard PCR-based assay, and those two seem to be concordant. And in this table over here are the commonly associated driver genes or significantly mutated genes for endometrial cancer. So the takeaway message from this figure over here is that number one, as I said, NGS-based and PCR-based MSI calling methods are highly concordant. And secondly, 13 out of 17 samples that are represented over here shared at least one pathogenic somatic mutation in a driver gene. So this highlights probably the selective advantage conferred by these mutations in the process of neoplasm evaluation. So digging deep into MSI, so here are the percentage of unstable microsatellites between the two pairs. So as you can see, with the exception of probably one sample, the MSI severity or MSI instability increases from the precursor to the actual carcinoma. And going further in detail, I have shown here two cases out of these two. So here I have I have plotted the loci, the final set of loci for each of the two samples. On the x-axis, I have plotted d value of the precursor lesion, and on the y-axis, I have plotted the d value of the carcinoma. So for example, in this case, you can see a subset of the loci are becoming unstable and the other subset are pretty much remaining where they are. Whereas in this case, you can see there is a gradual increase in majority of the locus. So these two present perhaps two different perspectives in terms of microsatellite progression between precursor lesion and carcinoma. Lastly, we decided to increase the granularity of our analysis even more and decided to look at the genes which are associated with this microsatellite unstable loci. So in this plot over here, the pink represents the genes associated with microsatellite loci that were altered in both precursor as well as the final carcinoma. The blue represents the genes that were altered in the final carcinoma, and the greens represent the genes that were altered in the precursor only. And as you can see over here I have plotted the number of recurrent genes from the EC private group, and here I have plotted from the shared group. So frequently unstable microsatellites in EC are consistently located in driver genes. So FGFR2, PIK3R1, ATR, these are the common P10, KRAS are the common genes associated with endometrial carcinoma. So regulatory alteration of these genes may confirm selective growth advantages to the cancer cells. And whereas these genes, some of the other genes, we know very little about those. So further exploration of such genes is needed to determine whether such alterations are just the passenger events or they have broader implications in EC pathogenesis. So to end, I would like to just say that, I mean, NGS-based microsatellite testing offers a much broader perspective in terms of understanding the tumorigenesis. As I've shown in the last few slides, rather than just being able to determine the microsatellite state of a sample, it provides mechanistic details of which loci are altered and the probable implications of those loci in terms of the genes that they are regulating and the downstream effect, which were not possible with the earlier classical methods of MSI testing and are possible only because of the data that we have at our disposal right now. So with that, I would like to end my talk and thank my collaborators. So from University of Chicago Medical Center, I'd like to thank Dr. Siegel, Ritterhouse, and Kadri, and at my current place, I'm working with Joel and Jason. So I would like to end with that, and I'd be happy to take any questions if there are any. Thank you.
Yeah, thank you so much, Professor Sushant, for this such a wonderful talk. So maybe time for a couple of questions. So there is one question that amazing data, Mike. So from there is a question that does the data presented here are open access to other groups?
So we are in the process of publishing the software itself. So the software will be available on the GitHub soon. In terms of the data used, I have shown here just the TCGA UCAC data. So with the appropriate permissions, you would be able to download the TCGA UCAC data from the GDC website.
All right, so I hope that answered the question. So I would request also the participants to ask questions if there are any. All right, so maybe I can ask one question. So in this direction, how do you feel that the incorporation of data science in this direction could help in identifying or addressing those questions that you have presented? So maybe you can give some insights in that.
Sure. I mean, the data science itself, in my opinion, it's such a broad term. But one relatable example I can think of is they published a paper in Nature just last year about doing all the things I just talked about, that is determining the MSI status using just the histopathological slide images using deep learning methods. And the biggest advantage of that is that you get some prima facie information within just probably hours after after your FFP slide is ready, as opposed to maybe few weeks for the sequencing and then determining the MSI status. So I guess the avenues are endless, and we are certainly excited for the future.
Yeah, I think that's a very nice overview, I think. So with this, I think again I thank Professor Sushant to, and also we now are close to for today's session. We are almost at the closing stage now. We don't have any other talk for today. So let me, Dr. Abhishek, are you around? So maybe we can proceed for to summarize the day. It has been really a long day for us.
Let me come. Yeah, are you able to hear me?
Yeah, please.
Yeah, so basically it was a wonderful day. We learned a lot from starting the day with Sobha madam's talk, starting with the structural analysis coupled with the machine learning. And then we had a lot of image analysis in afternoon session. And then even I to tend to think about image analysis. So that's what is important in context of a meeting like this, that you learn a lot from each other. And then later in the evening we had a very nice talk from Jessica and Sushant. And unfortunately we started day with this few very long talks actually, because the talk for a general invitation is 20 minutes and 45 minutes for key lecture. So basically if the speaker is keeping so Asif could not present today because of this timing issue and time zone differences. So we are very sorry about that, and we are asking him to join tomorrow, or if he's not able to join, he will I have told him that meeting will extend to Sunday for one talk of his, and he's a key note lecture he will give actually. So I'm just expecting his talk to be there, and tomorrow, since this is Saturday, we will be able to accommodate him until, if it is possible, tomorrow or on Sunday. So other than that one talk we missed, otherwise everything went well. And then we are reporting today that tomorrow every speaker is keeping 20 minutes estimated times and 10 minutes for question, so they can go 22 minutes but not 42 minutes. And also just politely remind them before, as a chair I will remind them and politely before the start. And yeah, when it's a scientist, as scientists we all tend to tell the story, and then we get lost over the time, and that's natural. However, since the other speaker has to do something else, so we'll bring the Asif back hopefully tomorrow. And it was amazing learning from me, and starting from the director of IIT Jodhpur, and he himself is a machine learning person, and doing image analysis. I was surprised, and he out of his busy schedule, he came all the way to discuss with us. And then tomorrow we will have very wonderful talks, and then a set of new speakers. And in the morning, Institute of will start with the welcome session by Associate Director of Institute of Bioinformatics and Dr. Ravi, Professor Dr. Ravi, and he's an actual doctor. So MBBS doctor, and I see he's still practicing. So basically, we will have a good views from a clinical aspect from our associate director tomorrow, and then we will have a good proceeding. And over to Pankaj. You have to say something, Pankaj?
Yeah, so I would like to thank all the participants who eagerly joined and showed enthusiasm throughout the day. And I also would like to thank our chairs who help in sessioning taking care of the questions and introducing the speakers. So it was really helpful, and I think it was a quite fruitful day. We had tomorrow also we are going to have 4 or 5 speakers, and as Abhishek said, that we will try to also arrange the keynote that we missed from Dr. Asif. And yeah, so tomorrow we will also have two talks from industry, I think. So one will be from let me just check it once through the agenda. I think I think.
Yeah, tomorrow we have from industry.
Yeah, so 2 talks from industry, and I think 4 or 5 keynote lectures. So I hope with this, I think I hope that participants enjoyed the day, and hopefully we'll also see tomorrow most of them. So with this, I think thanks to all for like making this day successful. Yeah, so I think from my side that's it.
Yeah, so thank you very much. And tomorrow evening we may have to extend 1 hour, because if Asif is agreeing, we will give a talk, and then I will we write it to all speakers, those who are in late in from USA. For them, it's also good if they go say start at 10 or 11 a.m. on Saturday. They can sleep some more time. So we will discuss with them privately. And with this, I think we should we must. Neol, can you have a poster which is there with you, maybe for the sponsor? So we're really thankful for the supporters. So basically, not as sponsors, I can say that, because there is a support, because they're providing, all are providing support. So starting for me dot securities, which is a software solution company, they are providing us the web hosting infrastructure. And then Cassette is also supporting in a similar way, and others in helping us in training or participation on the data science, as well as a social media partner in campus, and society partner with BioClue, Clue, and Journal of International Journal of Computational Biology, where our proceedings will be published in the journal. And so we will ask um, all the proceeding authors to edit it one time more, so that it's if there is some typo or some mistake, we will take care of that during the publication, while we are depositing to the journal. So yeah, and also if anybody who is there, and we want this conference to continue on an annual basis. So in 2021, if you want to host it, maybe online.
Let's do that tomorrow, Abhishek.
No, because we have to keep it in mind so that they can propose tomorrow evening.
Yeah, sure. That's I think good idea that you mentioned.
Yeah, so basically we are proposing that somebody to take it over for next year, and so that then they have an idea, and then the two independent organization, or one, if you even one organization, then we'll help them. So yeah, so with this, we again thank everyone who are supporting our students, Dr. Pankaj, our all our chairs for today, Dr. Priyanka Singh, Dr. Neha, and in the initial session, I can't thank myself. Yeah, so it's my duty.
So yeah, and then all the speakers
I can thank you.
and the backdrop is back in backdrop. Everybody has organized the students who have organized the things to go smooth, and thanks to them. And then tomorrow we'll show everybody who all are involved once again to thank them, and thank you very much to all of you. And I think with this we will go offline slowly at YouTube, and then slowly from here as well. Thank you, all.
Thanks. Thanks, Mr.
So
Dr. Kumar.
I have a really pronunciation problem with your names.
Not an issue.
So I get alphabet vowels here and there. So, and it happens to even with my own name. So yeah. So with this, we'll stop in YouTube initially.
Yeah, I'm stopping it now.
Yeah. So you're going offline into YouTube.